ready, we can start. Lata madam, please begin the prayers. You unmute yourself, ma'am.
Sir, we will start with prayer song.
Yeah.
Okay.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रान्विता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिमी देवै सदा पूजिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा निःशेषजाड्यापहा.
Thank you, Vardhini. Hello. Hello.
Yes, you are audible, ma'am.
Okay, sir. Thank you. Good morning, everyone. Sir, am I audible, sir?
Yes, you are audible.
Yes. Respected patron-in-chief of this conference, Dr. E. Lakshminarayana, Principal, Giraj Government College, Nizamabad. Honorable Chief Guest of this conference, Sri Naveen Mittal Garu, Commissioner, Commissionerate of Collegiate Education, Telangana State. Respected our guest of honor, Professor Naseem, Registrar, Telangana University. Respected co-patrons, Professor B. Vidyavardhini, Head, Department of Botany, Telangana University. Professor M. Aruna, Department of Botany, Telangana University. Professor P. Kamlakar, Head, Department of Botany, University College of Science, Usmania University, Hyderabad. Srimati Leela Lakshma Reddy, President, Council for Green Revolution, Hyderabad. Dr. Sai Bhaskar Reddy Nakka, Founder, Member, and Vice President, Green Alliance for Conservation of Eastern Ghats, Hyderabad. I seize this opportunity to cordially welcome our distinguished resource persons of this international conference on plants and environment. Respected Dr. Maqbul Ahmad, Department of Botany, School of Sciences, Maulana Azad National University, Hyderabad. Dr. Sham Kumar Bhandari, USA. Dr. Chandrakant Mahendranathan, Eastern University, Sri Lanka. Professor Abdullah Muhammad Shohel, Department of Biotechnology, Genetic Engineering. Jahangir Nagar University, Sawar, Dhaka, Bangladesh. Professor Anjana Singh, Tribhuvan University, Nepal. Dr. Natarajan Amar Sen, Ukka Tarsadia University, Gujarat. Dr. Madhusudan Reddy, Department of Botany, Yogi Vemana University, Kadapa, Andhra Pradesh. C. Uma Maheshwar Reddy, Environmental Scientist, Council for Green Revolution. And the second convener of conference, Dr. Ahmad Abdul Alim Khan, Chairman, Board of Studies, Telangana University. Organizing Secretaries, Deepak Parmar, Assistant Professor of Botany, and P. Raviraj, Assistant Professor of Botany and Head Department of Biotechnology. This conference advisory committee members are Dr. Muhammad Abdul Rafiq, Vice Principal and Head Department of Chemistry. Dr. Y. Venu Prasad, Head Department of Economics and Controller of Examinations. Sri P.V.V. Satya Varaprasad, UGC and Autonomy Coordinator. Dr. N. Jyoti, Academic Coordinator. Dr. D. Adappa, IQAC Coordinator. Our college teaching and non-teaching staff members. Also, I cordially welcome professors, lecturers, research scholars, and students who are participating in this international conference from various universities, institutes of our country, and from other countries. Giriraj Government College was established in 1956 in Nizamabad town in Telangana state, India. It is named after the donor of the land, Sri Giriraj Malji Agarwal. This prestigious autonomous institution is affiliated to Telangana University, Nizamabad. Our college is spread in 29 acres of land in the heart of the town. This college has a big auditorium, lecture halls, well-equipped modern laboratories, Telangana skills and knowledge centers. Our college is the only institution in northern Telangana conferred with the status of college with potential for excellence by UGC. It is the most prestigious college and sought after by students for getting admission in the Telangana state. In this academic year, our college placed in second position in the online admission process for the undergraduate courses in Telangana state through DOST degree online services, Telangana. Only after City College, Hyderabad, which stood first. Our college offers more than 33 undergraduate courses with traditional and modern subjects and 9 postgraduate courses. Our college faculty are well qualified and experienced in research and teaching. They are committed and service oriented to teach and guide the students to fly to reach their aspirations. Many of our college students got jobs through campus placements. Many got selected in state and central government jobs, private organization. Some of the students are also pursuing their higher studies in IITs, central and state universities. Now, I would like to tell briefly about the Department of Botany. It is the founding department in the college and started functioning from beginning in 1956 itself with an undergraduate course, and later the Department of Botany was upgraded with postgraduate course of MSc Botany. Our department is very spacious, well-equipped labs, and presently with 10 faculty members. In the course of history, the department has developed botanical and rose gardens, greenhouse on the college campus. Our department has collaboration with Botany department of various other universities and research institutions such as Telangana University, Nizamabad, Usmania University, Hyderabad, Kakatiya University, Warangal, Regional Rice and Sugarcane Research Station, Agriculture Polytechnic, Rudrur, and Forest Department and Agriculture Department of Telangana State at Nizamabad. Particularly, I would like to mention that our department is intensively interactive with the Department of Botany, Telangana University, Nizamabad, and taking inputs and guidance from the learned professors for curricular and co-curricular activities to enrich teaching and research, including today's international conference. Our department has organized many national workshops and seminars funding by UGC, CERO, and autonomy grants for students, professionals, and lecturers to enhance their abilities. To name few seminars and workshops organized are National Seminar on Recent Trends in Plant Sciences in February 2013. National Seminar on Biodiversity Conservation Role of Students and Youth in December 2015. National Seminar on Importance of Ethnomedicinal Validation of Regional Flora and Fauna for Defending Value-Added Products in December 2017. Two-day workshop on Techniques in Biotechnology in collaboration with Primer Biotech, Hyderabad in February 2019. 4 days workshop on organic farming and techniques in soil and water testing in August 2019 in collaboration with Soil Testing Laboratory, Agriculture Department. Workshop on techniques in microscope in November 2019. Workshop on techniques in herbarium and pollen morphology in January 2020. Webinar on NIF genes in Klebsiella pneumoniae and Rhizobium in May 2020. Webinar on plant succession in May 2020. In addition, our department has organized webinars, online awareness programs, and quiz competition during the COVID-19 pandemic situation. Regarding research activities, our department has submitted recently 2 minor research projects sponsored by UGC CERO, Hyderabad. The NAC team, New Delhi, has complimented the Department of Botany as highly rated during their inspection and assessment visit about teaching and research activities. Regarding this international conference, we have invited expert resource persons. These scientists and professors are experts in their chosen field of subject who have a lot of experience in research, knowledge, and deep understanding. The participants can get new insights and findings related to the plants and environment in this international conference. Thank you for giving me this opportunity. Thank you.
యశ్వంత్ ప్రొఫెసర్ నసీం జాయిన్ అయ్యారు, తనని కోహోస్ట్ గా చేయి. యశ్వంత్,
Yes, sir.
ప్రొఫెసర్ నసీం జాయిన్ అయ్యారు,
Yes, sir.
తనని కోహోస్ట్ చేయి. షీ విల్ స్పీక్ అండ్ షీ విల్ లీడ్ ద మీటింగ్.
Done, sir.
Ma'am, you unmute yourself and start your screen, ma'am. Naseem, ma'am.
Hello.
Yeah, ma'am.
Thank you, Alim Khan, sir.
Now there will be addressed by Chief Guest of Honor, Professor Naseem, Telangana Institute Registrar. I would like to request ma'am to kindly give the address to the conference.
Our esteemed chief guest of today's international conference, Sri Naveen Mittal sir, Commissionerate of Collegiate Education, a distinguished guest of honor, Srimati K. Leela Lakshma Reddy Madam, President Council for Green Revolution, Hyderabad, patron of today's event, Dr. E. Lakshmi Narayana sir, Principal of Giraj Government College, Nizamabad, Rafiq sir, Vice Principal of Giraj Government College, co-patrons, Professor B. Vidyavardhini Madam, Head Department of Botany, Telangana University, Professor M. Aruna Madam, Principal of Education College Sarangpur, Professor P. Kamlakar sir, Head Department of Botany OU, Dr. Sri Bhaskar Reddy Nakka sir, Founder Member and Vice President of Green Alliance for Conservation of Eastern Ghats, eminent resource persons, Dr. Mahmool Ahmed sir from Manu, Dr. Shyam Kumar Bhandari sir from USA, Dr. Chandrakanta Madam from Sri Lanka, Professor Abdullah Muhammad Sohail sir from Bangladesh, Professor Anjana Singh Madam from Nepal, Dr. Natarajan sir from Gujarat, Dr. Madhusudan Reddy sir from Yogi Vemana University, Kadapa, Mr. C. Uma Maheshwar Reddy sir, Council of Green Revolution, Hyderabad, conveners of today's international conference on plant and environment, Dr. P. Lata Madam, Head Department of Botany, Government Giraj College, and Dr. Ahmed Abdul Halim Khan sir, Chairman, Board of Studies, Telangana University, faculty members, research scholars, and students of Botany Department of Giraj Government College, Telangana and Telangana University, delegates and participants of this event. A very good morning to all of you. The topic of this international conference is very important, that is plant and environment. I congratulate organizers, conveners, Dr. Lata Madam and Dr. Halim Khan sir for hosting and moderating this virtual international conference to enlighten, motivate, and inspire the young generation to undertake research in the concerned field, that is to make environment green. This event is specially organized by the Department of Botany, Giraj Government College in collaboration with Council of Green Revolution and Telangana University to provide multi-channel communication platform for researchers, scientists, and delegates to share experience, foster collaborations across an academic, and evaluate emerging technologies across the globe for a green environment. It is high time to go green for preserving our earth and to make the world a better place to live for the generations to come. As we all know, one of the most important contributions of plants to the environment is the production of oxygen and absorption of carbon dioxide, which is a greenhouse gas from the atmosphere during photosynthesis. With the increase in the quantity of greenhouse gases in the environment, its temperature increases due to greenhouse effect. Plants need water to live and grow, but high temperature will decrease the availability of water. Green plants are very important to the environment. It is estimated that 1 acre of trees in urban forests can provide enough oxygen for 8 people and remove 188 pounds of carbon dioxide from the air. With the increase in the number of industries, more quantity of greenhouse gases will be released in the atmosphere than that removed from the air. Deforestation will further increase carbon dioxide in the air, causing increase in temperature, that is global warming. Plants release water vapor into the air through their leaves during transpiration. It is a well-known fact. It is estimated that trees in tropical rainforest lower the temperature by 3.6 to 6.3 degree Fahrenheit. The Telangana state government flagship program, Telangana Haritaharam program, started by the efforts of TRS government since the state formed in 2014, has led to increase the green cover in Telangana by 3.67%. Telangana has recorded improvement in forest cover in all 4 categories. That is very dense forest, moderately dense forest, open forest, and scrub due to afforestation program investigate to plant and production of plants. The main reason for the increase in the forest cover in Telangana state are plantation and conservation activities, as well as improvement in interpretation. I'm sure that this virtual international conference will be an opportunity for all the participants to discuss about the emerging trends and novel techniques in going for green lifestyle to protect ourselves and future generations. Deliberations, discussion, and exchange of views would be constructive, useful, and motivating. I hope it will render fruitful results to our healthcare system during this pandemic period. I wish this event a grand success. I wish you all a very happy, healthy, peaceful, and successful long life. Thank you.
Thank you, madam.
Ma'am, principal sir unnara. Thank you, ma'am. Thank you so much. Very kind of you for giving such a beautiful words for the conference. Thank you so much, ma'am. Ma'am, Lata ma'am, can we have principal sir's address? Lata ma'am.
Hello, sir. Ready, sir. Principal sir, ready.
Yeah, he can start addressing.
Yes, sir.
Yashwant, can you make Chandrakanta madam as co-host?
Good morning to all. Today's international conference, chief guest of this great conference, Sri Navin Mintal Garu, Commissioner of Quality Education, and guest of honor, Professor Naseem, Registrar and Dean, Faculty of Social Sciences, and another guest of honor, K. Leela Lakshma Reddy, President, Council for Green Revolution, Hyderabad, and chief guest of another chief guest, Dr. P. Kamalakar, Professor, Head Department of Botany, University College of Science and Osman University, Hyderabad, and Dr. Sai Bhaskar Reddy Nakka, Founder, Member, Vice President, and Green Alliance for Conservation of Eastern Ghats Ecosystem, and all the faculty members from Botany of Telangana University, and HOD of Botany and convener of this international conference, Dr. Lata Madam Garu and Deepak Parmar Garu, and Biotechnology HOD, Dr. P. Ravaraj Garu, and all the Vice Principal Garu, Dr. Abdur Rafiq Garu, and all the HODs and coordinators of our college. So this is a great privilege to me to participate on an international conference. So on this auspicious occasion, let me congratulate the Department of Botany. So respected, honorable, all the participants of this international conference, let me say all the best to Department of Botany. So environment plays an important role in healthy living and existence of life on planet Earth, and plants are essential for the balance in environment. Plants are ultimate source of food and metabolic energy for all animals. Beside food, plant products vital to whom human include wood, fires, drugs, oils, etc. So it gives me immense pleasure that Department of Botany of my college is organizing an international conference on plants and environment. This conference is a truly international because eminent personalities and resource persons like from USA, Sri Lanka, Nepal, and Bangladesh are going to deliver their presentations. For this, I congratulate Department of Botany, Giraj Government College Nizamabad, and Department of Biotechnology, and other staff members of Botany Department. I'm very thankful to Council for Green Revolution, Hyderabad, for collaborating with Botany Department in organizing this conference. I am also very thankful to Department of Botany, Telangana University, Department of Botany. TU is always in forefront in supporting our colleagues in organizing seminars and workshops. I hope this international conference will provide right platform for the faculty and the research scholars from various institutions and colleges, and also for our students in acquiring the right knowledge on plants and environment. So on this auspicious occasion, let me congratulate our Department of Botany, and I may thanks to all the participants of this international conference. Thank you, one and all.
Thank you, sir.
Thank you, sir, for your kind words. As the time for technical session 1 starts, can we start technical session? Lata madam?
Sir.
Excuse me,
Vijay
sir.
madam.
This is Dr. Sai Bhaskar Reddy from Council for Green Revolution. I would like to say a few words. Is it okay?
Sorry, sir. Okay, sir, you continue, sir.
We are the partners. We are the
Yeah,
partners
yeah,
who are
yeah.
conducting
Because
this
of time
international.
short, I couldn't even introduce the council. Actually, in the valedictory, I'll be giving the briefing on council. Thank you. You continue, sir. Sorry, sir. Okay, continue.
Okay, let me briefly say about our organization, sir. This is a more than 10 years old organization, and today's international conference is very much relevant to our the kind of work that we had been doing practically in the grassroots level, and especially the plantation 3.9 million saplings we have already planted with the stake of the children, youth, and all kinds of civil society we have involved. And we also involved the universities, several universities, including today we are working with the Giriraj Government College or the Telangana University, and we have done several conferences too. And apart from that, Greens Alliance for Conservation of Eastern Ghats had been working on the conservation of this fragile ecosystem. And in this regard, now we are planning a conclave with the parliamentarians at Delhi. So to bring the consensus on this, and we have already documented all the challenges and brought in the form of a book. And we are also working on the sustainable development goals, and we are into capacity building, especially through the Earth Center for the Earth leadership. And today, I think this is one of the important international conferences happening, and I feel very happy that we have people across the globe, and definitely the kind of topics that are being dealt today, right from the micro aspects to macro aspects of the plants and the environment. And this will be very much useful for all the participants, especially the students who are attending today. And I welcome all of you, sir, and including the chief guest and everyone, as there is very little time now. And to make it short, thank you, and I wish best for this conference success, and also after the deliberations, hope we could bring some recommendations through the essence of this whole conference, and we could represent to the authorities, respective authorities, and share the information. Thank you very much. Thank you once and to everyone. Thank you very much, sir. Thank you.
Thank you, sir. We couldn't give introduction of council also. It is a time span because one speaker is waiting from USA. So now I would like to request Professor Vidyavardhini Madam to start technical session 1. Over to Vidyavardhini Madam.
Thank you, Dr. Halim Khan, sir, and a very good morning to everyone. So today we are starting the technical session 1 of the International Conference on Plants and Environment on Virtual World, organized by the Department of Botany, Giriraj Government College, and the Council for Green Revolution, Hyderabad, in collaboration with Telangana University, Department of Botany, Telangana University. So in the first technical session, we have 2 speakers, and the first speaker is Dr. Shyam Bhandari from University of Alabama, United States of America. So I want to just give a brief introduction of Dr. Shyam Kumar Bhandari. So he's a senior scientist and the Department of Pathology, University of Alabama at Birmingham, USA. And he has done from 2015 to 2018. He was a postdoctoral fellow in the Department of Pathology, same in Birmingham, the University of Alabama in USA. And from 2011 to 2014, he worked in the Institute of Physiological Chemistry and Pathobiology, University of Münster, Germany, under the supervision of Professor K. Grob. And from the year 2009 to 2011, he was a project fellow in the Center for Cellular Molecular Biology, CPMB at CCMB at Hyderabad, India, under Dr. B. Deshmukh. And he has completed his MSc Biotechnology in the year 2008. And he has many awards to his achievements, and a few of them are he has a travel award from American Society of Matrix Biology in the year 2018. He organized and chaired generated funding for the special interest session in the year 2018, and he has a travel award from University of Alabama. And that is Las Vegas, which was held in Las Vegas, USA from 14th October to 17th October 2018. He won the first prize for oral presentation at the UAB Postdoctoral Research Day 2018. And he won the John R. Durant Award for Excellence in Cancer Research in 2017, Outstanding Research Presentation Award in 2017, first prize for oral presentation in Postdoctoral Research Day 2017. He got selected as a session chair for Gordon Research Seminars in 2016. He received the pre-doctoral fellowship in 2011. And he has achieved as many awards as even as a student. He received the gold medal, the highest merit in life sciences, Usmania University. And from 1998 to 2002, he received merit scholarship from the Government of India. He has more than 13 poster and oral presentations and more than 16 research publications. He attended and delivered lectures in various workshops. With this, it gives me immense pleasure to welcome Dr. Shyam Bhandari to present his topic today. Over to you, sir.
Shyam,
Thank you very much,
Shyam,
Dr.
you can start sharing the screen, Shyam.
Hello.
It's possible అయితే లేదు అంటే పర్లేదు
Can you can I share the screen? Can you see the
Yeah, you are co-host, Shyam. You can share the screen. You start sharing the screen.
Okay, just a minute. I'm trying to share the screen, but somehow it's not working. Somehow I'm not able to share the screen. Just a minute. So can you see the screen now?
No.
No. Probably maybe I have to log in again. Maybe.
యశ్వంత్ శ్యామ్ సార్ కి కొంచెం co-host చేయవా?
Sorry, sir.
శ్యామ్ సార్ మళ్ళీ రీజాయిన్ అయ్యారు. Co-host చేయి.
Answer.
Shyam, can you try now? Dr. Shyam.
So can you see now?
No, screen not, you are visible, but your screen is not visible.
Somehow I shared the screen, but somehow it's not visible. I don't know why. So can you see now?
No. Shyam, you do one thing. Can you pass it to the WhatsApp? I can start sharing from here itself.
Yeah, just. Sir, I did send it to your Gmail.
I'm checking it. Wait 1 second. No, Shyam, I'm unable to see your mail in the inbox, and spam also I'm checking. It is not found in there.
So I did send it to your A Halim Khan, right? A Halim Khan at gmail.com.
I'm not able to see.
So it's A Halim Khan at gmail.com, right?
Yeah. That's right.
So is there any alternate email where I can send, or?
Wait, did you send to Yahoo or Gmail actually?
So just a minute. It's
if it is possible, you go with WhatsApp.
It's Yahoo. Yeah.
Yeah, you have sent to Yahoo. That is what you have. Yahoo mail. I know well.
So okay, what's your email now? So I'll send it to you.
Yeah, same. Just at the rate Gmail.
AAHALEEMKHAN at gmail.com.
Yeah.
So just. Yeah, so I send it to you now. I think you can see it now, right? The screen sharing.
Yes, sir.
Yeah, it is okay. Now you continue.
Okay, good. Hello, everyone. Sorry for the inconvenience. So actually, this talk is also kind of a special for me because I was the previous student from the Giriraj Government College. I'm always thankful to my teachers. Alim Khan sir also was my teacher who taught me botany, biotechnology, and microbiology. So with this, I'd like to start my topic. So basically, the topic for the today is molecular tools that drive scientific research and its basic principles and application of the plotting techniques. So if you see, somehow I'm not able to move the. Okay. Yeah. So what are the molecular tools that drive scientific research? So basically, there are some blotting techniques. These forms basics that drives all the scientific research. So without these techniques, any molecular biology laboratories that are working, it won't work properly. So basically, these are the techniques that are essential to drive all the scientific research. So what exactly are the blotting techniques? So what is the definition of blotting? So blotting is defined as in molecular biology terms, it defined as a transferring of DNA, RNA, or proteins from an electroporetic gel to a carrier. Then after transferring the DNA, RNA, or protein to the carrier, that is nitrocellulose or a PVDF membrane, and that membrane is subjected to further visualization techniques via hybridization of nucleic acids and chemiluminescence or some other methods which are used to develop for the visualization. So what is the application of this blotting techniques? So how exactly this is applicable, this blotting techniques? So as you can see, I can give you a best example. So if you want to find a hay, if you want to find a needle in the haystack, so what you will do? What you do is basically either you use the magnifying lens or you use some kind of magnet, right? So in order to find the needle. So in the same way, so our cell also has different proteins, DNA, and RNA. Let's say, suppose example, so if you wanted to find the specific DNA fragment, or if you wanted to find the specific RNA, or if you wanted to find the specific protein of interest in a cell or in a mixture of a protein, DNA, and RNA, so how do you find it? So basically, blotting is a technique which is used to visualize a specific fragment of DNA, RNA, or protein. So depending upon the type of blotting, so it's basically divided into the three types. So first one is southern blotting, and it is used to detect the DNA. And the second one is northern blotting, it is used to detect the RNA. And the western blotting, it is basically used to detect the protein. So these are the three different types of blotting. So first we'll go with the southern blotting. So southern blotting is a technique which is developed by Edward Southern in 1925. And as I have mentioned earlier, so it is basically used to find or detect the DNA fragments. Basically, this involves three steps. First step is digestion of DNA by restriction endonucleases and separation by agarose gel electrophoresis. And then second step involves the transfer of DNA fragments onto the nitrocellulose or a nylon membrane. And the third step is followed by hybridization with a probe, and then followed by a detection. So we'll go through the different steps now. So first step, as I have mentioned earlier, so first step is the digestion of DNA fragments. So first step, what you do is basically you'll take the DNA of your interest, so where you want to find the specific DNA fragment, and then you digest the DNA with the different restriction endonucleases. So what are restriction endonucleases? Restriction endonucleases or the enzymes or kind of a proteins, basically what they do is basically it will cleave the DNA at specific sites. As you can see here, this is a double-stranded DNA, and this EcoR1 is a restriction enzyme. Basically, it cleaves in between G and A. So basically, the DNA is very large, right? So if you want to detect a particular portion of the DNA, basically you have to digest it with the different enzymes. So those are called as restriction endonucleases. So then once the digestion is done, so then you proceed to the next step. So the next step is agarose gel electrophoresis. So what is agarose gel electrophoresis? So agarose by name itself is indicates that basically agarose is a polysaccharide, and it's a derivative of agar. And this agar is basically isolated from or kind of purified from the algae. And this agarose is basically used for separation of the DNA. So, and this main principle of this agarose gel electrophoresis is it separates the DNA based upon the charge. DNA is a negatively charged. The charge of the DNA is negative because, as we all know, it consists of the phosphate group. So because of that, it will have a negative group. So under the constant electric current, the DNA molecules, the DNA fragments will get separated and they move toward the positive charge. So as a result, the DNA fragments will be separated on the agarose gel. And basically, the separation mainly depends upon the agarose concentration. So agarose concentration, depending upon the agarose concentration, the pore size of the gel is maintained. So basically, if you use less agarose, then more pore size, and then more agarose, less pore size. So basically, depending upon this, the pore size is maintained. And also, depending upon the molecular size or molecular weight or molecular size of the DNA fragment, the DNA fragments will get separated, and also based on the molecular conformation. So this is how. So the first step is basically fragmentation of the DNA, and then second step is the separation of the different fragments by using the agarose gel electrophoresis. Then we proceed to the next step. So basically it is visualization. So as we all know, basically whatever you deal with, it's all invisible. Basically DNA, RNA, or protein, whatever you see, it's basically watery, right? So if you want to visualize the DNA and RNA, so you need some kind of a chemical or something which imparts some kind of a seeable nature to the DNA or RNA or protein. So in this DNA, so for DNA, what we use is ethidium bromide. So ethidium bromide is basically a chemical. What it does is basically it acts as an intercalating agent. So it basically stacks between the two, between the double-stranded DNA, and it binds to the DNA. It basically intercalates between the two DNA strands. And then once you illuminate that with a UV light source, so you can see, you can visualize the DNA fragments. That's how you visualize this DNA fragments. And basically this ethidium bromide is a carcinogen. So we should be very careful while using this ethidium bromide. And so that's why the new dyes have been developed. So these are called as cyber dyes. These are highly sensitive and less toxic. So now we have seen the DNA. So we have confirmed the DNA fragments by using the ethidium bromide, that is UV source, by using the UV source. Then we proceed to the next step. So in this step, what we do is basically, this is a blotting. So the blotting is a method where you transfer the DNA fragments in the gel to the membrane, onto the membrane. So basically the membrane that we use for DNA is a nitrocellulose membrane or a nylon membrane. So basically nitrocellulose membrane has a pore size. Basically it has a small pores, and due to the hydrophobic interaction, so what happens is that the DNA which is present in the agarose gel will go and bind to that nitrocellulose membrane. So for that, basically what you have to do is, the DNA which is present in the agarose gel is double-stranded. So you wanted to make the double-stranded DNA into the single-stranded form. So in order to make the double-stranded DNA into the single-stranded form, you treat that with the sodium hydroxide. Basically, you're treating it with the alkali agent. So as a result of which, the hydrogen bonds between the DNAs will get broken up. Between the two DNA strands will get broken, as a result of which it will form the single-stranded DNA, two separate single-stranded DNAs. So that is a step called as a denaturation step. And this single strands binds more efficiently to membrane. So then, so basically buffer is used to facilitate this transfer. And then basically this transfer of DNA fragments to the membrane basically relies on the capillary action. As I have mentioned earlier also, basically hydrophobic interactions are involved in the binding of the DNA to the nitrocellulose membrane. And in some cases, or vacuum or pressure, so different methods are used to aid this transfer. The faster and most efficient transfer is also achieved by means of electroblotter. So basically what you do is, basically you supply the electric current, as a result of which, due to the capillary action, due to the electric current, so from negative to positive charge, this DNA will move, as a result of which the DNA will get transferred to the membrane. So this electroblotting depending upon the fragment you wanted to transfer, it might take from 1 hour to 4 hours, in between 1 hour to 4 hours. So, and the next step after transferring is basically the hybridization. So the hybridization is a technique where you use a specifically radioactive labeled probe with the DNA fragment of your interest. So let's say if you wanted to detect a X fragment in the DNA, so you basically, what you do is you make the probe radio labeled to that specific fragment. And once that fragment are made, radio labeled fragments are made, basically what you do is you incubate that radio labeled probe with the your membrane, and then after that you'll do the washing steps to remove the unbound probes, and then followed by autoradiography. So what during the autoradiography, what happens is, as you are using the radioactive labeled probe, it will emit the light, as a result of which it will get transferred to the X-ray film, and there you can visualize the DNA fragment of your interest. So this is how this the southern blotting works. So basically what you do is you'll take the DNA, or you isolate the DNA from the source. The source can be either the plant cell, or the that is plant cell, eukaryotic cell, or any cells, like so the source can be anything. So where you wanted to look for a specific fragment of DNA, and then after that what you do is you isolate. After isolating, you'll fragment that DNA by using the restriction endonucleases, and then followed by the fragment separated by agarose gel electrophoresis, and then followed by the transfer. This is a blotting method. So in this blotting method, what you do is basically you use a filter paper and a gel, and on top of it, basically you place the membrane and again the stock of filter papers. And then in this case, you could put the weight, or in kind of electroblotting, you use the electric current. So what happens is, from down to up, from negative to positive, the current will move, as a result of which the DNA fragments will get transferred, or kind of blotted onto the membrane. So onto the membrane, basically these DNA fragments are blotted. And then once this blotted membrane is there, and then you basically you do the denaturation here, and then followed by adding the probe here, and then you do the autoradiography, and then you visualize it by using the autoradiography by using the X-ray film. So this is how the southern blotting works. And then we go to the north. Then what are these applications of this southern blotting? So basically, southern blotting is used for gene discovery, or it can be used for a mapping of the genome. So, or in order for the evolutionary studies. So it can be used, or it can be used for diagnostic and forensic studies. Let's say if you wanted to find a specific culprit, so you can use this technique to find the specific culprit, because every humans have a different DNA fragments, right? So depending upon that, so you can use this technique to find the culprit. So basically, this is a technique what they use in forensic studies. And then either you can study the deletions or insertions, or you can also study the point mutations. So if you want to know particular point mutations which occurred in the genome, then you can find it by using this southern blotting technique, or you can also apply this for using these structural rearrangements, or a presence of a particular bit of DNA in the sample. So these are the basic applications of the southern blotting. So then next we will go for the northern blotting. So northern blotting is also technique. Basically, it's used for transferring the RNA sequences, or kind of detecting the RNA sequences. This method is developed by James Alvine and James Stark. So RNA molecules have a much defined length, because DNA is very large, right? And RNA molecules are basically DNA is transcribed into the RNA, and these are of a specific size. So these are kind of a much smaller compared to that of the DNA. And then these RNA fragments are separated by using the same procedure, like gel electrophoresis. And then, so basic thing, what you have to care here is RNA is more susceptible for degradation. So you have to be very careful while isolating the RNA or doing the northern blotting technique. So always you have to do it in the cold conditions, and you have to wear the gloves. and you do not expose that RNA to your bare hands or anything bare surfaces, so that the RNA get degraded. And then RNA is also blotted onto the positively charged nylon membrane, and it's the same procedure. Basically, here you don't have to degrade the RNA with the restriction endonucleases, because these are already smaller fragments, so you don't have to degrade it with the restriction endonucleases. And then after that, basically what you do is you do the gel electrophoresis, and then after that you transfer that RNA onto the membrane, and then you detect it by the radiography. So this is how the northern blot works. So basically what you do is basically you take the sample of your interest, and then you isolate the RNA, and then after that you do the electrophoresis. So this is how you separate the RNA depending upon the size. And then after once this the RNA is separated, so then you transfer that RNA onto the nylon membrane, transfer onto the nylon membrane, as you can see here, and then you add the radio labeled probes. So which were these probes are exactly designed to the fragment what you want to know. So basically these probes are basically designed against the fragment of your interest, and these are radioactively labeled. So that's why you can use the autoradiography. So basically it involves the using of the X-ray film. So it has the radioactively labeled probes. So what happens is wherever this radioactive probe is bound, that area will get exposed to the X-ray, and it will emit the light. And as you can see it, you can visualize it as this kind of a black colored bands on the X-ray film. So this is how you detect whether the particular fragment of your interest is present in that sample or not. Then we go to the Western blotting. So basically, sorry, application. So what are the applications of this northern blotting? So this is a standard technique, basically, which is used to study the gene expression at different levels. And it is also used for detection of the mRNA transcript size, or study of RNA splicing, or study of RNA half-life. These are the different, some of the different applications of northern blotting. So then we go for the Western blotting. So Western blotting is a technique. Basically, it is used to detect the protein of interest from the mixture of a protein. So basically, if you want to detect a particular protein of interest in the mixture of proteins, so what you do is you basically use the Western blot. And this technique was developed by George Stark, Harry Tobin, and the name Western blot is introduced by Neil Burnett. So basic principle of a Western blot relies on the molecule of interest, that is a protein. And if you wanted to kind of know a particular protein of your interest is present in the mixture of many other similar molecules, this technique is used. And the molecule of interest is protein here, and the probe used basically in kind of a DNA and RNA, in kind of a southern blotting and northern blotting, you use the radioactive labeled probes, DNA probes, or complementary DNA probes, or you use the RNA probes. But in this case, in order to detect the protein, what you do is you basically use the antibodies. So what are antibodies? Antibodies are basically proteins raised against the protein of your interest. And these are basically used to detect the protein of your interest. So how exactly this Western blotting works? So Western blotting works in a different steps. So first step is tissue preparation. Second is SDS page electrophoresis. And third is transfer of that protein onto the PVDF membrane or nitrocellulose membrane, and followed by blocking, and followed by incubation with the primary antibody, and followed by detection using the secondary antibody, and detection using the different detection techniques. There are different detection techniques depending upon the secondary antibody you use. Either it can be the chemiluminescence, or a fluorescence, or a colored based detection. So first we'll go to the first step. So first step is tissue preparation. So the samples can be taken from the whole tissue or a cell culture sample, or you can take the bacteria or a virus or environmental samples. Any source can be used to detect the proteins. And basically what you do is, once you take that cell or a tissue, you add different buffers to it, and this buffers has salts and detergents, So what this salts and detergent does is basically it ruptured the cell membrane or a cell wall. So as a result of which all the proteins, DNA and everything, whatever present in the cell will come outside. Once it is come outside, basically it gets solubilized into that buffer. So this is how you basically use you do the tissue preparation for Western blotting. And always remember that the Western blotting, whenever you do any southern, northern or Western blotting, you have to do it under the cold conditions, because what happens is basically there are a lot of enzymes which can act on these proteins or DNA or RNA and degrade it before you detect it. So that's why you have to do all these procedures under the cold conditions. Then it is followed by SDS, polyacrylamide gel electrophoresis. So SDS is sodium dodecyl sulfate. So basically what you do is you'll take the protein of your interest, that protein solution which you have isolated in the first step, and then you add the loading buffer to it. So what happens is when you add the loading buffer, this loading buffer has basically SDS, that is sodium dodecyl sulfate. Sodium dodecyl sulfate is basically negatively charged. And what it does is basically it degrades or denatures the protein. And also it imparts a negative charge to the protein, because this is very important, because when you wanted to separate the proteins using the gel electrophoresis, you want your protein size to get separated based upon the molecular weight, not on the basis of the charge of the protein. Because if you see inside the cell, there are lots of proteins and they have the different charges depending upon the percentage of positive, negative or neutral amino acids. Depending upon that mixture of positive, negative, and depending upon the folding of the protein, the charge will be exposed on the surface of the protein. So basically you want to separate the protein basically on the purely depending upon the molecular weight, not on the charge. So that's why you use the SDS. So that is the role of SDS in this loading buffer. So basically it imparts a negatively charged to all the proteins. And then this SDS page involves the two phases. First one is a stacking gel and the second one is a resolving gel. Stacking gel is basically what it does is basically it stacks all the protein, and then resolving gel, it resolves all the protein. It separates all the protein based upon the size, purely depending upon the size of the protein, the proteins get separated on the SDS page electrophoresis. And then basically the principle of this SDS page electrophoresis is electrophoretic mobility of different proteins, because depending upon the size, the proteins will move in the electrophoretic SDS page gel. So the larger size proteins will move slowly and the smaller size protein will move faster. So depending upon their size, these proteins will get separated on the SDS page. So once that SDS page is done, so what you do is basically you transfer the proteins which are present on the SDS page onto the nitrocellulose membrane. So as I have mentioned earlier, nitrocellulose or a PVDF membrane, basically it has a charge. Basically it has a charge and it has a pores. So because of which, and it has a high binding capacity, because of which the protein can bind to these membranes. And basically the same procedure as I have explained in the southern blot. So that is a technique, that is a same procedure which is used. Basically you place the gel, and on top of the gel you place the membrane and then filter paper, and then you do the in case of the electrotransfer, you apply the current, and then the proteins will move. All the proteins are negatively charged here because of SDS, and they move toward the positive charge. So it moves from down to up. So the membrane is present on the top, so that the protein will get transferred onto the membrane. And then after that, you do the blocking. So basically, once the protein get transferred onto the membrane, you have to block that membrane. So blocking is a method where, so because as I have mentioned earlier, this nitrocellulose has a charge. So it basically binds to the proteins, all the proteins. So that's why when you add the primary antibody, that primary antibody can also go and bind to the places where the protein is not bound. So it results in the a kind of a non-specific detection of the proteins. To avoid that, the blocking is used. So in this blocking method, what you do is you use the protein, we use a dilute solution of protein. In this case, either it will be bovine serum albumin or non-fat milk. So this is used to block the spaces where the protein is not bound onto the membrane. So it basically, it prevents the non-specific binding of the primary antibody. So after blocking, so what you do is you have to wash it. So basically in detection technique, what you do is after blocking, what you do is basically you add the primary antibody. Primary antibody is a protein which is raised against the protein of your interest. Let's say if you wanted to detect a protein X or a protein, let's say I wanted to detect some of the DNA binding protein. So then I will use the primary antibody which is raised against that particular protein, and then I add it to this membrane. I incubate it with this membrane, and then after that it is followed by washing. So in this washing step, what it does is basically it removes the non-specific binding. Basically you use some detergent. So this detergent, what it does is it basically removes the unbound primary antibody. Then after that, it is followed by secondary antibody. The secondary antibody is the antibody, basically it detects the primary antibody and it is conjugated with the either the horseradish peroxidase or alkaline phosphatase or different tags which can be used for visualization. And once the secondary antibody is added, so you incubate it for 1 hour or so at 4 degrees, and then you wash it with the second using the this unbound secondary antibody can be washed by using the washing buffer. And then further you expose this membrane to the chemiluminescent substrate. So what happens is basically the secondary antibody has this alkaline phosphatase or horseradish peroxidase. It oxidizes this chemiluminescent substrate, as a result of which the light will be emitted and it will get exposed onto the membrane, onto the X-ray, as a result of which that can be visualized. So this is how this Western blotting works. And as you can see here, so basically you take the protein of your interest and you separate the proteins. And after that, what you do is basically you do the incubation part. So where you, this is your membrane with protein, and you add the primary antibody, and then followed by secondary antibody with the specific tag. And then you add the substrate. And once you add the substrate, this enzyme will degrade that substrate or oxidize that substrate, and it will release a signal. And that signal, basically what it does is this signal can be visualized by using the autoradiography on the X-ray film. So this is about the Western blotting. And basically there are lots of applications of this Western blotting. So one best example is it's basically used in medical diagnosis to detect the different antibodies against a bacteria or a virus in the serum. For example, if you want to detect the HIV, so what you do is you basically take the serum from the HIV infected patient, and then you do the either the Western blotting or ELISA to detect the whether the person is exposed to the HIV or not, or infected with the HIV or not. And this is useful for detection of the different proteins. For example, in kind of a prion diseases, where you can see whether these specific prion proteins are present in the person's serum or not. And then it is also used to determine the size and amount of protein in a given sample. So these are some of the applications of this Western blotting. And thank you very much. So sorry once again for the delay, and thank you one and all.
Thank you, Shan. Thank you very much. Thank you for accepting the invitation and giving such a wonderful idea about the blotting techniques. Very kind of you, a kind gesture of all. Now over to Vidya ma'am. Vidya ma'am, can you introduce the next speaker?
Yeah, yeah. Thank you, Halim Khan.
Abdullah sir, can you share the screen?
I would like to introduce the next speaker, Professor Abdullah Mohammed Shoail, who's talking on microbes in applied research to improve food and human health. Professor Abdullah Mohammed Shoail is a professor in the Department of Biotechnology and Genetic Engineering, Jahangirnagar University, Dhaka, Bangladesh. He has joined the International Rice Research Institute IRRI as a research collaborator after completing his PhD on agricultural biotechnology from Changbuk National University, South Korea in the year 2006. And he worked on the overseeing of aquaporin functions of drought-tolerant mechanisms in rice. While pursuing his JPS postdoctoral fellowship from 27 to 29 at the Suzuka University, Japan, his prior responsibility was to develop genetically modified lettuce plants harboring a taste-modifying protein, miraculins. As a research associate of the University of Florida from 2009 to 2011, he attained his research goal successfully by developing a model on greening and canker-resistant transgenic citrus cultivars. He had a long research track on plant tissue culture, commercial production of plant cells through bioreactor, food biotechnology, microbiology, and genetic engineering. He has published articles in many international journals and books. His total citation is 830 and his H index is 15 and I-10 index is 16 and his RG score is 20.49. His key research interests to isolate genes from local biodiversity, genomic engineering, and genome editing approach to develop environmentally resilient plants or crops. His area of interest are metabolomics, metabolic engineering, secondary metabolism study from various endangered species and their conservation. He's also trying to establish institutional biosafety and biosecurity guidelines and procedures. He's very much keen to arrange campaigns to create public awareness and motivation towards new technologies. He's a life member of the Microbial Society of India. With this, I would like to welcome Professor Abdullah Mohamed Shoaib for his presentation. Thank you. Over to you, sir.
Good morning and Assalamu alaikum. Thank you so much for inviting me in this very time-being and important conference, International Conference on Plants and Environment, arranged by Department of Botany, Giras Government College, in collaboration with Council for Green Revolution, Hyderabad, Department of Botany, Telangana University, Nizamabad. Personally, I don't know all of you, even though we didn't have any collaboration before, but this corona pandemic gave us the opportunity to come forward and interact each other. So it's a blessing of corona pandemic. Though lots of people are suffering, lots of losses we have, economic, physical, different types of losses we had, but fortunately, lots of things also we also gain from this corona pandemic. So I am basically a plant biotechnology person, and it's a better way to talk about plants. But today I choose a few other aspects rather than plants. This microbes in applied research, so where I will focus about plants as well. I was hearing from Mr. I forgot his name, but he's from Council for Green Revolution. And I really appreciate that what he had explained about their mission, vision, and what they are doing. Basically, they are doing a lot to make our planet green. So I appreciate that, and thank you so much for Council for Green Revolution for your kind and noble work, and I believe you will do more and more in future. First of all, I would like to introduce myself and my university. I am Abdullah Muhammad Shohail, now Professor, Department of Biotechnology and Genetic Engineering, Jahangirnagar University, Sabar, Bangladesh. So where is the university, Jahangirnagar University? Jahangirnagar University is a big university and public among the four autonomous university in Bangladesh. Jahangirnagar University, one of them. It's established in 1970 and public and co-educational. Our Chancellor, Honorable President, Mr. Abdul Hamid, our Vice Chancellor, Professor Dr. Farzana Islam. We have two Vice Chancellor. We have around 700 academic staff, I mean faculty, students around 15,000. Its total campus area about 700 acres, 2.8 square kilometer, and it's located near Dhaka. From the center of Dhaka, it's 35 kilometers away, and from the airport, it's 22 kilometers away. So it's very relevant and it's very nice example to share with you. Our campus is full of green. Jahangirnagar University campus is a landmark of the university campus in Bangladesh. Every day, thousands of people just came here for visit. It's greenery, it's lake, it's migrated birds, and it's Jahangirnagar University trying to keep the natural conditions alike as well. Millions of tree plantations are there, natural and planted, and we have very good numbers of artificial lakes where in every winter season, millions of birds, they travel from different area. Here, please have a look. Then I am cordially inviting all of you, if you have any chance, to visit in Jahangirnagar University, and also research collaboration with me as much as possible. I will very much interested to do that. So here are a few snapshots of our university campus, and recently we have several constructions and others work are going on. So it will be much more greener and nice campus very soon. Now about the microbiology and microbiologists. Around the globe, microbiologists are making a difference to our lives, ensuring food safety, disease prevention and treatments, green technology development, bioenergy production, GMO development. So most importantly, they are working and they are doing a great job tracking the role of microbes in climate change. Where microbes are not present, actually, if answer this question is very difficult. Where present is better than say where it does not exist or does not present. Why study microbes? We live in a planet that is completely dominated by microorganisms. About half of the Earth's total biomass is entirely composed of microbes, whereas animals just make up about 1 per 1,000th of the total biomass. Our knowledge expanded and began studying microorganisms at the larger scope when next generation sequencing technologies and metagenomics happened. Previous speaker, Dr. Sham, he was very nicely explained about the technique. That technique also useful for microbial studies or different types of microorganism study. What is our challenges and opportunity? In that case, I will explain both plants and microbes in together. First, I am going for plants. In plants, we are getting carbon dioxide, getting rid of carbon dioxide, and we are getting oxygen, but we can't feel that because it's very regular process of our life. But medicinal plants we are feeling every day because lots of disease and different types of health well-being. We are taking the medicinal plants raw or supplement or tablet or different types of form. So we know much about medicinal plants and their direct role in our daily life, rather than getting oxygen, eliminating lowering the carbon dioxide, lowering the temperature. So we are directly involved getting the plant-based medicine different types of crops, fruits, vegetables, and such and so. I am focusing about plant-based medicine production. In India is world leading medicinal plant producing country, and they are supplying around the world various types of medicinal plants as a raw material, active ingredient. But we have few problems about cultivation, manufacturing, and marketing and distribution. What are those problems basically? Botanical characterization. So many plants, they are very useful, and as a consumer end, it's very difficult to recognize is which plant I am taking. So here, adulteration with wrong plant species, very easy to do that. Field production, contamination, pathogenic threat, abiotic biotic and abiotic other stresses are very much threatening for field production of medicinal plants. Environmental pollution, contamination with heavy metals, organics, herbicides, pesticides, crop nutrition, altered profile of medicinal metabolites. During manufacturing, harvest, all plant materials are received to manufacturing facility at once, loss of medicinal compounds when it dry, then degradation of medicinal compounds when long time storage occur, and filler, binders, and other processing is sometimes is very difficult. Marketing and distribution, exaggerated efficacy always distributed or uh, publicized for the medicinal plant. So, what are the modern biotechnology can give solution to do that? This is cell culture, organ culture, and bioreactor culture could be a could be an alternative of producing valuable medicinal plant in vitro more precisely and more controlled way. So here, if you have a look about a picture that is showing a very uh, mm, valuable medicinal plants growing in the Korea, China, and Japan, it's called ginseng. And I just came to know that India in Himalaya region, NBRI and maybe Assam University, they also got few ginseng species from the Himalaya belt, and they are now trying to establish those culture system in vitro and in bioreactor as well. So first of all, culture initiation, and then cell line selection, then go for suspension culture, small scale bioreactor, and finally go for commercial bioreactor. So this is the alternative way of producing very rare and expensive medicinal plants in a large scale production for commercial use. What other studies we usually do in our lab, and almost all of you here, you are listening, your lab also. So this is the those are the very common workflow that regularly all of we are doing. What are those? Like callus culture, or either directly selected plants. We go dried sample, or we do the callus culture, organ culture, go for flask culture, go for bioreactor culture, then harvest the fresh sample. We do antioxidant enzyme assay, secondary compound assay, or we collect the dried cell from direct from plant or from tissue culture derived plant and go for making powder and go for extraction. Then we purify the extract and do various types of phytochemical analysis, antimicrobial analysis, anti-diabetic, anti-helminthic, anti-thrombolytic analysis, free radical scavenging activity, antimicrobial as a nanoparticle synthesis. So lots of characterization also we do, FTIR, XDR, SAM, spectroscopy, lots of things. Then in vivo study, a cytotoxicity test, cancer cell line using, or non-cancer cell line we do use. So these are the flow chart. And another workflow we do, this in silico work. different types of in silico work. I mean, if we don't have the laboratory facilities and if we know some known compounds, we can easily go for this ligand preparation, target identification, then protein preparation, energy minimization, receptor grid generation, molecular dynamic simulation, molecular docking, then pharmacokinetics like ADMET, QSR. So those studies we can do different types of target like Alzheimer's disease, Parkinson's, neurodegenerative disease, different activities against different types of virus activities. Now, I'd like to focus about the microbiological recent threat. All of us, we aware about that coronavirus pandemic. So, infectious disease like coronavirus disease are a global hazard that puts every nation and every person at risk in everywhere. Right now, nowhere is safe, nowhere is out of control of the corona. I mean, coronavirus spreaded everywhere around the world. I mean, only maybe space shuttle, it's a safer place right now. Otherwise, in the world, every corner, coronavirus spreaded. Where the mobility is there, coronavirus is there. The recent coronavirus outbreak is a prime example, knowing neither geographic nor political borders, often arriving silently and lethally. Microbial pathogens constitutes a grave threat to the health and human. Throughout history, humans have struggled to control both the causes and consequences of infectious disease, and we will continue to do so into the foreseeable futures. And our journey begins after this corona threat started, and we are getting the benefits of different types of corona vaccine already within short period of time, less than a year. Some sorts of vaccine are here. So those are the microbiological, biotechnological research avenue we are getting that. So I am not highlighting those things, and this is the possible loss, and I am not also focusing also. What are other topics in microbiology that must we do in our daily research or we should focus on? Number one, antimicrobial resistance, environmental microbiology, pathogenicity, and virulence. As far I know, in India, they are doing a tremendous progress working with those environmental microbiology, antimicrobial resistance, and pathogenicity and virulence resistant example. In so many, even though college labs and so many small institutes, small entrepreneurs, they are doing a good job of producing different types of products using several microorganisms for human well-being. Then biotechnology and synthetic biology, little bit high through output research and need much more investment, and getting the results is quite time consuming, and also scientific proofs are required. Studies of microbiomes and food biotechnology, that's also important, and this is very common aspect, and lots of efforts are going on getting the different improved food. Then, if we think microbial decomposition of organic matter, what is the contribution of microbial decomposition? Really, we can't imagine this effect without decomposition of what? Even though today we cannot survive, I mean, if no organic matter decompose from the beginning of the civilization, I will rather say civilization, or the from the beginning of the Earth's history or our planet's history. So it would be stacked over the world. But Almighty gives those microorganisms a good job to decompose all organic residue and release the energy again, so that the energy cycle, different types of organic and inorganic cycles are continuing. So this planet is recycling their own product for their survival, for our survival and others. Human microbiomes, it's a huge subject, and we are living with those microbiomes. Lots of them are beneficial. Very few are harmful, but most of them are beneficial. Plant microbiomes, marine microorganisms. So our studies are expanding, and we are getting benefits by utilizing those microorganisms or by doing research on those microorganisms in our daily activities. Now we are characterizing different extended spectrum beta-lactamase and metallo-beta-lactamase genes of non-fermenting gram-negative bacteria, hospital sample, Suarez sample, different types of fecal sample or other sample. We are doing those for our betterment. I mean, microbes is giving is good benefits to us, and sometimes it also getting their abundance and frustrated us. So sometimes we need to control them. I mean, and for time being, and I will say our different sorts of activities, I mean, over activities, those microorganisms are getting much more competent of surviving, so that our regular antibiotics, those are not working. So we have to use fourth generation antibiotic, and some of them are still not responding against the fourth generation antibiotic as well. So this is the alarming situation, and our act should be minimized. We should think for green planet, we should think for green, then maybe those threats will be minimized day by day. So I will not focus much about the research activity, because I think here most of the students from the undergraduate level. So if you just see this, our study revealed that those high doses of antibiotic, actually those are resistant already, those bacteria already resistant. So lots of papers you will find from different labs, different research initiative, they are explaining about these threats. Food microbiology, it's one of our research about probiotics and yogurt sample and milk, fresh milk sample, and we studied that. Parallelly, we have two different aspects, as we got very nice active probiotic bacteria from the locally made yogurts. On the other hand, we also found so many threatening bacteria as well in the yogurt sample that could be sometimes causing the severe food poisoning and others. So we have to be clean, we have to be practicing well. I mean, food, good laboratory practice we have to follow, and good hygiene we have to maintain, so that we can make our food safer. And also from the milk sample, we got different kinds of antibiotic from it, and antibiotic resistance is very, very alarming these days, not only Bangladesh, India, and other countries as well. And our another research on this health effect of street vending aloe vera juice, though all of we know the effect of aloe vera juice in our health, but our study was about how much safe those juice from the leaf, I mean, leaf made juice in the street vending condition. When in the street vendor are preparing those juice, and they are using another ingredients, water and other ingredients for preparing the juice, and how healthy at the final product that we are consuming. And here, eventually, we also got the very alarming situation, and lots of alarming bacteria with their antibiotic sensitive and resistant strains also we find out from there. Then plant antimicrobial studies. This is simple flowchart. We first of all, we choose known or unknown medicinal plants. Then we go for metabolites extraction, I mean crude extract, then purify, extract. Then we go for compound identification if those plants are unknown plants. If known plants, then we validate the compounds and its efficacy. Then compound selection, we go for active ingredient selection, preparation, and go specifically antimicrobial studies in the selected microorganism. We go for good works. We do several plants and this antimicrobial study in our lab. Recently, we are doing different types of spices, different types of herbs, and different types of plants. So I will skip this part. Neoteric research direction, this is very important, and for making the green planet, it's, I think, very much unique. And this research is recent research, and we should focus on that. Unique and common traits in mycorrhizal symbiosis. Mycorrhizal symbiosis is a natural process, natural way, and due to the symbiosis, lots of good effect to conserve natural biodiversity since the beginning of the earth, I mean this planet, green planet, and this way they are surviving. Scientific knowledge gave us outlook, gave us the in-depth ideas how those symbiotic relationship works, how they are contributing, how much their contribution, and how much we can utilize those. So we are getting those information from the nature, and we are utilizing those environmental friendly mycorrhizal symbiosis in our enhancement of the production, reduction of the different types of pollution and such other ways. So it would be a new research direction that we can focus in our daily research. Natural products of an actinobacteria in host microorganism and microorganism interaction. It's also new direction of the research, and from here we can isolate different novel genes. Those have different types of activities like pathogen, disease resistant activities, different types of nitrogen conservation activities, different phenolic compounds producing activities, medicinal activities. So we should explore different plants and this microorganism interaction to get the better result from there. So common principles and best practice of engineering microbes. So recently genome editing and other tools, as Dr. Sham is very much expertise on this molecular validation or molecular tools. So he explained few tools here. So we can utilize those tools, and we can go our betterment, and we can enhance our research for that. Recently, all of you know about this CRISPR-Cas future neoteric research and innovation. This not only in microbes, not only in virus, so not only in others like plants, animals, and everywhere this CRISPR-Cas mediated genome editing or other genome editing is now flourishing. Although the spotlight on CRISPR-Cas system has shown on their immense potential as genome editing tools, the field's origins are rooted in the microbiology phase bacterium interaction. Furthering our understanding of this process can uncover more systems and generate a new reagents with revolutionary properties. So this research idea came from the natural microbiological process inside that microbes are doing since long time back. I mean, from the beginning of their evolution. So application of CRISPR-Cas systems in establishing animal and cell models of human diseases, and you can see lots of progress around the world. Application of CRISPR-Cas system in disease diagnosis. Recently, even though corona diagnosis CRISPR-Cas system, an Indian developer, they developed the technique, this CRISPR-Cas system for diagnosis the COVID disease. Application of CRISPR-Cas system in genome scale screening, CRISPR-Cas system in gene therapy. So lots of uses right now ongoing, and lots of research are focusing about this CRISPR and their future aspect. We must learn not just how microorganisms, even though plants and others, affect climate change, including production and consumption of greenhouse gases, but also how they will be affected by climate change and other human activities. I mean, we are just thinking about climate change, but why climates are changing, we are not thinking much. Our thinking is we should change our thinking that climates we are changing, our activities are changing the climates because we are cutting the plants, forests and biodiversity we are destroying for industrial revolution, production and production. We are filling out the natural water reservoirs. We are monoculturing different types of plants, monoculturing of aquaculture. We are doing that. In that way, sustainability reducing, biodiversity losing, and climates are changing. Agricultural and other human activities, they are affecting microorganisms directly. Industrial waste, livestock waste, and different types of this step farming and other farming we are doing. And this all residues we are throwing our mother nature, and those are very much harmful, and making the different types of microorganisms much more competent and much more vigorous, much more dangerous for our survival. Climate change exacerbates the impact of different sorts of pathogens, not on human pathogens, animal pathogens, fishery aquaculture pathogens, different crop pathogens. So right now, for the mobility to the around the world, because right now this world we cannot stop our mobility here and there, and then we are spreading those pathogens from here and there. Recently, all of you know that this wheat blast identified 3 years back in Bangladesh, and after that it spread it in the India. What was the origin? Origin is not from South America. From South America, it traveled through here. So disease and those harmful microorganisms, they have no boundaries at all. They are moving everywhere. We should think, we should really, really think. So in my final concluding remarks, we have to go green, we have to think green, we have to like green, and we have to act green. Reduce carbon emission for the sustainable green planet. And we should think and act positively to save our planet, protect our biodiversity, not by hypothetical. This reminder for me and all of you as well, because hypothetically we are thinking, we are doing lots of things, we are making lots of seminars, symposium, lots of government activities, but practically how much we are contributing, we should think about that. From every person, if we think, if we start working from our end, so our planet will be green very soon. We should not think government will do, we should not think NGOs will do, we should not think UN will do, we should think we have to do. When we have to do, then our planet will be green very soon. So many factors influence the balance of microbial greenhouse gas capture versus emission, include the biomes, the local environment, food web interactions, responses, and particularly anthropogenic climate change and other human activities. So we have to think, we have to act very fast, and it's prime time to act. So for those instances, right now, Science Portal Bangladesh is doing a great job here, and I am organizing this Science Portal Bangladesh. If you are interested, you can visit our Facebook page and also our website, scienceportalbd.org. org, and you will see we are trying to reach general people to make awareness for the green planet, zero carbon emission, and good scientific endeavors. We are trying to reach or we are trying to transport those messages to the general people in their own language. So thank you so much. I am very much thankful to my university, my department, Biotechnology and Genetic Engineering, Ministry of Science and Technology, and other partners that really are helping us for making the progress about very tiny amount of research, because we are not high throughput researchers, we are very basic researchers, and we have very limited facilities for the research endeavor. But I am very much hopeful and interested to make active and fruitful collaboration with all of you, because we are doing very great job, and you are welcome to contact me for any kind of assistance. And I'm inviting all of you in Bangladesh in new normal situation, and also I'll be visiting your college and your institutes in future when new normal situation will be. Thank you so much. Thank you for your passion sharing. And I am sorry because I did some gibberish presentation here. I was so busy and it was very short notice. So I do apologize for that. Thank you so much.
Sir, it is a kind gesture that you have given a presentation, very beautiful presentation. With a short notice, we have requested you and you have accepted our request. It is a very kind gesture. Now I request our next speaker, Professor Anjana Singh, madam. Please, ma'am, can you start sharing your screen, ma'am?
Yes, thank you.
Yeah, over to Vidya, madam. Vidya, madam, can you introduce our next speaker?
Yeah, sure. Thank you, Halil Khan. Once again, good afternoon to all of you. Let me introduce Professor Anjana Maharajan Singh, who is a former head Central Department of Microbiology, Tribhuvan University, Kathmandu, Nepal. She has completed her BSc in the year 1989 from Tribhuvan University and MSc Microbiology from Tribhuvan University, and her PhD as well as from Tribhuvan University in the year 2004. She did a postdoctoral fellowship from the University of Virginia, USA, from the years 28 to 29. And her major activities include, she was the head Central Department of Microbiology, Tribhuvan University, Kathmandu, Nepal, from 2004 to 2008, as well as 2012 to 2016. She's an executive member of Board of Studies, South Asian University, New Delhi, India, from 2016 to 2018. She's the executive member of Nepali Society of Microbiology, NESCOM, from 25 to 28. And she's a president, Filters for Families Nepal, SSF Nepal, from 29 to 2013. She's an associate academician of the Nepal Academy of Science and Technology, NAST, from 2010 to 2015, and since 2015 for lifetime academician of Nepal Academy of Science and Technology. She's an executive member of Fulbright Alumni Association Nepal, and she's a life member of Women in Science and Technology in Nepal, and she's an executive member of Health and Environment Welfare Association Nepal. From 2019, she's a life member of Nepal Biotechnological Society, 2013 to 15, 2016 to 2019. She's a UGC cluster member board of for Tribhuvan University Research Committee, and she's Tribhuvan University subject committee member from 2012 to 2016. She is a BOS member of South Asian University, New Delhi, from 2016 to 2018. She's a NAST Senate member from 2017 onwards. She's a NAST Research Committee board member from 2017 to 2019, NAST Management Committee board from 2019 onwards, and coordinator for Women Scientists Forum Nepal from 2020 onwards. She has received many awards like from 2008 to 2009, she's Fulbright Scholarship, United States of Government, and Education Award, that is Shiksha Puraskar, Nepal, 2008, International Foundation for Science Grants, Sweden, 2007, Crown Prince Young Scientist Award, NAS, Kathmandu, Nepal, in 2005, Mahendra Vidyalaya Bhushan Award, Kathmandu, Nepal, in 2005, 2000 to 2004, University Grants Commission Fellowship, Government of India for PhD in India, 2000 University Grants Commission Mini Project Nepal, 1994 Tika Lakshmi Padak Tribhuvan University Medal for securing highest rank among girl students in BSc in Nepal. 1991 Mahendra Vidya Bhushan Award. 1991 again Birendra Aishwarya Scholarship Nepal. 1990 Aishwarya Vidya Padak for among girl students in BSc Nepal, and 1990 Hirarar Raj Roshni Award for standing top in chemistry in BSc Nepal. She has many publications in international, national levels, 125 peer-reviewed journals in around 125, 16 peer-reviewed journals, 3 books, book chapters around 3, general proceedings 28. She has submitted 9 abstracts, and there are 9 papers in the pipeline which are to be accepted. So she's supervising candidates in masters in 1999 and supervised 103 candidates, and she's a PhD guide and a co-guide, and completed 5 ongoing projects. She attended around 67 conferences. She's a peer reviewer for 6 journals and visited around 9 countries. With this, I would welcome Ma'am Anjana Ma'am to share her speak on symbiotic effect of mycorrhizae in plants. Over to you, ma'am.
Thank you, Professor Vaidya, for a very nice introduction. First of all, I would like to thank the organizers, Department of Botany, Giriraj Government College, in collaboration with Council for Green Revolution, Hyderabad, and Department of Botany, Telangana University, Nizamabad, for giving me this opportunity. I really, it's my pleasure as well. And as already been stated, my talk is entitled Symbiotic Effect on Mycorrhizae on Plants. Now, we just wanted to start what is mycorrhiza, and we all know that it has a kind of a mutual symbiotic association between a fungus and a plant, and it also refers to a role of fungus in the plant's rhizosphere and its root system. So when we say rhizosphere, it is around the root is there in a plant, and surrounding of the soil, we say it is rhizosphere, and on the surface of the root, we say rhizoplane. So rhizoplane as well as rhizosphere, it really plays a big role with here to having a mutual association with fungus and the plant. So mycorrhiza, it plays an important role in the plant nutrition, soil biology, and soil chemistry. We can also say it's a soil biota. Here I have shown the figure where we can see that when there is a mycorrhization in the plant, it has a very good proliferation of rooting system with a very fine rooting system here. And when there is no mycorrhization, that is a control plant, we can see that in the right, there is no mycorrhization. So we know from the history that mycorrhiza, it has started in 1880. The term has been coined by A.B. Frank. And since it is in a Latin word, so how that the word has become mycorrhiza. So there are various types of mycorrhiza. Firstly, we can say it is ectomycorrhiza. Here in the picture, in the line drawing, we can see that it is the ectomycorrhization, where the root or the mantle of the root is being surrounded by the hair-like structures, which is mycorrhizae. In the next, endomycorrhiza. When we say endomycorrhiza, it is within the cell wall, and it will penetrate in the cell wall and invaginate the cell membrane. So this is where the place you can find the mycorrhiza. So it is known as endomycorrhiza. So when we say it is ectendomycorrhiza, that is the third type, it belongs to the same Basidiomycotina and covers both gymnosperms and angiosperms. That is the class of the plants. So it is known as ectendomycorrhiza because it is having in the both family it is covering. So we say it is ectendomycorrhiza. And generally it shows extensively the growth in within the intercellular penetration. So it will begin a formation of heartic neck. It is a kind of a network of the mycorrhiza, and which it grows behind the apical meristem of the growing root. And this heartic net, it penetrates between the epidermal and outer cortical cells and later extends to the inner cortex region. So when we talk of applications of mycorrhiza, mycorrhiza we say in a singular form, mycorrhizae it is in the plural form. And it increases nutrient uptakes of the plant from the soil. So generally when we say it is the nutrients, it is in the mineralized form, that is the phosphorus nutrition and other elements like nitrogen, potassium, calcium, manganese, zinc, copper, sulfur, boron, molybdenum, iron and manganese and chloride, etc. And it will also increase the mycorrhization, it increase in the diversity of the plant also. And it produce uniform seedling, and it has a significant role in nutrient cycle, recycling also, and it is more tolerant to adverse soil chemical, and it is constraint which limit the crop production. It helps in the increase of plant resistance to various diseases and drought and salinity conditions, and also it helps to stop the pathogenesis of the plants. And it stimulates the growth of beneficial microorganisms, which will help for the promotional effect like PGPRs, which will help the plants for the growth and promotion of the plants and improving the biomass, etc. And it will also improve and help in soil structure, that is conditioning of the soil, and it stabilizes soil aggregate and hyphal polysaccharides that bind and aggregate soil particles, right? So this will help the conditioning of the soil. It increases absorption of phosphate by crops or some other medicinal plants, flowering plants, and forestry plants, etc. It will help to uptake zinc, and it also increases its availability in the root system. And it also helps in uptaking water from the soil with the capillary action. And it is believed that from the root system of the plants, it almost goes to 100,000 feet away from the rooting system. This mycorrhization can help in uptaking the nutrients and water to the plant. It increases uptake of sulfur from the soil. This has been already been said also. And also concentration of cytokinins and chloroplasts in plants is increased. And it will also protect during the stress conditions in salinity, in the harsh conditions like high pH level and in drought condition, etc. So in short, the types of mycorrhizal fungi, it has 7 classes here. You can see 7 types. Ectomycorrhiza, arbuscular mycorrhiza, ericoid mycorrhiza, arbutoid mycorrhiza, monotropoid mycorrhiza, ectoendomycorrhiza, and orchidaceae mycorrhiza. Here you can see in the picture that how these 7 types of mycorrhization it occurs within the rooting system of the plants. Of course, it is in the line drawing. And it, as I have already told you, it has a very vital, big role in overcoming the drought stress condition. Like when there is a vitrification in the glass formation, and protection of membrane is being taken if the sugar is very high. If protein is very high, then dendrites and heat shock proteins, these can be also maintained by the mycorrhizas. And antioxidants and various types of enzymes that can be also maintained by the mycorrhiza. And there are free radical scavenging processes also helped by the use of mycorrhizas. And osmoregulation, that is the water uptake, is also being regulated by the mycorrhiza. So in totality, almost it has been observed that 90% of plants are colonized by mycorrhiza, and mostly these falls under economically important crops. So when we talk of beneficial part of mycorrhiza, we will divide into 2 parts, like for the beneficial for the plants itself and the beneficial portion that is for the fungus itself. When we talk of plants, that is connection between root and soil around the environment we are talking of, that is the rhizosphere part, and it helps the growth. It helps as a growth promoter for the plants, and it will help in the nutrient uptake, increasing the surface area, increasing mobilizing unavailable nutrients like rock phosphates, which is not easily being taken by the plants itself. And obviously there are some microorganisms like other bacteria also help in chelating this rock phosphorus, but however this mycorrhiza can also help in chelating the rock phosphorus, which can be easily taken up by the rooting system of the plant. And excreting chelating compounds or sometimes ectoenzymes is also been transferred by the mycorrhiza to the plants. When you talk of benefits to the plants only, fungus only, it helps to transfer metabolites to the from the plant to the fungi. It will take up, and plants transfer almost 10 to 20% of carbon to the fungi, because as we know, this most of the fungi, it lacks in chlorophylls also, like some of them are also mushrooms, where this mycorrhization helps to uptake the nutrient materials to the fungus. And nowadays, this period, there is a kind of a arbuscular mycorrhiza-like fungus, and mostly the arbuscular mycorrhiza, these are known as Glomus mosei, and there are so many types of fungus which can grow within the root, but their sporulation can be seen in the soil as well, but it cannot be cultured in the lab alone. But very surprisingly, there is arbuscular mycorrhiza-like fungus. We say like because it is all the characteristics is mimicking with the arbuscular mycorrhiza, like AMF fungi. But this Piriforma spora indica, it is easily culturable in the lab condition also. That is why we say it is AMF-like fungi. And nowadays, this Piriforma spora indica, it has been known as Serendipita indica, which is also the same, only the naming is different. And here we can see that this is the root section. Staining has been done by the trypan blue, where you can see that in the section of the root, you can see that within the cell wall, we can see that there is a spores growing within the root cells, right? And these has been magnified here to the extreme right lower side. This has been magnified, so let you see that how the spores are looking like. It looks like the shape of the spore is like a pear shaped. So because of this, how the name has been coined as Piriforma spora indica. This was first isolated in association with spore of Glomus mosei from this is the AMF fungi, Glomus mosei, and it was associated with this fungi. So this has been isolated from the rhizosphere of two shrubs in the Indian Thar desert, that is the northwestern Rajasthan, in 1990. And the paper was published for the first time by Virma et al. In 1998. So soon after, the fungus was characterized as a mutualistic endophyte because of its ability to colonize plant roots without causing any visible disease symptoms in roots or shoots. So instead of, it confers a strong growth promotion and induces disease resistant to microbial pathogens. So most of the laboratories within the world, almost 23 laboratories are working in this fungus nowadays. So molecular phylogenetic analysis have revealed that P. indica belongs to the Basidiomycetes, that is the order of Sebaceales. So it is also known as Sebaceales now. The salient features of AMF-like fungi, that is the P. indica, you can see that it is exonically culturable on synthetic media. So this is a very different drastic characteristic from the AMF fungi, which is not culturable, but of course you can grow this fungus AMF fungi within the plant root, in the laboratory as well as with the plant in the greenhouse, and also in the ex situ and in situ condition. But this B. indica, you can just take out the spore and culture in the synthetic media in a petri dish. This is the difference that has been observed from the AMF fungi. And it has no clamp connection, and there is anastomosis is occurring frequently. Hypha-hypha aggregation is often observed, no hyphal knots, and simple septum with dolypores and continuous straight parenthesomes are being seen. And chlamydoid spores are a little bit smaller in compared to AMF-like fungi. AMF fungi. And that is 16 to 25 micrometer in length and 10 to 17 micrometer in wide. And then 8 to 25 nuclei spores has been seen, which I have also shown in the trypan blue staining, where you can see the nuclei within the spore. So what are the functional similarities with AM fungi? So when we look through it, we can see that broad and diverse host spectrum as similar to AMF fungi. Hyphae extramatrical and inter and intracellular is being seen within the root region. And hyphae never invade the endodermis layer. And chlamydospores in soil and within cortical tissues is being seen. Sexual stages till date is not seen. Positive phytopromotional effects on tested hosts is being seen. Phosphorus mobilizer, phosphorus transporter, tool for biological hardening of micropropagated plantlets is being seen, and it's doing really good. And potent biological control agent against root pathogens has been also tested. And thus, this Piriforma spora indica, besides the biological hardening, that its protoplast can be also applied and for the fundamental researches and genetic transformations. And when comparing 18SR DNA sequences that is obtained after the PCR, it is showing a kind of fungus apparently that occurs in the rhizosphere, which is almost similar, which has been found in Pakistan and Philippines. That is with the similar to rice plants and some few grasses. But however, it is not the exact findings is being seen, but it is similar. The host spectrum of Piriforma spora is very much alike like AMF fungi. That's what I'm saying still, that it is known as AMF-like fungi, where we can see here similar to AM fungi, this has also 90% of the terrestrial plants colonization, and specifically mostly economically important plants. And it is also shown that it is well inoculated in medicinal and forest and ornamental plants, in horticultures, in floricultures, viticultures, etc. It has been successfully tested. And one of the striking differences is that, unlike AM fungi, it has a host range of Piriforma spora indica with terrestrial orchids, where this AMF fungi is not colonizing in the terrestrial orchids, but this Piriforma spora indica is it is colonizing terrestrial orchids. So nowadays, since it's showing as a biofertilizer, it's acting as a bioprotector, and as a also like a herbicide effect or bioprotecting effect. So right now, it is also known as a root tonic. So this is the exact form of electron microscopy of Piriforma spora indica, hyphal and the spores, where you can see with this white arrow, it's been showing this pear-shaped spores has been seen. That is how this name has been coined. And there is a hyphal coil here in the middle, you can see. And there is, when it has been magnified, the hyphae has been magnified, you can see that there is a kind of a funnel type of apristoria is being seen. And this apristoria is very much helpful in transporting the nutrients to the plant. Now, when we talk of mycorrhization, how it can be done? It seems simple, it can be done, So in the laboratory, in the in situ or within the greenhouse, we can do this mycorrhization, and we can do the mass propagation of this Piriforma spora indica. Here you can see that soil inoculum is there, obviously, like similar as with other AMF fungi. And you take one flower pot that is semi-sterilized. We say semi-sterilized because since we are using with the soil and sand here, now, this cannot be completely sterilized as you do with some other bacterial media, right? So soil and sand is been taken as 1:3 composition, and it has been successively, it has been autoclaved in alternate days, heating, cooling system. Then it has been, the sand is well washed with the acid, and then with lots of water you wash off all the acids remaining. So the crevices in the sand, there is no other natural mycorrhiza left or any other organisms left. So you make sterilized the substrate. Now you say 1:3 composition of sand and soil composition. Then what you do is one layer you put this lower layer, the substratum. Then in the middle layer, you put the inoculum, that is a soil inoculum containing the Piriforma spora indica. Then again on the top layer, you put the substratum. Now you go for the planting of the seedlings. Here has been the corn maize seedling has been seen, where you can implant this seed within the layer of the inoculum. This is how you do the mycorrhization. And the second style you can do is, the technique you do is, you take the culture, as I have told you that it can be easily cultured in the laboratory, in the synthetic media, that is a MM media, that is a mineral media, or even in potato dextrose simple medium you can do it, because this is a fungus, right? So it can easily grow in the synthetic media where you can similarly grow the other fungus. After growing this, about 5 to 7 days, it will grow radially and it will cover the whole media when you place a plug of inoculum in the, at the center of the culture media. So you make with a sterile borer, you can make a punch hole, then that or cock borer can be also taken, and that plug is you take as in the substratum, as before in the earlier slide I have told you, make the substratum as 1:3 sand soil composition. Then you put the plug about 2 centimeter deep, right? Then you plant a seedling, or you can also put the maize seed within it and then cover with a substratum and wait for 15 to 3 months. Then this is how the proliferation or mycorrhization can be produced. This can be also similarly worked for the AMF fungi. Since this is an AMF-like fungi, you can also do the similar technique. So exenic culture shows that these are the spores of the Piriforma spora indica on carrot root organ culture, where it can be also done in the laboratory condition. You can get lots of spores in this case, in this way. So for identification, you can easily collect the spores and then take out the protein out of that, and then get the antigen. So you can go for the immunization in rabbit or mice to get the antibodies. So how that way also in serological also by using serological technique, you can also characterize the fungus. So in the lower side, you can see the spores sporulating in the root organ culture, that is in the carrot root organ culture. And this is a biological hardening that has been done with the Withania somnifera, which is a medicinal plant, of course. And here you can see with the mycorrhization plus, that is the mycorrhization minus means that is the control plant without mycorrhiza. That is again Piriforma spora indica, that is AMF-like fungus I'm talking about. And you can see that there is a proliferation of leaves. The plant is very nice and very dark green in color, and lots of biomass has been produced. And later it's also been shown that fruiting and flowering and fruiting bodies is also more in case of mycorrhization in compared to non-mycorrhization. So as I have told you, in the serological identification technique, it's not only that for the pathogenic organisms, for the human disease that you can use, or the plant disease, or in animal disease that you can produce antibodies against it, but for the taxonomy also, you can raise antibodies and then use for the identification of the that particular fungus. Here we are talking of Piriforma spora indica, where antigen is being taken from the spores of Piriforma indica by crushing it in a buffer, and then you take this, obviously the protein, it is the protein. We all know that when you take the antigen, it can be protein or it can be some kind of peptides you can take, right? And the antibodies is raised against it, and there is secondary antibody, that is the enzyme leveled HRPO for the color formation. Sorry, this has been done in the ELISA reader in the and then you can see that there is antigen and antibody complexes being formed with the help of secondary enzyme leveled secondary antibody. There is a fluorescence is being seen, and this reading has been taken in the ELISA reader. And similarly, as earlier also, one of the previous speaker has also talked about Western blot technique also. For the here, this is the dot blot is also done for the identification of the antigen and antibody that is being raised against Piriforma spora indica for identification of the particular strain, that is the Piriforma spora indica. And when it has been, this is just the figure to show you that how it can be cultured in the synthetic media. This is media, that is the minimum media, where a plug of agar containing the spore of Piriforma spora indica is placed, and after 7 days it has a radial growth growing. And similarly, the other sebaceanil, that is Sebacina vermifera, which is almost similar to Piriforma spora indica. This Sebacina has been identified in the European countries in 1960s, and this Piriforma spora indica, it has been identified in the in India in 1998, right? So it has both of it. It is in the same family, almost having the same type of growth, but however, these two are different. So I was talking about ectomycorrhiza also. These are all the ectomycorrhizas. The name is given here, like Trichoderma and then Amanita, etc. So these are also culturable in synthetic media, similar as Piriforma spora indica. Only that different is Piriforma spora indica is AMF-like fungi, but these are the ectomycorrhiza, because these does not colonize the cortical region of the root. It only develops the mantle around the root only. So when Piriforma spora indica is being taken, this A is controlled without any mycorrhiza, A controlled without any mycorrhiza. B is for Piriforma spora indica. You can see the proliferation of the root here, plantation here, shoot I mean to say. And this C is for Sebacina. So similarly here, this is the control. This is Piriforma spora indica. This is Sebacina. These are Glomus mosei and another Glomus species. So you can see here that comparing to another AMF fungi, and this Sebacina is also AMF-like fungi, and P. indica, this is also AMF-like fungi. It's showing the higher shooting and rooting effect. So these are the maize plants, and this rooting system is also from the maize plant, where control, there is no proliferation of the rooting system. In P. indica, you can see that their rooting system are proliferated and it has become lower and hairy and more in biomass. Similarly with Vermifera also, but in this is AMF fungi, you can see Glomus mosei, it's a little lesser than in compared to P. indica. when we talk of biomass this is grown in the broth culture, and this is from the 5 days, 7 days and 10 days replica. And we see that similar method has been followed for the Sebacina, that is the another kind of AMF-like fungi, which is found in the European countries, and P. indica, that is in the India, which is found. We can see that almost similar it is, but however, P. indica is a little bit higher in rate in compared to Sebacina. So this is the cytochemical staining by trypan blue. You can see here, this to the left is all P. indica, and to the right is all in Sebacina varifera sensu, which is almost similar. And through microscopy tests, it's difficult to identify. It looks same. So in the end, I can see that I can say that there is lots of research area has been opened due to this experiments previously done by, that is myself also. I did my PhD in the same fungus from Jawaharlal University under Professor Dr. Ajit Verma, who's my mentor. And similarly, there are so many other students has already done the same work, similar work under the Piriforma spora indica, but still there are yet to do, long way to go. But however, in the end, I can give recommendation as such that it acts as a, this is the Piriforma spora indica spores in the trypan blue staining. It acts as a plant promoter, as a biofertilizer, as a bioregulator, as a bioherbicide, and immunomodulator, and phytoremediator. Biocontrol against insects and pathogens has been also seen. It has a stress tolerance effect also. And like salinity, temperature, etc. and drought condition also. And it acts as a stimulator with PGPRs, that is plant growth promoting rhizobacteria. And it acts as an antioxidants and drug enhancer. There is yet to do lots of work in this area, and it also enhances anti-aging agents also. So yet to go far ahead for the in experiments and researches. And it also acts and promotes anti-cancer drugs, and yet to go more to come. So with this, I would like to end my talk here, and thank you for listening.
Very kind act of kindness, ma'am. You were so kind enough to accept our invitation and gave such a colorful and wonderful talk on mycorrhiza. Surely we'll be summarizing your talk in the valedictory. Very kind of you, ma'am. We expect that many more such kind of talks could be expected from you in future. We can request you for many more talks in future. Thank you so much, ma'am.
Thank you, Ahmed Khan, for keeping in touch with me and inviting me. Thank you all.
Thank you, ma'am. Thank you. Now I request Chandrakanta ma'am to share her screen. Chandrakanta ma'am, over to you. If you start sharing in the screen, I'll be doing the introduction. Okay, ma'am, wait a minute. I'll be starting your introduction. So, yeah, it is proud privilege to introduce our next speaker, Dr. Mrs. Chandrakanta Mahendranathan. She is the PhD in post-harvest pathology and technology at Eastern University, Sri Lanka, and Cranfield University, United Kingdom, as a Commonwealth Scholar of the United Kingdom. She has been serving as senior lecturer for last 20 years at Eastern University, Sri Lanka. Her research findings have been published in several national and international reputed journals, as well as in many national and international conferences and symposiums, including USA, Australia, New Zealand, Turkey, Dubai, China, Mauritius, Singapore, India, and Sri Lanka. She became one of the nominees among the first three, in fact, the first ever female nominee for the Vice Chancellors of the Eastern University, Sri Lanka in 2018, to be appointed by His Excellence, the President of Sri Lanka. Throughout her career, she has been engaging in several administrative positions at the Eastern University, Sri Lanka, such as Head Department of Botany, Director Center for Information Communication Technology, Director Center for External Degrees and External Extension Courses, open distance learning, director, career guidance unit, etc. In addition, at present, she is holding the following national positions at this university. President of Sri Lanka Sports Association, which is affiliated to International University Sports Federation and Asian University Sports Association, Vice President of the Federation of University Teachers Association, FUTA Sri Lanka. In addition to that, due to her interest in social service, she became a member of the International Lions Club, in which she was the charter president of the campus Lions Club of the Eastern University Sri Lanka in 2010, and currently engaging as regional consultant for the Eastern Province Sri Lanka in this international service organization. With this words of introduction, over to Chandrakanta ma'am. Chandrakanta ma'am, you can start your presentation. Thank you for accepting the invitation and giving the talk for us. Thank you, ma'am.
Thank you very much. First of all, good morning to you. Hello.
Yeah, you're visible.
Hello, can you hear me, please?
Yes.
Let me join my screen. Can you hear me well now?
Yeah.
Hello. Yeah. Can you hear me well now?
Yeah.
Okay. Thank you so much for such a wonderful and kind introduction. I'm so pleased and privileged to join at this occasion with you all. I'm happy to again welcome you all on behalf of Eastern University, Sri Lanka. And it's my pleasure, as well as I'm so privileged to join with you at this international conference on plants and environment. It's a virtual mode one. And this is organized by Department of Botany, Government College in collaboration with Council for Green Revolution, Hyderabad, and Department of Botany, Telangana University, India. So I'm so pleased for requesting, finding me from Sri Lanka to join with you as usual. I have been, I think, even in this year, this is my fourth webinar, international webinar that I'm sharing with India as a resource person. So thank you so much. I'm so pleased.
Ma'am, Chandrakanta ma'am. Now there is a power failure at her end. She'll be joining soon. Please be patient for some time. Ma'am, you can start your screen, ma'am. Already you are made co-host.
Can you hear me now?
Yeah,
I
yeah.
have back.
Okay.
I'm so sorry. It's sudden power cut, unfortunately.
No, sorry. Okay, okay.
Let me share again my PPT. Can you see me, my PPT now?
Yes, yes.
Okay, thank you so much. Thank you. I'm apologizing for the it's a sudden power
Please,
cut.
don't use all those words actually.
Thank you so much. Thank you again for the introduction, and by thanking to the organizers, particularly the head of the Department of Botany, of this yes, the Giraj Government College. Thank you, sir. Well, the topic that I'm going to talk about is the post-harvest losses of fruits and vegetables in developing countries. Why I'm selecting this particular topic actually is the currently, due to the COVID-19 pandemic, we are always talking about our immunity these days. Not these days, of course, last 1 year, we have been talking and very, very serious about this, about our immunity, increasing our immunity to fight naturally against the COVID-19 pandemic. So I just want to, with regard to my field of expertise, I just want to connect my field, connecting with the COVID-19 pandemic situation, just nothing but this is how can we increase our immunity naturally to fight against this coronavirus. So I don't need to repeat all these things, facts again behind these fruits and vegetables, how this is the vitamins that we get from the fruits and vegetables are helping us to increase our immunity in the natural body, in our body naturally. So in these aspects, I'm just going to talk about how much we have to be concerned about these losses that occur worldwide in these post-harvest produce, that is the fruits and vegetables, which are, we are being blessed for, of course, to talk about our Asian countries, particularly Sri Lanka, India, all other Asian countries. We are being blessed with throughout the year with all sorts of fruits and vegetables, unlike the European countries, which have different temperature, the weather conditions. So in these aspects, why we have to concern always about the loss of the loss of the fruits and vegetables naturally or artificially occurring, and what is the role of being academics, what is our role to have a look very seriously on minimizing this post-harvest loss of the fruits and vegetables, particularly in the developing countries. Why I'm focusing on developed countries? Because if you take the developing countries like our countries, we are here, actually, we are the one facing nearly 50%, up to 50% of the harvested produce loss of the fruits and vegetables. So this is where I really want to focus about the post-harvest losses on developing countries. So let me briefly just talk about my university. First of all, I belongs to Eastern University, Sri Lanka. I'm glad to say that this is the university I graduated as well. My first graduate degree was obtained. And also this is what you see, some of the pictures of my university, the gateway and also the other building, the Senate buildings and all. So here, this is actually the university was established in 1981. Before that, it was also as a college, it was functioning. And as a fully fledged university in among the 15 universities at the moment in Sri Lanka, 15 state universities, which is situated in the eastern province, where the sun rises in Sri Lanka. So I'm so happy this university belongs to Eastern University, and all multi-language. The medium is actually in our faculty is English, whereas in other faculties we have all other ethnic groups as well. So with that brief introduction, let me come to my subject. First of all, let me talk about something in general about the characteristics of fruits and vegetables. As you all know, why I just want to briefly, though you know the subject, because this is my concern at the moment, why to bother, why to concentrate on minimizing the post-harvest losses. Because what is the characteristics of fruits and vegetables? Actually, it is a very good source of nutrition. and it has high water content and easily damaged, then it is the problem is, since it is highly water content, that is perishable, so this is get damaged easily due to any, particularly by the diseases or any post-harvest handling practices. So this deterioration begins actually at harvest. At harvest, the deterioration or the post-harvest loss begins actually. So why, this is where I'm talking about post-harvest losses, because the losses start at harvest. So we are always talking about, when we talk about the loss of the fruits and vegetables, so simply look into the post-harvest stages, because the loss starts at harvest. Until the fruits and vegetables are attached with the mother plant, no problem at all. But once we start to harvest, the problem starts as the post-harvest loss. Again, the fruits and vegetables are very nutritious and then perishable. Also, seasonally available. Most of the fruits and vegetable seasonally available. This is also one reason why we have to always look into minimizing the post-harvest losses. So again, the fruits and vegetables are an important source of carbohydrate, protein, organic acid, vitamins and minerals, and of course for the humanitarians. Then, however, the fruits and vegetables are highly perishable products, particularly once they have been harvested. So reducing losses is the key to sustainability and food security. This is what, being the academics, we also have to focus at the moment. The highly nutritious products like fruits and vegetables are often more perishable, and investment and lost when produce degrades. And of course, the investment of land, investment of energy, seed, fertilizer, water, labor, etc., money and things are get invested. But if we lose, or if we are going to lose right after the harvest, you can just imagine how much that we are going to lose as a whole. So to increase the food security, we must reduce the producers the post-harvest loss. If you look at here, the short, the diagram, it is schematic diagram that I'm trying to show you how this, if you look at the post-harvest losses, it's ranging from 10 to 50%. Actually, if you take the developed countries, that is still they are maintaining below 35% or even 30%. But whereas if you take the developing countries, still that is at 50 or more than 50%, the post-harvest losses. So as you see here in this slide, it starts at the harvest. That is the post-harvest losses starts at the harvest, and it continues during the transportation, storage, packing houses and marketing, and even at the consumer levels. You could see we just face the loss of the fruits and the vegetables. If you look at here, this diagram, this slide, actually, if you take the world, actually, the production is there. Though that is enough or not, the production of the fruits and vegetables is there. But the problem is, are we getting the 100% of the product that is given by the government? So the simple answer is no. Why? Simply the post-harvest losses. But look into the one side of the world, the poverty. The poverty is a big problem. Whereas in the other way around, if you look at the other side, loss of the harvested produce, the loss of the foods is in the other side. So where we are here, how we are going to connect it? How we are going to eradicate the poverty? By minimizing even these post-harvest losses. So this is one major role that we can play, being an academics, that we can play, how to eradicate poverty by minimizing this food produce or the post-harvest produce. Then if you look at the quality, the produce quality or the quality of the fruits and vegetables, actually, this is a high concern when we talk about the post-harvest loss, because to provide the high quality of fruits and vegetables throughout the year, it is required to start from high quality material at harvest. But of course, at harvest, we obtain the high quality produce. But what is happening after that? After the post-harvest chain, from the point of harvest. So the product quality is only maintained after the harvest, but it is not improved. We have to concentrate. We should, look into that. The quality is not improved after the harvest. It is just maintained. So our duty is just maintained because the quality one is already been given from the plants. So maintaining that quality is our prime duty until it reaches the consumers. So the pre-harvest and post-harvest factors, we can, of course, the pre-harvest factors are very minimal. It is playing a role, but the post-harvest factors in losing the post-harvest quality is playing a major role. So the quality is the most important factor for marketing and export as well. As you all know, in your country, the India or any other countries in Sri Lanka, we export the fruits and vegetables, flowers, etc., etc. But where we have to be very much concerned about the quality, not even as exporting with regard the export in the market, even until it reaches the consumer level. So what is quality? What if we are talking about the quality of the post-harvest produce, the quality? So we can categorize as external quality as well as internal quality. As described here, the external qualities, you can see the what are the external qualities and what are the internal qualities. And when we are talking about minimizing the losses or maintaining the quality after harvest, we have to look into this both, the external quality, to always to keep our produce fresh, which are being marketable and which are consumable by the human being, as well as internal. Consider about the internal quality, because this is where we are talking about the all sort of the vitamins, gaining the vitamins and all what we need to improve our immunity in our body. So if you look at here, I have shown you here two different pictures, but we are talking about this one, the pineapple internal quality, and here in the case of tomato, the external quality. So both have to be looked into very, very seriously when we are talking about post-harvest losses. So the total quality, if you look at that, the production quality, harvest and storage quality, packaging quality, transportation, etc., etc. So all these means the total quality of the harvested produce. So again, this quality again depends on the, of course, the pre-harvest factors and more than the post-harvest factors as well. That is how we take care at harvest without any damage, causing damages to the fruits or the vegetables that are being harvested. Then packaging houses, how we maintain the quality. At the storage conditions, what is the quality of our storage facilities? How we maintain the quality? Because always, again, I'm repeating, we are not going to improve the quality. We are going to maintain the quality. So at each and every level, at harvest, at packaging houses, at storage, at transportation. So how we maintain this quality is the question. So again, the quality, the post-harvest losses of fruits and vegetables, if you come to the loss, actually, we can again categorize this qualitative loss as well as quantitative loss. Again, the both are important. If you lose the qualitative loss, no point of talking about the export or the marketability, etc., etc. Acceptability by the human being as well, or by the consumers. Also the quantitative, that is what I said. In developing countries, we are facing nearly 50%, up to 50% of the quantitative loss so far, which has not been rectified, though we are with so much of research in this field. So this is what will happen ultimately, the loss of acceptability by the buyer, loss of weight, loss of calorie and nutritional values, and loss of edibility as well. If you look at here in this slide, actually, the estimated post-harvest losses of fresh produce, here I have taken just few examples, how we can compare the developed countries and developing countries, what is happening. How this is, I mean, the post-harvest loss, the percentage wise, what is happening actually. So if you look at here, the locations, different locations, what is the range between the loss, percentage of loss between the developed and developing countries. So if you look at, first of all, the from production to retail sites, the range in the developing countries is 2 to 23 only, whereas in developing countries, it goes up to 50%. Again, the same way at the retail, food service and consumer sites, even here, if you look at here, the range but in developed countries, it is going up to 5 to 30%, where fortunately we are still with the 20% of the up to 20% in the at the consumer side or the food service side, the losses. So the ultimately what we are talking about, the estimated post harvest losses at each and every level, we face this percentage. I mean, the loss is at almost as 50% of the loss in the harvested produce. Then if you look at the causes, what the reasons for this post harvest losses, particularly in developing world or the developing countries, because of the simply talking the economic problem, where we don't have, still we didn't meet the needed level of the sophisticated or the high technological side in all these post harvest chain. That is post harvest transportation can be, or the storage facilities, and etc etc. Still we are in the way of improving our all such facilities. So if those facilities, particularly the storage facilities or the transportation facilities, are being improved as in the developed world, of course we can bring down these losses also from 50% to further down and down. So the first of all, if you look at the high temperature after harvest, is a very big problem, because our country, mostly the vegetables or the fruits and vegetables are produced. So due to the high temperature after the harvest, that is sun exposure, lack of cooling facilities, cold transportation facilities or cold storage facilities, this high temperature and the loss due to this the hot or the high temperature, we lose a higher percentage of the harvested produce. So this high temperature leads to the water loss, then physical damages, decay. Also, all these are happening because of lack in all these facilities, the cooling facilities or the cold transportations, cold storage. So this is where the simply we are in the dry zones, mostly the dry zone, the vegetable, the agriculture is being undertaken. So then high temperature, what is happening, or how this is actually the high temperature affects the produce after the harvest. So here the temperature related to the water loss, the related and over ripened. So simply the decay, the water loss leads to the decay of the fruits and vegetables, or the water loss simply the deterioration of the quality, the water loss due to the high temperature. So they limits the time for marketing after harvest. Again, another problem. So the farmers must sell in very short problem. They don't have enough storage facilities with cold facilities or the cold room transportation facilities, mainly the storage facilities. So the farmers tend to sell it as quickly as possible because they are also poor people. So in this case, what will happen? So the quality is questioned there. Then loss is questioned there. Post-harvest deterioration is questioned there. The loss is questioned there. So unable to collect the quantities required by some buyers also, because the farmers trying to sell it as quickly as possible because of simply the high temperature. And then, so the recommended versus, as an example, I'm trying to show you here with some few countries, including India. So here, what is recommended temperature, or what is recommended temperature, and what is the measured temperature, and in case of tomato fruit. So if you look at here in India, of course, as everywhere, the recommended temperature for tomato storage or the transportation is just 15 degrees C. But at the farm level, you can see it is almost 10 degrees C higher than what is recommended. And if you look at the market level, so particularly the wholesale market level, it is exactly double the temperature. So this is where we are very much worried about the higher percentage of the post-harvest loss, that is after the harvest. If you look at the all developing countries, including Sri Lanka, this is the case, particularly at the retail or the wholesale market, the temperature is double than the recommended temperature for this harvested produce. So if you look at here, I'm trying to show you some situations, particularly these pictures are being taken during the COVID-19 period. It's so pathetic to share this actually. Maybe this is the situation in the other countries due to the lockdown during the COVID-19 period. So this loss, 50% loss, was increased dramatically because of the lockdown between the district to district, farmers to the wholesalers or farmers to the markets, farmers between the farmers to the transporters, farmers to the in between the farmers to the buyers. So there was a long period of the lockdown. So ultimately, the loss became double and double. It reaches up to 85 to 90% because the farmers couldn't sell at all their produce. Not even sell. The farmers didn't harvest. If you take the situation in our country, particularly in the northern east, which is under lockdown during the COVID-19 first wave, and even particularly the first wave, the farmers couldn't even harvest their produce from the plants, particularly the vegetables, even the fruits. They completely stop harvesting the produce from the plants because if they spend the money to harvest, they can't take it to the markets or they can't sell it to the buyers. So ultimately, at a point, they stopped even harvesting all these produce, millions of kilograms of the vegetables as well as fruits. Just it's left at the field itself without usage. Whereas, as I told you, in other side, we see the poverty problem. In one side, we see complete loss of the harvested produce or at this particular. So this is where you see how at the wholesale market or the transportation, very, very poor conditions at the transportations, very, very poor conditions at the wholesale market level, storage level in the developing countries. You could see this is why we are still facing up to 50% of the loss of harvested produce. Here again, I have taken some more pictures in Sri Lankan situation of the post-harvest losses at the wholesale market level or the to see how this, after spending the labor, the money, the time and everything, the poor farmers, how they face the problem. Of course, at a point, the economic problem. At another side, the lack of the availability of fruits and vegetables. And this, because of this, actually, if you take as an example, the COVID-19 pandemic period, particularly the first wave, the farmers couldn't sell for example 1 kilogram of tomato even for 5 rupees they couldn't sell, whereas in the other district we couldn't buy even for 200 rupees per kilogram a kilogram of tomato. So this is the gap. Why this is? That is because of course a particular period, but still due to this loss, the post-harvest, due to the lack of facilities, the high technological facilities, simply the cold storage or the cold room transportation facilities as in the developed countries, here we face the loss. And because of that, simply the prices are being increased at the retail markets. So again, in one side, we just simply throw the fruits and vegetables just due to the post-harvest losses in one side. In the other side, we just spend more money to buy the fruits and vegetables. So this is the situation almost in everywhere in the developing countries. So I have taken few newspaper articles as well. Here one from Indian one and another from Sri Lanka as 2 examples. India incurs rupees 2 trillion per year post-harvest losses of fruits and vegetables, which is from one newspaper. And if you look at Sri Lankan situation, rupees 18 billion annual loss from post-harvest waste. So this is the pathetic situation, very, very pathetic situation of the developing countries. I have just taken 2 countries as example, our country and our neighboring country, the India, whereas almost in all Asian countries, all the developing countries is the same situation. The billions of money is being wasted due to this post-harvest losses. So this is what the Economic Times says about Indian situation, the post-harvest losses, as I showed you. So developing wholesale markets in the situation in India, actually, developing wholesale markets together with enhancing the cold storage capacities of the facilities and regional markets, and in the district markets or the regional markets are key factor. It's a key factor. We are introducing the cold storage facilities. not to throw the produce because of the higher temperature at the market level, the deterioration of the fruits and vegetables at the market level. Why this is happening? Because the people or the farmers or the people at the market, the wholesalers, the retail sellers, they don't have the proper cold storage facilities, and due to that, if they couldn't day or two a week, they have to just throw it out. So this is leading ultimately the trillions of money of the country is wasted due to these losses. So if you look at the situation in Ghana, so this is where you can see how the wholesale, the markets conditions are here, how the wastes are here of the post-harvest produce. Not just the fruits and vegetables, it's our energy, it's our money, it's our immunity is thrown here, it's our energy is thrown here, our money is thrown here as the fruits and the vegetable. It's a very pathetic situation in developing countries. So despite everything we know, why have post-harvest practices remain so poor still? What can be the reasons in developing countries? So simply the reasons for the low adoption of the lack of initiatives, the lack of incentives at the market level, incentives for the farmers or at the market level. Again, lack of the resources, as I said, simply the cold storage facilities, proper transporting methods, or the incentives for transporting the produce by the farmers is not there as they expected, as they needed, because still we are developing countries. Again, lack of information among farmers, handlers, and marketers actually. Of information, say for example, the buyers, the wholesalers are sometimes they buy say for example a kilogram of tomato 10 rupees, but at the market level they sell it for 100 rupees. So this is the situation in almost all over the country, in Asian countries or developing countries, because in between the brokers or in between the lack of information between the farmers and the sellers or the wholesalers. So the policies, again, that in with the best practices have to be adopted. The policies have to be adopted. There are many other reasons as well. So ultimately, as conclusion, what I could say, the poor can be considered and it should be minimized. Why? Just to extend their storage life. Extend their storage life of the post-harvest produce. This is our prime, not just to throw right after the harvest or after a few days of harvest. We should not let the farmers or the market at the market level to throw our produce unnecessarily just because the problem what we face is the extending the storage life with the at least the minimum facility. But the problem is the simply the technology. Simply we talk as the post-harvest chain. During the try, it can be the harvest.
I express my deep sense of thanks to Chandrakanta ma'am for giving such a wonderful talk on post-harvest loss of fruits and vegetables. So it is a great information what one should learn. Not only doing a new research is important need of the hour, it is also very important to take care of the what we got from our hardships. So it is most of the students are attending this conference or meeting. So I hope that students have taken a message by looking the what is the loss we are doing for nature, how we are unnecessarily wasting so much amount of valuable things by eating so many kind of designed foods like sandwiches and all, without any antioxidants or something like that. So it is a great presentation, ma'am. I express my heartfelt thanks for your such a wonderful talk. Sure, I'll be in valedictory. I'll be summarizing the talk. Thank you for joining for this event. A big thank you, ma'am. And over to Deepak Parmar now. Deepak Parmar, you can start your thing. And Amar Natarajan, you can start your screen sharing. Over to Dr. Amaresan. Dr. Amaresan.
Thank you very much, sir, again for giving opportunity on behalf of Eastern University, Sri Lanka. Thank you so much.
Very kind of you, ma'am.
Thank you, ma'am.
Thank you.
Amar, you are not visible actually.
Yeah, 1 minute.
For a while, you can start your video so that we can introduce you.
Yeah, sir.
Over to Dr. Mr. Deepak, you can start the introduction of.
Yeah, sir. Thank you, Halim Khan, sir. Good afternoon to everyone. Once again, I welcome you all to this one-day national conference on plants and environment. And it's my privilege to introduce Dr. N. Amaresan, who did his Bachelor of Science in Microbiology from University of Madras. And then he did his Master of Science, that is MSc in Microbiology from May.
To everyone, once again I will.
From Bharati Darshan University, and he did his PhD in microbiology from Bharati Darshan University in year 2012. Then his sir specialization are molecular microbial diversity, plant microbe interaction are some of the specializations that he is working. And he joined as the lecturer in Kanchi Sri Krishna College of Arts and Science, which is affiliated to University of Madras, and he worked there from 2005 to 2006. Then he joined as assistant professor in C.J. Bhakta Institute of Biotechnology, which comes under Uka Tardasya University. He joined there in the July 2013. Okay, and sir is also the member of editorial some of the journals of the he's a member of editorial boards around 7 international journals. He is either the associate editor or he is a editorial board member in this 7 international journals. Then he also participated in many professional and research training institute. Then he has some 24 research paper, both international and at national level. And he was also, he wrote around 4 books on the microbiology. So it's again a privilege for Department of Botany, Giriraj College to have the eminent resource person as Dr. N. Amaresan. So I welcome sir to deliver his talk on microbial mediated mitigation of environmental stress in plants. Welcome, sir.
Thank you, sir. Am I audible, sir?
Yes, sir. You are audible, sir.
Yeah. Yes, sir. Sir, thank you.
To have the eminent.
Thank you very much, sir, for nice introduction. And before to my talk, I would like to thank
microbial mediated mitigation of environmental.
I get some disturbance. So before going to talk my today's title, I would like to thank organizers, especially Dr. Alim Khan, for inviting me to share my share our research work. So my today title talk entitled Microbial Mediated Mitigation of Various Environmental Stress in Plants. So all we know that the plants encounter a number of environmental stresses, which can be biotic stress or abiotic stress, which leads to a loss of more than 50% of crops. So nowadays, increase in world population, we have to find out alternative technology to improve the food crops. Then only we can feed the increased population. But what happened due to many anthropogenic activities, the environment and thereby the yield is lost or the reduction in the yield. So there are many stresses in the environment which cause or which impact the plant growth, especially the major the abiotic stress salination disturb the iron homeostasis stasis and membrane damage or a higher
Amar, your sound is a little bit disturbing.
transpiration water, then cause a water
Talking behind.
it is not audible?
No, some disturbances coming. There is some connectivity problem, it seems. Okay, now it is okay.
Sir, I'll connect through my mobile. Just
brain damage or higher
Redmi.
Somebody is listening to your presentation. There, somebody side to you.
It is not audible? No, some disturbances coming.
Amar?
Yes, sir. Is it okay now?
Yeah, it is okay now.
Okay. So the plants encounter various stresses as I described earlier, like the major stress.
Yeah, maximum full presentation mode.
Yes, sir, but it's not working.
Yeah, it has come now. Okay.
Yeah. So the major abiotic stress is salinity. Then it's drought stress. The drought decrease the photosynthesis efficiency of the plant, and also it inhibits the water transportation. Then if we see the look at chilling and cold effect, that decrease the biochemical reactions in the plants. Also, it decrease the carbon dioxide fixation, and the ice crystal mediated damage also taken place, and there will be free radical formation due to chilling and cold. If you look at highlight stress, where we can see inhibition of photosynthesis and increased photooxidation and elevated oxygen species. In the case of heavy metal, the heavy metal pollution leads to bioaccumulation of different metals in the root region of the plant. So thereby it inhibits the protein synthesis, as well as it retards the growth of the plants. So there are many abiotic stresses which affect the plants. But if we look at plants as its own mechanism to overcome these stress, but even that their own mechanism, the yield is always lost, or it's because the biomass is lost, or yield and the growth is reduction in the growth. Everything happens because of abiotic stress. So to manage this abiotic stress, the better option available at present is the use of plant growth promoting organism. So plant growth promoting bacteria helps plant directly or indirectly to promote plants. So the direct mechanism of the beneficial microbes includes nitrogen fixation in the plants, phytohormones production like cytokinin, gibberellins, etc. And then production of ACC deaminase, which reduce the ethylene synthesis. Then production of water-soluble vitamins. Also, many microbes solubilize insoluble phosphate into soluble phosphate that can be absorbed by plants. Then potassium solubilization. Then siderophore production, which chelates the iron molecules. So apart from direct mechanisms, the beneficial microbes also indirectly helps plant to survive against various stresses, both against abiotic and biotic stresses. So if we look at the indirect mechanism, the plant growth promoting bacteria increases the induced systemic response against various biotic and abiotic stress. Also, some organism produce antibiotics, some enzymes or siderophore, which makes deleterious to the plant pathogens. So how these microbes helps to mitigate abiotic stress. So when we inoculate a plant growth promoting bacteria or the plant growth promoting microorganisms which are present in the rhizosphere region, it acts differently against different types of abiotic stress. So if we look at drought stress, the plant growth promoting bacteria produces cytokines. The cytokine which act as antagonizing the abscisic acid. So in general, abscisic acid is a phytohormones, but excess production of phytohormones in the case of a drought condition, which leads to the inhibition of seed germination and post-germinative effect growth in response to environmental stress. When the organism produce cytokines, these cytokines act as a antagonizing the ABA mediated inhibiting effect. So likewise, same in the case of drought and salt stress, there will be production of antioxidants. The antioxidants may be enzymatic or non-enzymatic antioxidants, which inhibit the reactive oxygen species, ROS, the damage caused by ROS. So what happened? The production of ACC deaminase, which helps in the breakdown of a compound called ACC. This is the precursor for the ethylene production. So the ACC is converted into alpha ketoglutarate and ammonia. So thereby the production of ethylene level is reduced. So same way, some plant growth promoting bacteria produces volatile compounds. So these volatile compounds contribute to reduce the nitrogen uptake. Also, it translocate the sodium ions in the plants. So thereby the microorganism confer resistant against salt and drought stress. Also, some organisms produce IAA cytokines or some other unknown metabolites, so which alter the root morphology and the transporter activity. So thereby the increased accumulation of nitrate and phosphates can be managed. So there are many mechanisms the plant beneficial microbes possess, and these mechanisms always helps plant for their better growth, better yield, and better biomass. So in the few years, we have studied some research. Our research shows the inoculation of plant growth promoting bacteria or fungi enhance the plant growth in response to salinity stress or heavy metal stress. So I'll be sharing some of our research work recently carried out with respect to salt tolerant and heavy metal stress. So if you look at salt stress management, so this salinity is the major abiotic stress worldwide. So this salinity unfavorably affects crop production and pose a major threat to farmers worldwide. Not only in India, it's globally it poses a major threat. So if you look at the salinity stress, there are 20% of the land, the total cultivated lands are affected by salinity. And if we look at irrigated land, 33% of the lands are affected by salinity. So how we can manage this salt stress? The simple, the beneficial or cost effective method is to use beneficial microbes to manage or to mitigate salt stress. So some of our work recently carried out, this work was carried out in the Vapi district where we find the soils are alkaline in nature. We have isolated some set of organisms in the soils, and these isolates were screened against the screened for their salt tolerant properties. The bacteria which found higher salt tolerant capacity were selected and we put tested on cucumber seedlings. You could see here the control plant, the growth is reduced, whereas the plants inoculated with bacterial isolates, the root and shoot length were increased. Also, apart from that, increasing in their biomass, the inoculation of this salt tolerant plant growth promoting bacteria also enhance the uptake of nitrogen and phosphorus from the soil. So you can see these graphs clearly. The control plants stem and root as lower nitrogen and phosphate uptake, whereas in the case of bacterial inoculated seedlings as more nitrogen and phosphate uptake. So not only without any sodium chloride amendment, we can also see the enhanced uptake in the presence of 100 millimolar NaCl. So this result shows that you can look at the organisms identified in this. So you may ask, some may ask, all these are relatively human pathogens. So that's why the researchers always says that we need to go for indigenous organism for managing any abiotic stress. So if we take Surat district, you have to isolate a organism which are indigenous to Surat district that can be formulated for the managing any abiotic stress. So this paper we recently published in Journal of Plant Growth Regulation, and in another study with the same salt tolerant organism which was carried out in the Daman region, where you can see the Daman region is the cultivated lands are the alkaline condition is high in nature. So we collected isolates from Daman cultivated lands. Then we could isolate some set of organisms, and these organisms were screened against different plant growth promoting properties like indole acetic acid, phosphate solubilization, siderophore production in the presence of NaCl. So the results you could see here, even in the presence of sodium chloride concentration, the isolates used to produce indole acetic acid, solubilize phosphate, and produce siderophore production. There is a little reduction in the production, but you could see the it is not totally inhibited. You can see in the 2% of NaCl, there is 54%, 54 mg per ml production, but at 4% NaCl, it's a 50%, 50 mg per ml. And these isolates were inoculated with the rice seedlings under the salt stress condition. The results are very encouraging. We got encouraging results where the isolates enhance the root and shoot length and also improved the biomass of rice plants. So apart from enhancing biomass of the plants, the inoculated organism also supported in the photosynthetic pigments product synthesis like chlorophyll, also enhancement of proline content, which is a antioxidant enzyme where it acts as a metal chelator during the stress. So some of our some studies of salt tolerant bacteria were carried out in the Andaman Nicobar Islands after post tsunami. You all know that after tsunami, the salt water intrusion was more in the cultivated land. So many parts of Andaman Nicobar Islands cultivated lands are lost. So when I was working over there, we collected some soils, the cultivated land which are affected by saline water. So we could isolate some bacteria like Bacillus, Pseudomonas, and these organisms were tested for their salt tolerance ability against rice seedlings. So you could see here, in general, if you look at 4 deciments per meter, it is a or in general 4 EC, electrical conductivity. So in general 4 EC is not suitable for cultivation of any land. Okay, but here you can see here even at 5 deciments per meter, even at 10 or 15 also, the isolates showed some promising growth compared to control. Okay, so these isolates also increase the rice germination, also increase the biomass even in the presence of higher salinity concentration. So not only for bacteria, we also carried out some experiment salinity management with some fungal isolates, especially Trichoderma species. The Trichoderma species also showed a promising effect in enhancing maize plant in the presence of different concentration of different salt stress. You can also see the enhancement in the proline content, also enhancement in the total phenol. You all know that the phenolic compounds inhibits the lipid peroxidation and chelates the metals. Okay. So apart from this salinity stress, we also carried out some experiment with the metal stress management, especially in the Surat district, where you can see Tapi River is the major river which is used for the agricultural irrigation purpose. But when we analyzed the soil samples, the soil sample used to have a higher amount of copper, iron, and zinc. So all we know that copper, iron, and zinc are micronutrient in nature, but the excess amount of this zinc, copper, iron also cause toxic to plants. So then we thought that it is a one way of like a phytoremediation into biofortification. So where one way we are going to clean the environment, in another way we are going to mobilize these micronutrients into the plant pots, so that the plant is fortified with a certain amount of micronutrients like copper, zinc, and iron. So in a study carried out in the Tapi River region, so we have isolated a set of organisms over 250 microbes, and these microbes were screened for their tolerance ability against zinc, copper, and iron. Okay, So after based on that tolerance ability, we could select separately for a zinc resistant, iron resistant, and copper resistant. So the zinc resistant bacteria were tested in the presence of zinc amendment. So you can see our results where you can see even in the presence of 2 gram of zinc per kg zinc stress, you can see the improvement in the root length and shoot length. Also, even in the presence of zinc stress, the bacteria enhance the total phenol content, chlorophyll content, proline content, and ascorbic acid contents. You can see all these are antioxidant and non-enzymatic antioxidant and enzymatic antioxidant where which reduce the production of ROS. Also, you can see the inoculation of this plant growth promoting bacteria enhance the zinc uptake in the plants. You can see here clearly in the presence of zinc, the microbes which are resistant to zinc tolerance mobilize the zinc into the plant pots like roots and the stem. So that way, one way it is fortified with zinc content. So it is a coin with two sides where one way it is a cleaning the environment, another side it is a fortification of important or essential micronutrients into the plants. The similar results also observed for the iron tolerant plant growth promoting bacteria in the brassica. So these iron tolerant bacteria were belonged to the genus Bacillus rhodococcus licinibacillus species, and this iron tolerant bacteria also showed production of plant growth promoting properties like IAA, siderophore production, potassium solubilization. And these isolates were inoculated in the controlled study by using brassica plants. So you could see here, even in the presence of higher iron concentration, the bioinoculants used to enhance root length and shoot length. Also, it increase the antioxidant properties in the plants. So where you could see the enhanced phenol concentration and proline concentration in response to iron stress. Also, the inoculated organism enhance the iron content into the plants. You can see all the plants, the microbes mobilize the iron which are present in the soil, mobilized into the root system, stem system, and the leaf system. So this experiment is not carried out in the artificial inoculation of the iron content. This experiment was carried out with the natural environmental soil, where we brought the soil from the Tapi River region and set up the experiment. And there is no additional amendment of the iron. You can see here without additional amendments, whatever the iron present in the soils are mobilized into the various parts of plants like root, stem, and leaf parts. So this indicates that the plant growth promoting bacteria not only enhance the plants against iron stress, also it mobilizes the iron into the plant parts. So same way, the similar results was also observed for the copper accumulation, where we identified Stenotrophomonas multifila as a potential copper resistant bacteria. You can see here the bacteria inoculated with copper, which enhance the plant growth compared to control. So also it improved the chlorophyll content. Also it increased the copper accumulation in the plants. So in all three cases, you can see these three micronutrients the higher means excessive amount of these micronutrients cause toxicity to plants, but inoculation of uh, beneficial microbes reduces the toxicity caused by micronutrients, as well as the inoculants mobilized these micronutrients into the plant pots. So as I already told, this phytoremediation and biofortification, it's a coin with two sides. So this technology could be used for cleaning the environment as well as fortifying the important micronutrients. Some other experiment also carried out with the heavy metals like chromium tolerance. So this experiment again carried out with the Vapi region, where the soils are heavily contaminated with metals. So we have collected soil samples, and the soil samples were used for the isolation of bacteria, and these bacteria were screened for chromium tolerance. The bacteria which has chromium tolerance properties along with the plant growth promoting properties were further selected, and these isolates were tested under pod studies with chickpea plants. So you can see the results. There is an enhancement in the root and shoot growth even in the presence of chromium stress. Also, the inoculation of chromium tolerant plant growth promoting bacteria increases the photosynthetic pigments also, which helps the chromium accumulation in the plants. So this we cannot use it as this experiment cannot be used for what to say edible plants. This can be used for non-edible plants where for the cleaning of chromium polluted environments. Similarly, we have tested for cadmium mobilization also in using Sesbania bispinosa. The results are very encouraging, and the isolates which are resistant to cadmium stress promoted the growth of Sesbania bispinosa root and stem. Also, it increased the other antioxidant properties like proline, phenol, ascorbic acid, catalase. Apart from that, the isolates also helped in mobilizing cadmium into the plants. So these are some of the research we recently in the past few years we carried out. So in conclusion, what I would like to say is that the microbial mediated mitigation of any biotic stress or abiotic stress is viable, and it's cost effective, and this can minimize the other the environmental damage which are created by using physical method or chemical method. Also, it is very important to search indigenous flora for cleaning the environment. So we cannot because say for example, if I collected I have good number of isolates with me, but if we use these organisms in other parts of India, it may not work because it may not survive in the different soil condition or different environmental condition. So it is always we have to look for indigenous microbial flora for mitigating any biotic stress or abiotic stress. So I would like to thank organizer for giving me opportunity. Thank you. Is it?
Dr. Amar, it was a very big research work what you have carried, and I could see that very big list of inoculants you have tried, like some of the bacteria, and mostly you have done work on bacteria for different kind of stress. So surely I'll be reporting your findings in a summary of the valedictory, in the summary in the valedictory. So it was a kind enough, you were very much hesitant that you can't able to present the things, but you see so much you have presented. I am very shocked. All these are the papers you have reported. I was keenly looking. 2020 only you have published so much. You have published zinc resistance, iron tolerance, copper accumulation, chromium, cadmium, and salt. So much work you did here. Really, you told that you will be not able to do, but see, you did a very big job. Actually, you are marvelous. Really, you rocked the thing.
I didn't do, sir. It is because of
Whatever it
my,
is, but
my students.
you are the group. You are in
My
the group.
students are pillars.
You have published in Springer, you have published elsewhere, you have published in Taylor and Francis. Such a big personality you are, really. Thank you so much, Natarajan Amaresan, actually, for the kind acceptance of the request. And thank you so much. As we are going for the next speaker, thank you so much, Dr. Deepak. And I request Maqbool Ahmed sir to please start sharing the screen. Maqbool Ahmed sir.
Okay.
Hello.
Maqbool Ahmed sir.
Yes, sir. I'm sharing. Just
over to Deepak.
Yeah, once again, good afternoon. It's my privilege to introduce Dr. S. Maqbool Ahmed, who is Associate Professor in Manu University. Dr. S. Maqbool Ahmed has completed his MSc and PhD from Regional College of Education, Barkatullah University, Bhopal. He is presently working as Associate Professor of Botany with an additional responsibility as a nodal officer of Bachelor of Vocational program in School of Science. He also worked on different academic positions like OSD examinations and overseas coordinator, in-charge coordinator of UGC schemes, and in-charge horticulture officer, and he also worked as head department of botany. He was the recipient of Best Teacher Award from former Chief Election Commissioner of India, Mr. T.N. Seshan. He has teaching and research experience of 25 years, and before he joined the Manu, he worked as research fellow in Regional College of Education, Bhopal, in one project which was sanctioned by CSIR NERI. He also worked as lecturer in botany in Regional Institute of Education, Bhopal. Dr. Maqbool Ahmed has visited Kingdom of Saudi Arabia for conducting convocation, that is certificate distribution ceremony of the Manu students. He has published number of research articles in national and international journals, and also presented papers in many seminars. He has published 3 books, and 2 more books are under the process of publication. His research areas are plant physiology, plant ecology, and environmental science. Now I invite Dr. Maqbool Ahmed sir to deliver his talk on the world of mushrooms. Welcome, sir.
Yeah, thank you, sir. Audible, sir?
Yeah, sir.
Yeah, is it audible, no?
Yeah, sir. It is audible, sir.
Yeah, thank you, Deepak, sir. Good morning. Sorry, it is almost good afternoon. Good afternoon to all. I profusely thank the Department of Botany, Government Giriraj College, even Council for Green Revolution, CGR, and Department of Botany, Telangana University and especially when I thank my friend, my
आपका स्क्रीन पावर पॉइंट पे नहीं है सर, जूम मीटिंग पे है आपका स्क्रीन शेयर हो रहा सो।
हाँ या
और आपका कैमरा भी आपका मतलब करेक्ट नहीं है, फोकस कैमरा फोकस आपका करेक्ट नहीं है, आपका हेड आधा ही, हाँ आप ठीक है। अभी स्क्रीन का देखिए सर, स्क्रीन चेंज करिए
ना,
नहीं अभी स्क्रीन चेंज नहीं हुआ आपका, जूम का ही दिखा रहा, जूम क्लाउड मीटिंग्स का स्क्रीन दिखा रहा।
अच्छा अच्छा 1 मिनट बेटे ये देखो तो सिर्फ कम या so what I should do sir?
आपका पावर पॉइंट वाला जो है उसको शेयर करिए,
पावर पॉइंट शेयरिंग या नाउ
अभी भी नहीं चेंज हुआ, इसको क्लोज करिए, जूम को क्लोज कर दीजिए,
जूम को क्लोज कर दो, जूम को क्लोज करना है
या मिनिमाइज करिए उसको,
जूम को क्लोज नहीं करूं, जूम को
मिनिमाइज करिए, शायद आप आउट मतलब साइन आउट हो जाता, जूम को मिनिमाइज करिए, हो रहा है यहाँ पे, हाँ इसको मिनिमाइज कर दीजिए।
नाउ सर
आप शेयरिंग एक बार बंद करके फिर से शेयरिंग चालू करिए सर।
हाँ वन मिनट शेयर करो सर, स्टॉप कर दो ये वाला, स्टॉप शेयरिंग ओके, share screen right okay then this also on this side come now sir
अभी शुरू नहीं हुआ सर अभी, हाँ आ गया, अब आ गया, राइट राइट चलिए सर आप चालू करिए, बिस्मिल्लाह।
या आ गया सर ना,
आ गया
या thank you very much sir. So thank you professor, my colleague, my friend Professor Ahmed Abdul Halim Khan sir for inviting me to this international webinar conducted by in collaboration with CGR and Telangana University, that to government Giriraj College. And my today's topic is I'll be discussing about biodiversity of mushrooms and so importance of mushroom and its nutritional, medicinal properties or values. This mushroom, as we know that they are the fleshy fungi and macroscopic, which belongs to Bestiomycetes class, and we all know that fungi, these are the special group of plants which do not have chlorophyll, so they are unable to synthesize food material, so they may be parasitic in mode of nutrition or saprophytic, or sometimes they show symbiotic mode of nutrition also. As far as a species is concerned, basically if we say estimated species of mushroom on earth, more than 1,40,000 species are there, but out of which only 10% are known species. So 10% means it comes to nearly 14,000 species. So we can say that in the world nearly 14,000 species have been identified, and out of which nearly 2,800 to 3,000 are edible in nature, and nearly 250 having medicinal properties. Whereas production is concerned about mushroom, as per the data of Food and Agricultural Organization, some countries which are producing mushrooms in the world, leading countries I can say later on, but here you can see China, Iran, Italy, Poland, Netherlands, United Kingdom, France, Spain, and the larger amount even you can see in the parts of United States of America and in Canada also, mushroom production is there. But three major mushroom producing countries in the world are, this is the world, the first topmost leading country in the world, that is China. Second, United States of America. And the third one is Netherlands. The above three countries itself produce more than 60% of world production of mushroom. But if we see the share of this single country, China,
सर आपका आवाज म्यूट हो गया सर, अनम्यूट करिए.
yeah sir.
हाँ, और स्क्रीन मोड पे रखिए, फुल
So yeah, sorry, I'm extremely sorry. So I was discussing that estimated species of mushroom on Earth, nearly 140,000 species are there, but only 10,000 species are known to us. So that means here together I have shown here a picture that in the world at present we can see that 14,000 species which have been identified, out of which nearly 2,800 to 300 are edible in nature, and 250 species are they are having a medicinal importance as well as medicinal value. When we talk about mushroom producing countries in the world, as far as Food and Agriculture Organization FAO of United Nations, it says that these are the countries which are producing mushroom in the larger extent, like China, Iran, Italy, Poland, Netherlands, United Kingdom, France, Spain, and larger area, larger parts of even United States of America is also producing mushroom in a larger extent. Even you can see here, Canada also, Canada is. So three major mushroom producing countries in the world. These are this China, Netherlands. These are the three more important countries where we get nearly 60% of world production of mushroom share is coming only from these three countries: China, Netherlands, and China, Netherlands, and United States of America. But from this single country, if we see from this only single country, China, mushroom production or the share of production, it is nearly 46% of share is there. That means you can imagine that how much production in the world China is doing. This is a very great job. They not only producing, they're doing high kind of research. They are using medicinal mushrooms in medicines. So this China, it stands top in the world in production of mushroom. But as far as country mushroom production, it is nearly 3 to 4% is there. And hope I think the data might have the percentage might have increased now. But as far as position is concerned, position maybe India is coming on 13th or 14th position. So this is the what you call the graph I have shown here, Odisha, Punjab, even Haryana, these are the states where they are producing a good percentage of mushroom production, even Himachal Pradesh as well as Uttarakhand. But very low production is there in southern states as well as western states. So when we see the production in our country, in the northern belt, this is what you call near Uttarakhand as well as in the Himachal Pradesh area and some parts even in Jammu Kashmir, you can say, button mushrooms are grown. Button mushroom, which is known as Agaricus bisporus, scientific name, and this is a top production of button mushroom in our country, and this is the in the northern belt. And in eastern area we have a very good mushroom production that is known as shiitake mushroom. Shiitake mushroom, this is Lentinula edodis, scientific name Lentinula edodis. And in the central part of our country, like in the Chhattisgarh, even Orissa, and some parts of Madhya Pradesh and some other states, like in the central India, we have the production of a pleurotus species of mushroom, that is oyster mushroom. You can see the picture of oyster. It is a very beautiful, it looks very beautiful in different colors. And when we see about southern belt, in the southern belt, in the eastern southern as well as the western southern areas, we have the production of two different types of mushroom. One is known as a milky mushroom, which is known as Calocide indica, and milky mushroom as well as this Valorella volvacea, paddy straw mushroom, both are grown in the southern part of our country. And as I just said that button mushrooms, Agaricus bisporus, its production is very high in our country. 30% production, it is more than 30. This is of Agaricus bisporus. And when we talk about pleurotus species, that is oyster, it is in second rank, second position. And in the third position, shiitake mushroom production is there. So I'll be talking about one biodiversity issue, that is mushrooms biodiversity. Some interesting mushrooms you might have seen in what you call even on websites, different websites. This is what you call named as bird's nest mushroom. It looks like the nest of a bird, and inside that nest you are observing small white egg-like structures, but basically they are peridioles. These are eggs or the peridioles, and these mushrooms, bird nest mushroom, actually it is a cyathin type fungi members, and they belong to Nudulariaceae family, and they are not edible category. They are inedible in nature, and largely found in the European countries. And nowadays it has become endangered also. And this is about the bracket mushroom. Uh, Many people might have seen on the logs on the stem. This is what you call. It looks like a shelf. It looks like shells. That's why this bracket mushroom is named as shelf mushroom or shelf mushroom. And the fruiting body, this is the fruiting body, is also known as conchs. Conchs. So this is a group actually polyporous fungi, and you are observing the lower surface white structure. This is the main what you call spore producing structure, from where you can see that this is the spore producing center, and from this area what you call bestiospores are produced, and it is a bestiomycetes member. And in Japan, even a certain type of bracket mushrooms are used in medicine purpose, even in Japan. This is the another kind of mushroom. It looks like cauliflower, and This is also called as parasis, and it is a parasite, and even some saprophytic in nature also there, and it looks like head cauliflower or a brain-like structure. And this is another kind of mushroom. It is a Ramadia botrytis. It looks like a cluster of corals. Coral. It looks like coral. That's why it has been named as a coral mushroom. And this is what you call. It looks like a black ball-like structure. Basically, it is used in an old folk remedy for night cramps. So that's why it has been named as a cramp ball mushroom, and it also named as Galdenia, or else it is also named as cramp balls also. Okay, it's grown on even decaying wood. And very interestingly, this picture, it looks very differently. This is, of course, you are noticing, you're observing that this finger-like projections are coming from the ground level. Basically, this is a saprophytic mushroom, saprophytic fungi, grown from rotting or injured, what you call trees or stumps. And it looks like dead man fingers. You can see it. These are the what you call apex terminal part. It looks like what you call fingers, and this is actually Xyleria polymorpha, and this the common name is the dead man's mushroom. Now come to the another category. This is the most poisonous mushroom. So this, whenever you see some yellowish color is there, so you can blindly say that this is a poisonous one. But it is very difficult to differentiate what you call the edible with the non with poisonous also. Only experts can say that this is a poisonous and this is edible. But edible, we have already some hundreds of species we have identified, so need not to worry. We can have these edible mushroom. But this is what you called this is known as Amanita phalloides. This is the most poisonous mushroom. In this genus, Amanita, some species are there like Amanita phalloides, Amanita verna, and Amanita virosa. These are the most poisonous mushrooms. Even a single bite can lead to death. Directly, it attacks to the nervous system. It attacks to the brain, and on the spot, death may occur. Even you might have heard the story of Shakespeare Hamlet, where King name, what you call Claudius name. So Claudius' death occurred due to this mushroom. There is a long story. I am not going to discuss this in detail because we have given a very short time to deliver a lecture. So this, at least you should have an idea that King Claudius' death occurred due to this mushroom, death cap mushroom, which is Amanita. And another, this is what you call the earth tongue mushroom. Sorry, this is, yes, earth star mushroom. This mushroom fruiting bodies, it is like a stomach shape. It is like a stomach shape, and that's why it is also named as a stomach fungi. And actually, these unfolded structures, they cover this spherical body, stomach structure. Whenever water falls on this fruiting body, and these outer layers, which are known as peridium, this peridium split or it uncurl and forming star-like structure with nearly 4 to 12 ray-like structure. That's why it has been named as earth star mushroom. And this is another kind of mushroom, it is in the northern Africa, Asia, and Europe areas, which is black in color, and it is earth tongue mushroom. And very interestingly, another is there. It looks like a bridal wheel. It looks like a bridal wheel. You are noticing here. This is the wheel-like structure, and commonly called bamboo mushroom. This is commonly called bamboo mushroom. And this wheel, you know, this is a skirt. It looks like a skirt, a lady's skirt, and this is a white color, and this is a yellow wheel or yellow what you call the lady's skirt. It looks like a lady's skirt. So these are of different colors of Phalaenopsis species, and this is also you can say poisonous mushroom. And again, you have when we see the diversity, plenty of mushrooms are there. Only an interesting one. This is a lattice shape that is named as a basket stinkhorn mushroom. It is in northern Europe grown. What you call northern Europe, it is present in the forest areas, lattice mushroom. And this is also another lion's mane mushroom. See, this is what you call, it is edible in nature, and it has a medicinal value, medicinal property in the Northern America area. It has a very good production of medicine in Chinese. So whenever you say that whenever some hair-like structures appear on the face, which is named as a mane. So hair-like structures are present in this mushroom. That's why it is named as lion mane mushroom. Okay, and this is normally you might have seen in the pictures in even what you call small story books. Also, you might have noticed this is fly agaricus. It has an ability to attract the flies. This mushroom, it has an ability to attract insects and flies, and it kills. So that's why it is named as fly agaricus. So this is it looks very beautiful. It is beautiful in color, and that's why it is named as fly agaricus. And another one is the stinkhorn mushroom. They are known for foul smell and also for sticky spores. And this is another one is a trumpet. It is a trumpet shape. You all know that one musical instrument comes, it is a trumpet shape, and it is the shape of this mushroom is trumpet shape. That's why it has been named as trumpet mushroom. It is a pleurotus species and edible in nature, and even found in the Middle East area, Northern Africa, and in cool climate areas they are found. And this is finally umbrella shaped mushroom. It is also known as ink cap mushroom. This it appears, you know, in grasses in overnight. You know, in overnight, within a short span, it appears, and within 24 hours it decay. Also, it disappears. That means it leads to a death. So overnight following day, that is in 24 hours of rains. So this is the another type, and this is the finally you can say that auricularia species. Also, this is edible in nature, known as wood air mushroom. Now to nutritional value. This is the important part. What I'm going to discuss here. Actually, mushrooms are greatest, I can say that it is an untapped resources of nutrition and palatable food of the future. Because mushrooms have very high copper levels, It helps to reduce even colon cancer. The last speaker, he was even talking about minerals. I'll be giving some more stress on this part later on. So mushrooms, it contain nearly 80 to 90% of water and 8 to 10% of fiber. And this is, I can say, excellent source of vitamin C and vitamin B. So all earlier speakers were discussing about the last 1 year pandemic situation. So during this pandemic situation, every doctor, every person was discussing, saying about the immunity power. So these are immune boosters. I can say that mushrooms are very much immune boosters. They have rich vitamin C, vitamin B, and also vitamin D. No other food you find vitamin D. You never might have heard about vitamin D in the food item. People generally say, go and sit in the sunlight, you will get vitamin D. But here, as I said, that nearly 80 to 90% of water and 8 to 10% of fiber, and it is a source of vitamin C, vitamin B, especially folic acid, thiamine, riboflavin, even niacin is also there. And when we talk about minerals, mushrooms, they are rich with potassium, sodium, even copper, phosphorus, magnesium, zinc. All these minerals are present: potassium, sodium, phosphorus, even copper, zinc, and magnesium. So this is a zinc. Zinc is a very, very, very important mineral, I can say here. And even I have shown here selenium. I think selenium is missing here somewhere. Yeah, selenium. So selenium, it is in the form of selenoprotein found in mushroom. It is having anti-cancerous property. Anti-cancerous property. So in the next slide, I will show how the mushrooms are having importance in the medical field. So mushroom effective even against, you can say, cancer, in cholesterol reduction, stress, insomnia, asthma, allergy, and so on. So this is where I would like to say that Indian diet is primarily based on what you call cereals like wheat, maize, and rice, which are deficient in proteins. But you can say that mushrooms have a very rich amount of proteins, very good amount of amino acids like tryptophan and lysine, which are present in rich amount, tryptophan and lysine. And just now I was discussing that mushroom is the only crop which contains vitamin D. Means mushroom means vitamin D means basically directly you can say we don't have vitamin D. Actually, it has an ergosterol. It is a precursor of vitamin D. Whenever this ergosterol enter into the human body, it converts into vitamin D. As we say that what you call carrot, carotene is there. When enter into the human body, this carotene converted to vitamin A. So same thing here. I can say that this is an ergosterol. Ergosterol is present. It is a precursor of vitamin D. When enter into human body, they convert into vitamin D. Vitamin D. Now come to the benefits of mushrooms. As here it has been mentioned that first come to cereal order, I can say it improve digestive health, it reduces BP and prevent even various types of cancers.
Sir, your voice is lost. We are not able to listen to you, sir. Mahbul, sir. Mahbul, sir, check it to the end for sound. You are not getting a sound.
Level and yeah, sorry, some disturbance was there. Now, is it audible? No, Dr. Ahmed, sir?
Yeah. It is audible.
Yeah. Now, just I was saying that about I don't know how it has gone. Yes, sir. Yes, sure. Once again, I have to. This side. Yeah. Sorry. So here I was saying that. Sir, it is audible, no?
Yes, you are audible, sir. Right.
Yeah. Sorry. So I was saying that it is playing a vital role in reducing blood pressure, and it has been noticed that pleurotus species, that is of a soger kaju, which is oyster mushroom. In oyster mushroom, lovastatin is found. Lovastatin will help in reducing what you call blood pressure. And when we talk about potassium as well as the sodium ratio, potassium and sodium ratio, it is nearly 110 to 1%. That means 110% of potassium is there in this sodium, very meager amount of sodium is present. That means we can say that potassium, it lowers the blood pressure to stay healthy. So people need more amount of potassium to control blood pressure. So if we eat the more potassium you eat, the more sodium it will process out. Even mushrooms, we have very little amount of sodium. And when we talk about parasaccharides and some other of bioactive compounds are there, like polysaccharides, diterpenoids, glycoproteins, even immunomodulating compounds are also has been noticed. So we can say that mushrooms promote immune system, or it boosts health also. And here it prevents various types of cancers. Among various types of cancer, prostate, it has been studied in the various research. It has been found that it prevents prostate cancer, because fresh mushrooms are capable of arresting action of 5-alpha reductase as well as aromatase enzyme responsible for growth of cancerous tumors. Okay, and next slide. This is I can say this is a very interesting Himalayan vigra, which is under threat. You might have seen in the newspapers recently. This is actually, this mushroom is not saprophytic. This is a parasitic mode of mushroom. This is very interesting. Parasitic mushroom, basically it grow on a caterpillar. So you can imagine like capsicum. It looks like a capsicum and a small what you call petiole-like structure. It has been noted. I'll discuss. This is actually this caterpillar. It is found in the Himalayan areas. It is in the elevation of more than 11,000 feet, which is known as caterpillar mushroom. So, for growing this mushroom, or it requires temperature, very low temperature. It requires winter temperature below even 0 degree, and this is what you call it is commonly named as Himalayan vigra, but the scientific name of this mushroom is Cordyceps sinensis or Ophiocordyceps sinensis. And in the northern belt, it is known as Kida Jadi, also Ersagomba or Jivan Bhuti. Sometimes it has been referred as Kida Ghas, caterpillar fungus, and even people called as a Sanjeevani Bhuti. Sanjeevani Bhuti. Sanjeevani Bhuti, you might have heard about the name. So this is what we call how this is spreading. This mushroom what called attack on the insect body, and this is what we call the spore producing area. Whenever spores are produced, and these spores directly it attacks the caterpillar, and you are observing here infected larvae is it moves below to soil surface and facing its head upwards, and small projection starts appearing from this area, and this fungus grow out of the inside. This is the fungus is growing.
Because of his network issues, we are facing difficulty, it seems. Sir, can you resume your...
It's not moving. Yeah. So this is what I was saying, that this is what you call caterpillar mushroom. This mushroom is on the caterpillar, and finally it leads to death of a caterpillar. Caterpillar will die, and this is the protruding structures which comes out from the mushroom or the caterpillar body. And I have mentioned here that which is named as Himalayan vigra. It is also known as Himalayan vigra, and it is having a very important medicinal value. It used to cure cancer, as an anti-protein drug in China as well as in the world. It has a very high market. Internet it is there, because 1 gram of powder of this cordyceps, it is nearly fetched to 3,500 to 4,000 rupees. So it is like more than gold price. 1 kg of powder of this what you call Himalayan Kida Jadi mushroom, it is in the market nearly. Okay, I'll stop sharing. I'll continue with this only, because only 2-4 slides are there. So I was saying that it's having a medicinal value. It is used to cure cancer. It has anti-potent drug. Even athletes were using this mushroom, because in doping test this will not be traced out. This what you call drug. And we in our lab we are even growing this mushroom like Calocide indica, and we have started doing research on this mushroom, and hopefully we are going to set up a laboratory also in future. So what I would like to say that mushroom is considered to be a complete health food suitable for all age groups, from child to even aged people. And in mushrooms, what you call pharmacologically active compounds are present. It is showing anti-microbial property. It has antiviral, anti-tumor, anti-allergic, immunomodulating, and anti-inflammatory, anti-therogenic, and hypoglycemic, and hepatoprotective. So all these properties we can find in mushroom. So finally, I suggest, I would like to say that mushroom can prevent and treat many diseases, and even researchers have a very good scope to do research on this area, and it boosts even overall health. So as well as it helps to heal complete planet. So I recommend and I hope you also recommend to eat mushrooms. So thank you very much, and also I apologize for interruption. A lot of interruption was there. And finally, I thank my colleague, my friend, Professor Halim Khan Saab, as well as I thank Giriraj College, as well as CCG, for providing me an opportunity to speak on this international seminar. Thank you, sir. Thank you, one and all.
Thank you very much for giving us beautiful insights of mushroom, the macroscopic forms of fungi. It was wonderful to listen the whole colorful world of mushrooms. So we are 1 hour late of the schedule. So quickly we'll get back. So I request Dr. Madhusudan Reddy sir to share his screen, and I request Dr. Raviraj to start giving introduction of Madhusudan Reddy sir. Madhusudan Reddy sir, you can start sharing the screen, sir. Dr. Raviraj. Dr. Raviraj. Sound problem is there, Dr. Raviraj. Please, you check your background. Yeah, you can start introducing, sir. Sir, you can start sharing the screen, sir, by the way.
I'm sharing, but it's not showing my desktop. Wait one second.
Meanwhile, you can start, Raviraj sir, because already we are delayed by 1 hour. You can start. I have pinned him. You can start. Raviraj sir. Disturbance minimize చేయండి Raviraj sir. Raviraj sir, introduction start చేయండి sir ది.
ఇది ఉండని నా దాంట్లో
It's a proud privilege to introduce our next speaker, Dr. A. Madhusudana Reddy, sir, Associate Professor, Yogi Vemana University, Botany.
Okay.
Dr. Madhusudan Reddy, sir, is Masters in Botany from SK University, Anantapur, with the plant physiology as a specialization. He conferred his PhD from SK University, Anantapur in plant taxonomy, especially in grasses of AP. His areas of research interests are plant taxonomy, biodiversity conservation, medicinal plants, and lichenology. Positions and honors of Dr. Madhusudan Reddy, sir, are: he was visiting research fellow in Chemtech's Environmental and Industrial Laboratory, Texas, USA, during 2007 to 2013 April, and he was he was acquired a position of biologist in the same laboratory during 2004 to 2007. He was assistant professor in Nagarjuna University PG Center, Guntur and Ongole, during June 2003 to May 2004. Honors and awards: Biodiversity Conserver Award 2019 received from AP Biodiversity Board in view of World Biodiversity Day in 2019. Vanam Manam Appreciation Award 2018 announced by AP Forest Department, received from Prime Chief Minister of Andhra Pradesh in 2018. Andhra Pradesh Greenery Award 2017 announced by AP BUDC, received from Chief Minister of Andhra Pradesh 2018. Best Plant Conservationist Award 2017 announced by AP Forest Department, received from Kadapa District Collector on July 2017. Associate Fellow, Andhra Pradesh Academy of Sciences, March 2016. He was visiting research fellow in Texas, USA during 2007 to 2013. Young Scientist Award during the International Seminar on Medicinal Plants and Herbal Products by SV University, Tirupati in 2010. He was conferred with Dr. S.R. Yadav Award at Annual Conference of Indian Association of Angiosperm Taxonomy, University of Calicut, Kerala in 1998. Best Teacher Award in 2011 honored by SRI Madasi Venkatappa Memorial Trust, Galividu, YSR District. New taxa described are 4 and 10 are in progress. Brachystelma Vemane.
Sir, sorry interrupt you. That's okay. Enough. I can start my way.
1 minute. I will finish up. So he was Stigmatochroma microspora, Diorygma carnulensis.
Diorygma.
Diorygma. The thing is, ongoing projects to his credit are ex situ conservation and propagation of medicinal plants through model nursery by Ayush, Government of India, New Delhi, and APMA PB NMP. Ex situ conservation and propagation of threatened and endemic plants of Eastern Ghats of India. Then biodiversity assessment and prospection of lichens in Rayalaseema forests of Andhra Pradesh, India. Then ex situ conservation of wild medicinal plants, Agri Science Park project sponsored by Ministries of Industries and Commerce, Government of Andhra Pradesh. Large-scale cultivation and commercialization of elite varieties of banana for Rayalaseema region. He has number of guest lectures and he organized a number of conferences and symposia. With these few words of introduction, I would like to request Dr. Madhusudan Reddy, sir, to give his talk. Thank you, sir.
Thank you, Dr. Sir. I am audible, right?
Yes, you are audible.
So my screen share is okay, right?
Yes.
It's looking right?
Yes.
Okay.
You start slide, this mode.
Oh, okay, right?
Right.
Good afternoon to everybody. It is too late. I'm sorry. I'm going to click on within my stipulated time. I'm very much thankful to the organizers, especially International Conference on Plants and Environment. This is a really wonderful topic, plants and environment. It's a recent times, especially Corona times, we have to discuss on plants and environment. This is really very useful to all, and also we have to look on plants and environment, how it is going on in our surroundings. So what is happening in our future also, what is the new things coming out, and how threat is coming to our human life. So recently we faced a threat to everybody is scared about our threat, how we are going to face this problem. So like this, these people are conducting plants and environment organized by Department of Botany, Giriraj Government College in collaboration with the Council for Green Revolution, Hyderabad, and Department of Botany, Telangana University. I am very much thankful to you all for inviting me this international seminar. So here I'm going to speak on practical oriented things which I have done last 10 years when I am joined in the university as a botany assistant professor. Then now I am associate professor. What I've done and what I contributed to the university and what I contribute to the nature and what I contribute to the extensive conservation and especially on endemic and threatened plants. So before going to my topic, this is brief on Eastern Ghats endemics and threatened plants and Eastern Ghats status. Then real picture I'm going to do what I've done last 10 years. I will show you that things. So my title is Extensive Conservation of Endemic and Threatened Plants of Eastern Ghats of India. So what are the plants are endemic and threatened plants are present in Eastern Ghats, and what is the threat phases are facing, and so what is the suitable things to conserve the endemic and threatened plants. So I did my little role to conserve the some of the plants in our botanic garden. Also rehabilitated the various places in botanic gardens. I distributed the various institutions also. So with this, I will start this my lecture. So this is the satellite map of Eastern Ghats. Eastern Ghats, especially, you can see the states which are connected with the Eastern Ghats. And when you see the rainfall, tropical, and also the temperature in Kadapa, we are also facing 45, 46, 47 also. And water resource, water resource, rivers, there are several rivers and waterfalls, also numerous waterfalls, and also natural springs also many are found in the Eastern Ghats area. So when you see the vegetation types, evergreen forest, semi-evergreen forest, moist deciduous forest, dry deciduous forest, dried savanna forest, scrub, dry evergreen forest. Why I'm showing these things is, so with this various kind of natural habitats, we brought the threatened endemic plants to our extensive conservation centers. So these are some example for the vegetation types, and these are some of the other examples. And plant diversity in Eastern Ghats, pteridophytes is nearly 120 plus, gymnosperms are 8, angiosperms are 3000 plus. Total flowering plants 3200 plus. Still new species are coming every day, every year coming and the number is increasing. And cultivated and domesticated 185 species, and medicinal plants more than 1800 species. Die yielding plant species are 50. Aromatic plants are still the number is increasing. So dominant families, and when you see in the Eastern Ghats area, dominant families, Poaceae is one of the highest and richest family in the among the angiosperm plants. Poaceae and followed by Fabaceae, Cyperaceae, Euphorbiaceae. And when you see the genera wise, Crotalaria in Fabaceae is the highest number of taxa, and Cyperaceae is 20, and Lucas followed by the other species also you can see. And these are some of the ethnic groups present in the Eastern Ghats area. Various Chenchus, varieties. There are several people. These people are really wonderful natural conservation people, and also they are not going to harm to the forest. So these are some fauna, and these are some of the other life. So Eastern Ghats face mass extinction. So Eastern Ghats has decreased by almost 200 square kilometers between 1920 and 2015. You can imagine 1922 2015, 32,200 square kilometers area of the Eastern Ghats has decreased. The overall forest cover spread over 43.4% of Eastern Ghats in 1920. So reduced it to 27.5% by July 2015. So the area to have a last maximum green cover are Mahabubnagar, Telangana, and Gajapati in Odisha. So these are the two places maximum green cover as every day is going on. Very less is going to take the, there are several threats. So it's not just loss of green cover, plants and trees endemic to the Eastern Ghats. Hello.
Sir, your slides are moving, but you are not visible, sir.
So my power lost from the monitor.
You're going ahead of your slides all. So you're not
Oh.
able to see your slides.
The slides are moving, huh?
Yeah,
So
moving.
I'm not.
Cleom viscosa.
No.
Please bear for us because we are waiting for sir to join back because of his I mean to say some issues. He couldn't power failure or what, we don't have any idea. Please wait by for a while and we'll be getting back to you again. Yashwant, keep monitoring. Sir may be joining at any moment because he was having a power failure, power interruption. So Yashwant, please be keen about joining of the sir.
Sure, sir. Sure, sir. I'm on it.
Yashwant, if it is late, can I introduce a council? Can I talk about the council for Green Revolution? What do you say? I wanted to talk in the valedictory, but as sir has some time, so can I introduce what is a council activities and council?
Please go ahead, sir. Please go ahead.
Okay, then, it is a proud privilege to speak few words about Council for Green Revolution. So Council for Green Revolution is an environmental organization founded on Earth Day, 22nd April 2010. It works for the protection of the environment and promote sustainable development. It is a group of concerned citizens comprising of eminent environmentalists, scientists, ecologists, journalists, and supported by others who are deeply concerned about the planet Earth. CGR main focus areas are green revolution, massive tree plantation through community participation, concerned citizens, school children, local bodies, institutions, government agencies, public sector, and private sectors. So far, CGR facilitated the planting of about 3.9 million saplings. CGR founded Greens Alliance for Conservation of Eastern Ghats in 2011 for awareness, synthesizing the knowledge and policy advocacy for the conservation of Eastern Ghats. CGR published a world-class book entitled Eastern Ghats Environment Outlook, which was released by Sir Pranab Mukherjee, former President of India in New Delhi in 2019. For the emergence of the Earth leadership for achieving sustainable development, CGR founded the Earth Center at Ansampalli, RR District, Telangana. Geospirit programs, Earth leadership programs are conducted involving youth and community members. CGR has actively brought several publications. Simultaneously, it is organizing a series of webinars, competitions on key sustainable development goals, relevant health, education, environmental policy and legislation, and village sustainable development. CGR has received Harita Mitra Award consecutively for 2016 and 17 from the government of Telangana. CGR also received Justice Kuldeep Singh National Award from Capital Foundation Society. The organization received Vishishta Seva Puraskaram from Dr. MCR Foundation in 2019. In 2012, APSC ERT included a lesson about CGR's mission in its 5th class science textbook. In 2020, Telangana Model School entrance exam asked a question about CGR. With this few words of Council for Green Revolution, I hope Yashwant.
Yes, sir.
Did he join?
No, sir. Actually, there is a signal issue. Sir, he's on there.
Yeah,
Just a minute, sir. Just a minute. Just a minute.
You can make him co-host.
I will, sir.
Sir Halim Khan, sir, I can read Umameshwar Reddy sir's profile.
No, because again, Madhusudan sir has joined.
Oh, okay.
Yeah, he's co-host. Madhusudan
He
sir,
has
over
joined.
to you.
Yes, sir. Madhusudan sir is joined.
Sir, your screen is visible, sir. Sir, please unmute yourself.
Now I'm hearing.
Yeah, you're audible.
Yes. So endemic and threatened plants of Eastern Ghats face mass extinction, already discussed. Then endemism is the ecological state of a species being unique to the defined geographical location, such as islands, nations, country, or other defined zone or habitat types. So the extreme opposite of endemism is cosmopolitan distribution. Then there are two types of categories, two subtypes in endemism. Paleo-endemism, neo-endemism. So causes of endemism: narrow ecological amplitude and poor adaptability, geographical barriers, changing climatic conditions. And India shares about 11.4% flora among the world. Angiosomes in India, 19,259 plus taxa. Endemic in India, 4,000 species plus, and it is a 23% 90 taxa. Endemic in Eastern Ghats, 450 species. Out of 450 species, 44 are covers in red list categories. The Eastern Ghats are homeland for 96 species of wild relatives crops. Example, wild rice, wild pepper, wild banana, wild turmeric, wild pigeon, etc. And these are some of the endemic species: Dimorphocalix carnulensis, and Pimpinella tirpatensis, and Cleome viscosa, and variety, and Ophiorhiza chandrasekharani, and Euphorbia sengupta. This is Barlegia stocki, endemic to AP, Hibanthes vastavini. And these are the very famous, everybody knows about the Pterocarpus sandlanus, endemic to Eastern Ghats, and Justicia nilagrensis, Croton scabiosus. And this is the Decosphere species. 3 species are endemic to this in the Eastern Ghats area. And Areoliana hookeriana and also Areoliana lushingtoni, both are endemic to Eastern Ghats. And threat to biodiversity. There are several threat to the biodiversity: destruction of habitats, fragmentation of habitat, and overexploitation, hunting of for poor economic purpose, pollution, and also agriculture, and also predators and population growth, exotic species. There are several ways of the threat to the biodiversity. So these are some photos for the threat of biodiversity, how it is going to threat to the various factors. And these are other photos, and also some of the areas, forest areas, cultivating the podo cultivation, and forest lands are occupying, and threat to the native species. And see how the lands are, forest lands are cutting to the huge trees and converting to the cultivation. And IUCN threat categories, International Union for Conservation of Nature and Natural Resource, they categorized the threat categories. The concept and structure of data book has promulgated by IUCN. The aim at categorizing the species at risk according to the severity of the threats facing them, and also estimating the immense for their extinction. The concept has been adapted by the several countries which have started to put together their own lists. In India, Wildlife Act 1972 lists in its schedules of species, animals according to their status in the world. So scheduled species, these are the very important scheduled species: Schedule 1, Schedule 2. Like you might be heard about the black buck who is hunted by the Salman Khan, he jailed sometimes. So this is very serious Wildlife Act in India. And the Botanical Survey of India and also Zoological Survey of India published the data books on threatened plants, animals in the country respectively. So these are some categories. I don't want to go details about this category. So in the latest red list categories, IUCN 2014, IUCN evaluated 6 plant species as extinct. 61 are critical endangered, 151 are endangered, 120 are vulnerable in India. So threatened species in Eastern Ghats, total 44, possibly extinct 3, endangered 17, rare 14, vulnerable 10, intermediate 3. So you can see some of the species are IUCN threat categories, and IUCN fall under the IUCN red list categories. So 2010, this paper published in the Journal of Threatened Taxa, Cycas bedomai. In 2003, the species is critically endangered. And 1997 endangered, and 2003 it come to the 2000. After 6 years, it is critically endangered. You can see how much threat having that each species, especially threatened and endemic species. So this is another endemic species, Illigardia papalipolia, which is restricted to Eastern Ghats. And this is another paper published in the Journal of Threatened Taxa about this threatenedness. And this is Serpigia spiralis. And conservation of biodiversity. I'm going to the next topic. So in Convention on Biological Diversity held in 1992 stressed the need of the conservation of biodiversity for sustainable development and perpetuation of human beings on Earth. So conservation in the protection, preservation, management, and restoration of wild life and natural resource. So through the conservation of biodiversity and the survivability of many species and habitats which are threatened due to human activities can be ensured. So there is an urgent need not only to manage and conserve the biotic wealth, but also restore the degraded ecosystems. So there are two basic strategies for biodiversity conservation. So these are most effective and efficient mechanism for conservation of biological diversity. One is in situ conservation, another one is ex situ conservation. So natural biodiversity conservation, in situ and ex situ conservation. In situ conservation, on-site conservation, ex situ conservation outside the natural habitats. These are the example for ex situ. These are the example for in situ wildlife sanctuaries, national parks, biosphere reserves, reserve forests, and ex situ seed banks, zoos, and botanical gardens. So in situ conservation, we don't want to go for about full details. Everybody knows about the in situ conservation. And ex situ conservation, it's outside the natural habitats, especially botanical gardens, zoological parks, and seed banks, gene banks, germplasm banks. So then we will coming to the real picture, botanical gardens. I'm going to take the ex situ conservation one example as a botanical garden. So a botanical garden is a place where flowers and fruits and vegetables are grown. And most of them have started keeping the exotic species or plants for educational and research purpose. So many rare endangered plants lives in botanical gardens, which have taken task for conservation of plants in real sense. So in India, first botanical garden was established in Bombay in 1830 by Agriculture Society. And in 1838, one more botanical garden was established in Madras. So these are some of the pictures of the worldwide botanical garden pictures. So when coming to the botanical garden, Ekavemana University, then we are going to what we have done last 10 years. I will show you the next coming slides. So Ekavemana University Botanic Garden is intended to educate the students, researchers, and common man about plant biodiversity and its importance in human well-beings. So our botanic garden mission is promote ex situ conservation of the threatened plants resource of the Eastern Ghats region of India through research and education. So our goal is serve as a national repository of living plant specimens of Eastern Ghats towards ex situ conservation and propagation of endemic and threatened plants of the region. Maintain live plant collections for the purpose of research, education, display, and aesthetics. Serve as a center of excellence for conservation of research and education. And established in 2008 and area 18.67 acres. Total number of species conserved 1,565 species so far. And total RET species, rare, endemic, and threatened plants, 150 species. So this is the barren land which I have selected for the 20 acres, nearly 20 acres land which I have taken in 2009. So you can see very few amla trees and 2 neem trees are present in this 20 acres area, and we made a fencing. So this is the first after 3-4 years it came small entrance and it existed like this. So this is the present situation of the garden, present above that entrance gate. After receiving the fund from the Ministry of Environment Forest in 2017, nearly under the ABG scheme, I received nearly more than 80 lakhs for 3 years of the conservation of the, especially on endemic and threatened plants in this region. So this is the garden where the garden entrance we highlighted with the RET plants, especially endemic and threatened plants with art, with the good paints with our fine art students. When you enter into the entrance of the botanic garden, then the people can aware that what is going inside. That you can see the red sander logs also, the paintings. So this is our elevation, 140 meters elevation. So you can see the Google map also that enter area or university area, it can looks like white soil, it's alkaline soil. It's very difficult to grow the plants. So this is the plan of our botanic garden. There are several blocks in the botanic garden. Nearly 24 blocks are present in the garden. So each blocks its own identity, its own kind of plants are established in each block, like wild edible fruits, then RET plants, plant dinosaur section in the plant dinosaur section, especially only on cycads and wild ornamental plants, which are the plants are very ornamental in forest. Those we planted in the botanic garden. Economic plant section and also cactus section and landscape area, especially lotus and lily plants, woodland area. So these are the sections, and these are some of the infrastructure we developed in the inside the garden, and net houses and construction of the ponds and also other paths. So this is that after entering to the botanic garden, you can see the some of the court and sports and also vision and mission of their garden. So with the barren area, it looks like this. This is the one aquatic pond we established, artificial pond. Here also we given one quotation water awareness. No water, no life. Save every drop of water. See, so these students has to aware the water conservation, especially students in hostel living. So they are wasting their water. So we are going to explain there when I come to the visit the botanic garden about the water importance. We never know the worth of water till the well is dry. Thomas Fuller has given wonderful quotation. Well dry, we don't know the worth of the water. So see, this is the 4 years back. The pond is a very dry area, very just we kept the water and also given the water lilies. Just now it is looking very greenery, and also it's having a nearly 50 to 60 species. It is occupied with all aquatic species, with grasses, other aquatic plants. So these are some of the areas, landscaping area, and so these are the net house facilities. We have 5 net houses and vermicompost area also we established. So this is the some of the areas, landscaping areas constructed in the botanic garden. So this is the one area which the plants are occupied very thick arboretum with trees, all endemic and threatened trees. So this is the herbarium and lichen herbarium, and also herbarium also established in connect with the botanic garden. Then when the students will come, then they have to see which are the plants are herbs, especially herbs which are documented in the herbarium house. So research activities, germplasm collection, I and my students along with the other team, we are visiting to the various forest areas and collecting the RET plants with the proper care and taking them to the botanic garden and massively propagating in the botanic garden. So this is the some of the areas, cycad collection and also nymphia collection. So these are the seed Cycas perica collected from the East Godavari, Pinjarikonda Hills. Nearly 2,000 seeds we collected, one phase, and the other phase we collected 5,000 seeds, and all seeds are germinated. Now all the plants are growing with 3 years age. Nearly Cycas bedomi and Cycas perica, we are the highest propagating persons in the world. I have to proudly say, these 2 species are 5,000 and 4,000, nearly 10,000. 2 species are 10,000 individuals are germinated and growing very well. I will show you that slide also. So shortly, after 2 years, we are going to rehabilitate in the again going to the forest, and also we will plant it in the wherever the plant we bought it from the area. So these 2 plants also risk. So this is the tissue culture lab. So this is the process where we are doing in the net houses and with the massive propagation with in the net house and also outside the net house, and treating the seeds and also seed scarification, like that. We are doing several methods and propagating the RET species. So these are the some of the Hildegardia papulifolia and also Boswellia vavilifolia, then Syzygium alternifolium. These also other RET species are growing in the botanic garden. So this right side Hildegardia papulifolia, you can see back side we have distributed to the city forest Kadapa. Now it is 1 year back, this photo, it is growing very well. Wherever we distributed, again, we are going to visit and taking the photos and whether a plant is growing or not. And then we are distributed to the people. And also I am asking the people how we are distributed important plants, whether it is growing or not, other good condition or bad condition. Like that, we are taking the photographs and also we are updating every time. So these are the other RET species which are growing in our net houses in the massive way. So these are the list of the RET species. I don't want to read out all the species. So this is the Cycas beddomai and Cycas verica in the wild. You can see how beautiful it is. Both species are in wild, both male and female cones. You can see seeds we collected from the Sheshashalam Hills, Cycas beddomai and Talakona, especially Talakona, and Cycas verica from the Pinjarikonda Hills, East Godavari. So this you can see lab from field to net house to land. These are the Cycas Sheshashalmensis, Cycas perica, Cycas revoluta, Cycas beddomai. All are growing very well in the land, in the botanical garden, and also especially you can see inside the net houses how much individual population we are propagating. This is only Cycas beddomai, Cycas perica individuals, and Cycas beddomai. I will show you the videos, but time is not permit me. Then I will show you the videos also. So this is another important species. Recent last year we introduced to our botanic garden from Sikkim Botanical Survey of India Sikkim Center. They contributed to us our botanic garden. Ginkgo biloba, living fossil. Everybody knows about this plant. So this is another Boswellia vavalifoliata from the wild, how it is looks, and we bought from the wild, and now the nearly 2,000 individuals are growing in our botanical garden. So this is another species, Hildegardia papalipolia. Hildegardia papalipolia, all over 3 years, we propagated more than 5,000 and distributed to the all over India and botanical garden, forest people and beautification people, several people. So this is recent red standard Pseudocarpus sandlina also. We propagated massively and also distributed. And Syzygium alternifolium also propagated and distributed massively. And some of the herbaceous plants also growing in our botanical. Boswellia umbellata, earlier it is to be Caroloma. You can see some of the Caroloma species, Caroloma diffusa and Caroloma umbellata. Now it is changed to the Boswellia umbellata. So these another Carolomas growing in our botanical garden: Caroloma crelunata, Caroloma lacinantha, Caroloma diffusa, and Caroloma ascendens. So these are another conservation of the other herbaceous plants. It is very difficult to conservation of herbaceous plants, especially Brachychelmas and also Cirpigias, Brachychelma annamacharya, Brachychelma ciliatum, Brachychelma colarensis, Brachychelma maculatum, Brachychelma pullai, Brachychelma seychelles. So like that, herbaceous plants also, this every season it's coming and going. So this is another, recently we discovered a new variety, Brachychelma maculatum variety. In Redia it is accepted. So these are other Cirpisia species, Cirpisia gentiana, Cirpisia pullai, and Cirpisia spiralis, and other Decastesia corpensis also growing in our botanic garden, and Decalepis hamiltoni, Dalbergia latifolia, and other Cirpisia bulbosa, Lucy, Cirpisia candelabrum. So this is another important thing I'm going to express here. Last year I introduced the world's largest leaf, Victoria amazonica. I collected from the Calcutta Botanic Garden and introduced to our Botanic Garden. This is a very big size pond, especially for this plant only, Victoria amazonica only. We created this pond and established it. It's growing very well, and now it is not a season. It's a last year photo. It's going very well, and also it's within 1 year we established it very beautifully. So these are the leaves which are grown in our botanic garden. You can see how much size it came. And see, it is the first time in Indian history the leaves bear nearly 10 to 29 kilos of weight of each kid. In Botanical Survey of India also, in Calcutta also, they used only 12 kilos of baby they kept on the leaves. So in our botanic garden, we tried up to 28 kilos. You can see how one day we did a festival on Victoria Festival. So all kids came and they have taken pictures. You can see Narshita, 28 kilos. This girl is 28 kilos, and even though it is bearing that leaf, I got that very good response from all our Kadapa district, and also various people are visited, and we got the good citation to our botanic garden. So this is how the people, how many students are visited, and how many students are visiting, and awareing the plant wealth of botanic garden, and also especially on conservation aspects. So these are some water lilies growing in nearly 5 to 10 colors of water lilies are I'm conserving in the botanic garden. So this is Acacia pistula. So Akira Miyawaki, I think everybody knows this method of plantation. Akira Miyawaki is a Japanese botanist. I don't want to go for a detail about within a short time, within a short period. So he's developed one method, high density forest, very close to plants are growing, and developed within the 10 years or 20 years of span. With the 300 years, it will take time 300 years to grow. So that is the forest you can create within the 20 years. So like with that method, we started nearly 10 acres of sites, 5 acres, 2 sites we selected in the university, and nearly the 40 species of wild plants we selected. Total individuals are 1,20,000 plants are each hectare, each plot. 5 acres plant, 55, 650, around 50 plus 1,000 plants. Another 5 acres, 50,000 plants. It is a highest number of individuals, or highest number of high density forests in India. I am proudly say 10 acres of nobody is maintaining. So this is the earlier time how they started the. They spend 1 crore rupees for our university, and especially this Akira Miyawaki plantation, and with the MGNRC grants, and the daily wage people are coming and they are doing the works. So now you can see high density plantation 2019-20, just 1 year back we started. 2019 December we started. Now, so this is like this. The plan. This is also 6 months back photo, not current photo. Current photo is more than 10 feet height each species. So very fastly grown. Within a span of 1 year, the total area is created a small forest, forest-like forest. So the education awareness, we are giving good education awareness to the students or botanic garden, especially on RET plants and conservation of natural resource, and also especially on plants, especially on threatened plants and endemic plants, Eastern Ghats. So these are the people are visited to our botanic garden, and famous scientists from Lucknow, then Rasingham BSI, and CSR ex-director, and also Sanjay Prasad. There are several people visited, and also NAC director. And these guys, like this, we are distributing to the people. SR Yadav from Kolhapur University, we distribute to their botanic garden plants, and they are also growing well. They are also sharing the photos. So various people are coming and planting our area. Then recently last year, Dr. Hamid from BSI Kolkata also we are distributed, and also every forest department. So education awareness programs, connected RET plants awareness programs for schools and college students, connected RET plantation programs, resource materials we prepared on brochures, endemic lichens of Eastern Ghats, RET plants, posters, booklets, and plants for Ecom University campus, handbook on endemic and threatened plants. So these are some of the brochures we started. Ornamental plants, and also to aware the people. So these are the brochures we prepared for the botanic garden. So this is also another brochure. So this is a giant water lily brochure. So this is the lichen brochures. So these are one of the new species we discovered, Brachyshalma vemane, with this project. Also recently, also one last 2 months, another species published, Brachyshalma Lepidognathis Rajasekare, former vice Rajasekare Reddy, we kept the name Lepidognathis Rajasekare. So awards we received 2-3 times Chief Minister, Vanam Award, IP Green Award, and AP Best Biodiversity Conserver Award. And these are some of the news clips and covered about our botanic garden. So this is one flower. Why I'm showing this last slide is, so whenever we have a functions or whenever we have a any guests are coming, just we are collecting the Botany garden pond and giving to the bouquet without spending the plastic, without buying from the shops. Just we are giving our natural bouquet. So I'm suggesting to people, then you can prepare on our own bouquets and give to your guests. Thank you very much.
Thank you so much, sir. Thank you for such a colorful presentation and your efforts to conserve the local area of Eastern Ghats. We are very proud to be you with us in this meeting and inspiring so many people who are attending this conference. So thank you very much. Now I request Uma Maheshwar sir to start sharing his screen.
Thank you very much, Halim.
Thank you, Halim sir.
Now, Raviraj, can you start introducing sir?
Yeah, I'm sharing.
Yes,
Raviraj,
sir.
over to you.
Yes, sir, I am ready.
Start introducing him.
Okay, okay, sir. First of all, I thank to Dr. Masan Reddy, sir, because I attended the refresher course at Kadapa, Yogi Vemana University. And
Thank you, sir. Thank you, sir. Thank you very much.
Yes, our next and last speaker, Dr. Uma Maheshwar Reddy, sir, garu. So I'm welcoming on behalf of our Giraj Government College and Telangana University in collaboration with the Council for Green Revolution. Dr. Uma Maheshwar Reddy, sir, has been an environmentalist with 25 years of work and academic interest in environmental protection and nature conservation. He completed his master's degree in applied geology and a PhD in environmental pollution of Musi River from Usman University. He worked with national and international organizations such as International Water Management Institute, CRISAT, and the National Academy of Construction, Center for Economic and Social Studies, etc. He has been part of various international projects on environmental management, water management, sustainable development. He has authored 3 books, The Eastern Ghats Environmental Outlook, Changing Face of Rural Andhra Pradesh, and Power of Collective. He is presently a technical advisor, environmental technical advisor, and a Council for Green Revolution, environmental organization. With this introduction, we are heartfully welcoming Dr. Uma Maheshwar Reddy, sir. Over to Uma Maheshwar Reddy, sir.
Thank you, sir. Am I audible?
Yes, sir.
It's a great opportunity. I thank, first of all, the organizers from Giriraj College and Telangana University, Department of Botany, and of course, we are also a collaborating organizing partner. And I thank you for your kind introduction of me. Thank you very much. And I also thank Abdul Halim sir, and my special thanks to Venkatesh, who has coordinated this program from CGR. It's a wonderful program, and I am afraid that after a series of eminent speakers, professors, it's very difficult for me to hold the attention of the participants in this webinar, but I will try to do my best. I'm a earth scientist from Usman University, and I did my research on Musi River, and I worked on various nature conservation and environmental projects. And moreover, I have a passion because I hail from a region called Eastern Ghats from the foothills that inspired me to work on this, to take this as a lifetime work for me. And I'm here at CGR, Council for Green Revolution Environmental Organization. It's a 10 years old organization working for the, focusedly working. It was founded on 22nd April 2010 in a small village called Anmaspalli. And its focus areas of massive tree plantation, conservation of ecosystems, environmental education, earth leadership, sustainable development. And it's a group of concerned citizens comprised of eminent professors like Professor K. Purushottam Reddy, Dilip Reddy, and other many people from different fields. And this is our office. You see, I'm inside this building now speaking to you. This is a sprawling premises near Hyderabad on the highway of the Ring Road. And this is patronized by Srimati K. Leela Lakshmareddy and K. Lakshmareddy. They founded and patronizing this organization and this office and other initiatives as well. And this is inside our premises. I'm just beside this wall. And, you know, we have already, I told this is a 10 years old organization. Its flagship program is 10 million tree plantation campaign. And it is through children actually. And the other intention is to seed the environmental consciousness in the young minds. And so far we reached 39 lakh plantations in villages in Telangana and Andhra Pradesh. And we could facilitate the preparation and distribution of 7 lakh seed balls and 10 lakh students we have reached so far. This is the work through this Vanasankalpa 10 million tree plantation campaign. And then these are some photographs in the field, children planting the saplings, and they are given 2 plant fruit-bearing trees and 3 shade-bearing trees, and they are sensitized before giving the trees. And likewise, there are so much of success in survival rate of the saplings. This is a passionate work CGR is doing. And these are some areas, a brief forest, tiny forest we have created. This is one school in Khanapur near Hyderabad. It is like now, if you go there in the premises, you will feel like a small jungle inside around the school. And this is somewhere in a cotton mill in Amangal near Hyderabad. This is also created by CGR plantation. And this is a village. It is 200 houses and nearly more than 10,000 plants are there. This program is green village actually, Silarpalli. This is one landmark and success to share. And these are the seed balls prepared by the children. And these are the seed balls. Here you can see our President Leela Madam. She's with the children who are scattering the seed balls in the nearby wilderness in the hills around Hyderabad. So like that, we are involving children, and like that, we are inculcating some environmental leadership and consciousness. And another initiative, already this was briefed by Abdul Alim sir and Sai Bhaskar sir, but anyway, in pictorially, I would like to present what we do, CGR do. This is about Eastern Ghats initiative. Already our co-speaker, our speaker, Dr. Masoon Reddy sir shared. This is one of the unique and prime ecosystem of India, one of the prime mountain ecosystem of after Himalayas and Western Ghats and Eastern Western Ghats. So there are so many endemic and endangered species, and its biodiversity is rich. So we have organized 5 regional and national conferences and released a book called Eastern Ghats Environment Outlook. So this book, a voluminous book, and you can find it in our website www.cgrindia.org. And sometime back in 2019, this was released by Pranab Mukherjee, the former president of India. Here you can see our advisor, Professor K. Purushottam Reddy, and Leela Madam there, and Tulsi Rao the other, and Professor M.C. Mehta is one of the eminent leading enroll legal activist, and Dilip Reddy Garu, former ITI commissioner. This was in Delhi. So these are the tasks through Conservation of Eastern Ghats, and this is we are coming up. Actually, the structure is construction is over. We are coming up with a world-class environmental academic training and research center near Hyderabad Airport. This is called the Earth Center, and we would like to start it in a strict academic discipline and applied research to take up ambitious work and to reach out in manifold to the people around the world for environmental stewardship and ecological stewardship. And then now coming to the subject, because we are running short of time, and it's almost afternoon. In the fag end, it's difficult to keep your attention. But anyway, I will try to be crisp and onto the precise to the topic. And this is my topic is plants and the plants for a living planet. The Earth is the only living planet in the cosmos, in the space. We don't know where else we find a space, a habitat to live, and we are trying where various space missions are sent to the space, atmosphere, and space to probe some planet or some space to live. But we don't know when we will find. But certainly we have around our home. This home is Earth, and Earth is a living planet. It's also called blue planet, but it is many people call a biological scientists and other call it a living planet. So all the life forms and mammals, plants, and everything are thriving in this Earth system because it has a life supporting systems like atmosphere, air, water, and all these things. These are the basics actually. And coming to the plants, they are the important building blocks of the ecosystem. So any ecosystem, grassland, maybe forest or a mangrove, whatever it is, the basic organisms are the trees and plants, and from tiny algal forms to the mighty sequoias, and that the organic link between living and non-living creatures. A tree itself is an ecosystem. One of the study by Cornell University revealed that a redwood tree hosted something like 400 organisms and creatures, like small mammals, rodents, and everything. So a tree itself is a very big ecosystem. It's a home actually. They are the source of inspiration and enlightenment, and Buddha got enlightenment under the tree. And they are the foundations of civilization, and the ancient Gurukulas were there, and knowledge and contemplation was there. They are the green mantle. They are like skin and protective skin of an organism. If we peel off the skin, what happens? The organism will die. So are the important. And from tiny algal forms to grasses, herb shrubs, and gained sequoias, and also mighty fig trees, they are very diverse. And in the farm and habitats, they form dense forests, mighty woodlands, grasslands, and mangroves, etc. And they're very old and ancient. They came onto the evolutionary chart some 1 lakh years. Of course, the Earth's age is of 4.6 billion years, and plants came onto this Earth about 1 billion years ago. Very old, very old. And there are about 4 lakh plant species recorded so far, and still explorations in the Amazon hinterlands and Congo Basin, and even in the Papua New Guinea, even Eastern Ghats also, still there are new inventories. I think some academics publishing new inventories in the nature journals and botanical journals. Without them, natural history of the Earth is near blank, and without them, there would be no human civilization and progress. Without them, Earth will not be a living planet. It will be a dead planet. That's why I took this topic to share my concern. It's not a primary research work, but it's a prime concern that I would like to share with all the audience and participants in this webinar. And here also we can mention about some of the founding fathers of exploration and founding fathers of exploration of the natural world and plant sciences, like Alexander Van Humboldt, who discovered the flora and fauna of the Latin America, South American continent, and Charles Darwin, evolutionary theory. Alfred Russel Wallace, you might have heard about the Wallace line, which divided Indo-Malayan plant realm with the Australian Pacific botanical realm. It's good work he has done. Bentham, Hooker, Hutchinson, and Gregor Mendel, Carlos Linnaeus, Sir Jagdish Chandra Bose, a proud son of India, and plant breeders like Washington Carver and Louis Burbank, who worked to sacrifice their work who gave much of the time for the plant breeding and for the feeding of the millions of the people. And then, plants provide so many ecosystem services, like provisioning services, like water, agriculture production, and pharmaceuticals, medicinal, and the regulating services. They control watersheds, water flows, and they act like a gain sponge and release slowly, and they increase the stream longevity, and they act against hazards of the natural calamities. Like yesterday, what happened in the Terrigar, I think in the Uttarakhand, and human interventions there destabilized all the Himalayan geology and soils, and so many power plants came up, and human interventions that caused yesterday's devastation and fatal loss of life. And there are cultural services, spiritual, recreation, biodiversity, and ethnological diversity, and supporting services, growth of vegetation, production, photosynthesis. These are all the selfless services rendered by the plants, and they are the perennial food basket. Without plants, there is no food on the planet, and without plants, there is no pharmacy. And half of the medicines in the world are derived from the plants. Many are from tropical ecosystems along the equatorial forest, and Madagascar periwinkle. It is a lifesaver of many leukemia patients around the world. It's a wonder plant. It was ignored before, and it has a wonderful metabolite that helped in countering this leukemia and other diseases. And like so many, nature is a wonderful pharmacy, and it's a source of energy also, like trees, plants, and for fuel wood and many things. And it is habitat for people. And they are the water towers. Half of the world's water comes from the forested watersheds. It is regulated, and it is perennial over the year because of this forest act as a water tank, slowly releasing throughout the year a stream base flows, operating stream base flows with its inner maze of the roots and all these things, and it makes a climate balance, soil health, biodiversity, space, economic, social, and spiritual spaces. And recently, I think last week, there's a news that Supreme Court appointed a 5-member panel to evaluate the valuation of the trees, and they said that a tree is valued 74.5K into multiplied by its age. That means if 100 years old tree into multiplied by 74,000 rupees, 74.5 thousand rupees. So a old tree is more than crore rupees, but it's difficult to evaluate a tree because its services are so immense. If you take a keystone species, a fig tree or something like that, it can give birth to many other trees around the region. So it is very difficult. But anyway, it is at least monetarily put on a high value, but still I doubt it needs much more deeper evaluation and to come to a real conclusion, not on monetary terms. But anyway, and let us have a synoptic view and a peregrination and a bird's eye view of the global forest cover, because we are talking about a living planet. And the tropical forest, we have 3 types of global biomes, forest flora: a tropical forest vegetation, boreal forest cover, and temperate flora. I'll go quickly. These are the boreal forests and the circumpolar forests, thin soils and short growing periods and cold climes, and they are around in the Scandinavia, Siberia, Alaskan United States, and Norwegian tundras. And much of the logging is going on there, and they are under plunder. And these are a picture of the boreal forest from Canadian Arctic. And these are the tropical forests along the equatorial region. These are the highly diversified, very high with biodiversity. One of the national park in Manu in Peru, it has nearly 2,000 tree species in a small plot of 5 to 10 acres. So they are very rich. Losing a tropical forest means losing our a vast gene bank, actually a precious gene bank. And these are the temperate forests like in the Western United States and Europe and Eastern Siberia. These are all, and southern Argentine regions. These are also important. Here are the species like maple, oak, beech, and here in the tropical vegetation we find dipterocarps and hardwood, mahogany, and teak and other very hardwood species we find, and canopy is the characteristic of this region. And here in the boreal forest, I'm just getting back a few slides, and in the boreal forest, maple, spruce, pine, and short growing, and but tall growing trees and intermixed with the wetlands and all these surface features. So these are the brief profile of the forested landscapes and vegetation of the planet, major vegetation types, and this is the temperate forest. You know, this is the beech, oak, and maple in between polar regions and the equatorial region. You see, characteristic of this is xanthophylls and reddish color in the season, autumn and spring. So these are the forest and vegetation wealth of the planet, and we have vital signs. You know, before industrialization, population was I read that it was only 100 crores or 150 crores, but now it is 8 billion people in a limited infinite land space, and it is hotter by 1.5 degrees centigrade before and after the industrialization. And you know how many ships passing through the Suez Canal and this Panama Canal, and also this um, eh, uh, South Pacific Seas and South China Seas. There is so much commerce, trade, and communication. Goods are moving, and resources are extracted and processed and moved around the world. So there is so much of trade that is posing an imminent threat to the loss of biodiversity, and the face of Earth is changing, and communities and social needs, requirements, and consumption patterns are changing in a rapid way. So resources are finite. We are pushing to the planetary boundaries. Need of the greed are high, and tree world in and outside the forests are in pressure. You know, outside forests there were about 7 to 10% of the forest cover, but it is limited to 2% actually. The outside the forest vegetation is lost so much. Now it is maybe 1.52%. You know, earlier in the olden days, there were many trees dotted around the villages when we go on a transect or something, like walk through a village road. But now where have gone they? So many trees, they are disappeared, and there are limits to growth, and our economy can't go in this unsustainable manner. And the flora by axe, machine, fire, and flow are farming, ranching, and planting, mining, logging, tropical forests, especially in the Amazon. It's a ranching ground, and so many cattle are there. And also coffee plantation, their soybean plantations are devastating miles and miles of forest in the East Asia, like Borneo and Indonesia, Malaysia. Palm oil is one of the major, and coffee, of course, and tea. India is the largest exporter of the tea, and they're all occupied the prime pristine forested areas in the mountain regions of India and East Asia, and affects the climate change. And there are even the forests are affected by this climate change impacts. 1.5 degrees means there are pests and other diseases, and temperate forests are highly impacted. And deforestation, a profile briefly, 13 million hectares of a year is lost, and causing 20% of greenhouse gases, and loss of biodiversity is alarming, debilitating the capacity of the carbon sequestration of the planet and accelerating global warming. Soil erosion is very rampant everywhere, on every continent, not only in the forested areas. Everywhere, 2 billion people lose sustenance and their source of economic and fresh water supplies are impacted, and pollination, frugivory, and other ecological functions are highly debilitated. And United Nations General Assembly sometime back in a decade ago has made a resolution and placed environmental concern beside peace, social justice, international relations, and atomic global security, all these things. It is one of the important, most prime concern, global concern. So environmental security is our future. Otherwise, we don't have future on this planet. And Mahatma Gandhi said the earth has enough to provide all the needs of all, but cannot satisfy the greed of a few. How long we go on plundering until the last tree is cut, until the last river is fished, until the last river is dried. And so we have to heed all these things. And then this is one classic example. This is Borneo Island. It was lush green forest, tropical, a classic tropical forest. And you see the changes, deforestation changes, and it has gone into the core areas, mountain areas, and just only in the central highlands there is some forest remain. And this is an Amazonian example. In 1975, this was a Rhodesia state, and a trans-Amazonian highway came, and then people started moving inside, and the fishbone pattern, the lateral roads came, and entire area gone out of vegetation. And these are some pictures in Amazon. And this is the palm oil plantations replacing a diverse tropical forest into a monocropping. And this is one of the example of Canadian lumber mills. And from Eastern Ghats, mining is in the northern Eastern Ghats, mining is so heavy and extensive, like bauxite, iron, and all other important ores. So much of forested areas are lost due to mining, stripping, processing, and transportation. And this is Podu cultivation, subsistence farming by the tribals. It's a traditional thing, but its ecological extension is so heavy, ecological impact today, because so many tribal people and each one vie for some patch of land to cut and grow some crops. So in the northern Eastern Ghats, it is so evident and conspicuous. And it is one of the area Podu, close shot of a Podu cultivation in Araku. And the cashew plantations are steeping up to the hills in Visakhapatnam areas. And urbanization in this is what creeping up into the forested mountain. Or Visakhapatnam city is going up and up into the mountains. And logging for the road expansion in the northern Eastern Ghats. And it's a forest frontier near my village. People moved just beside the protected area, Tiger Reserve there. It's a contact point, and forest is pushed to its limits to the highlands. And perennial food basket is impacted, and food supplies are impacted, and local indigenous communities like Chenchus, very old primitive tribal groups are affected. And their firewood and basic sustenance lifestyles are impacted. And water towers are impacted, and habitats protected areas. Only 5.4% of the area is earmarked for the protected area so far in India. It's very short and very small area. Even that also under pressure. So I will say a quotation I will quote John Holdren: "The biodiversity of the planet is unique and uniquely valuable, and have been steadily burning down one wing at a time. Before all the biodiversity is cataloged all the books, let alone read them all. Before we reading them all, we are burning a library. It's like what we are doing to the forest and woodlands on our planet." There are some classic examples of what happens if we cut trees and woodlands. There are examples like Easter Island, It is one of the island in off the Argentina coast, and some castaways thrived there 5,000 years ago. They developed a civilization, but due to egos, human egos, they cut all the forests, and it has become barren, and whole civilization vanished. Even it is same in the pre-Columbian civilization, South America, like Aztecs, Mayas, in Guatemala, Peru, Honduras, and other countries. And Great Dust Bowl in USA of 1930s pushed the people to migrate away from their central plains, American central plains, because its productivity uh, fell down due to the soil erosion. And famine in Sahel, sub-Saharan Africa is a classic example. So what happens if we lose all our green mantle of the earth? What happens? That is a concern. And this is called Svalbard, a archipelago of Norwegian Arctic, and they, with the help of some philanthropists, scientists have created a seed vault. Actually, their concern is if something happens, if planetary disaster happens, if the biodiversity is lost with this scale, if something else happens, we should preserve the seeds, whatever the nature holds and nature has and had, and like that. And they created a Svalbard seed vault. So this seed vault is a classic concern and example. So it's a beckoning light, actually warning. It's a warning what will happen to the future of the biodiversity, floral biodiversity on the earth. So seeds were earlier on in the nature and in the earthen parts in the every home of the villages. Now they have gone to the corporate like Monsanto and others. But even now they are also under threat from vanishing from the nature. So Svalbard seed vault is a classic example. So what will happen to the future if we don't concern? This is they are labeling and sequestering all these seeds brought from the hinterlands around the world, like cereals, pulses, millets, and fruits, and natural wild varieties. And you can browse in the internet. And sustainable. Everyone knows this. United Nations has a framework of sustainable development goals. In that 17 goals, out of 17 goals, the second one, zero hunger, sustainable farming, all this one, and the climate change and the life on land directly linked to the plants and the plantation and conservation of plants, and our concern and the passion to the plants. So if these are the basic primary goals, we based on these other goals are they were catalyzed and something like that. So, and even water, the sixth, and clean water, sixth, is also interdependent, and many other sustainable development goals are interdependent. So we have now reforestation is the one of the most important job and moral duty that human societies has to take, and every individual's responsibility. And we have examples like Ashoka and John Muir. Richard Saint Baker is known as Tree Man. His life totally dedicated, and his original name everybody forgot, and is known as Tree Man. And Nobel laureate, she got Nobel because of her activism in Kenyan forested areas, Kenyan mountain areas, Aberdares and Mau Mountains and Mount Kenya, like that. And we have in earth defense people like Bishnois. In 1730s, 340 Bishnois were killed by a king who are defending against the felling of this Kejri trees. And it's a inspiring story. And Chipko movement of 1980s in the Himalayas. Yesterday's example also, Chipko, it is linked. If there is a forest, and this would have not happened. And Silent Valley in Kerala, people stopped Silent Valley. And in Brazilian Amazon, a martyred source of inspiration, Chico Mendes, is a rubber plant tapper, and he defended the local indigenous communities, and he was shot on point blank. Same is the case with Dorothy Strong. She also defended the Amazon and indigenous tribes, and she was killed. And Diane Fossey, she was defending against the deforestation and mountain gorillas, and she was also lost life. And we have, of course, we have some success stories in Tizca National Park. It's a last Atlantic forest. It was reforested by Dom Pedro, a king of in the Brazil. And many of the trees planted, millions of trees sometime back in 1930s in Kenya. And civilian conservation corps in the USA after the Great Dust Bowl incorporated by Franklin D. Roosevelt done a marvelous work to stop the erosion and loss of vegetation. And Korea is a classic example. And Bhutan, they don't cut the trees, they conserve the trees. And in Greenbelt movement, they have planted 51 million saplings by women groups, and nearly 10,000 women groups. And there are efforts going on Great Green Wall of China to contain this Mongolian Gobi Desert advancing into the Chinese frontiers. And Great Smokies in the eastern seaboard of the United States is a classic example that if we stop the human interventions, the nature revives itself. And Catskills in United States, and this New York City spent $3 billion to protect the catchment. They're paying farmers and others to keep the catchment intact and not to disturb anything, that New York water supplies are secured. So Annapurna Conservation Area is a classic example, and the revenues from ecotourism are used for averting people from cutting the trees. And Bialowieza in Poland, it is one of the only virgin forested European forest, which is a home to the European bison, is protected by stopping the human interventions. And we have also examples. Felix Fingbeen, a 9-year-old boy who inspired United Nations and other world nations, that he has done marvelous work and planted millions of trees, inspired planting millions of trees. And Jadhav Prayang, a ordinary man from Brahmaputra village, recreated a forest in Brahmaputra river island. And there is one old woman called Timmakka in Karnataka. She has no sons and daughters, and she took as a mission, lifelong mission, and created a green avenues along the roads, kilometers of green avenues, like very big trees. They cover the road as a canopy, and we walk under the shade. So she got Padma Shri award. And there are many unsung men and women around the world, those who have passion and strength to inspire and work. So our even Indian Constitution also says that protection and improvement of environment is the role of government. It's the responsibility of government. And according to the 48A article, and according to 51A article, it is also the duty of the citizens to protect the forest, lakes, rivers, and wildlife, and have compassion to the living creatures. So we have to do so many things to reforest, because we have lost nearly 20-25% of the global forest cover from pre-industrial times to hitherto now. And hence we must work, and we must commit, and we must pledge. And that's what CGR is doing its own part as a civil society organization and a group of and that finally I would like to say I concern about our request, and I express all the other fellow beings and all other society to have work for the environmental responsibility, ethic, a land ethic and a planet ethic, and eco-resilience towards the natural background ecosystems and life forms. And we need to develop like ecological stewardship and earth leadership. And thank you. Thank you. This is the presentation. Thank you very much, sir.
It was a great talk, sir. Actually, we are very much inspired with your presentation and such a wonderful experiences you have shared. And it is a time and we need to alarm the generation for conservation. It is really a big job done or carrying out by Council for Green Revolution. We appreciate the efforts and it is a correct platform to have a focus of such kind of activities. So it is a great privilege and proud moment for us. You have shared your experiences, and as a time has come a long way, and I request Deepak Parmar to request our next person to initiate our valedictory. Deepak Parmar, can you request?
Yeah, sir. Thank you, Alim Khan, sir. I invite you all to the last phase of today's international conference on plant and environment. So I welcome our principal, sir, Dr. E. Lakshminarayan Garu, and also vice principal, sir, Dr. Abdul Rafiq, sir, to this valedictory function. And I also invite Professor Kamlakar sir, Head Department of Botany, Usmania University, for this valedictory function. So
And
first,
Indira Madam from New Zealand. Indira Madam from New Zealand has joined.
and I also welcome Indira Madam from New Zealand to this valedictory function. So I request Rafiq sir, the Vice Principal of Giriraj Government College, sir, to share his view on the today's international conference. Rafiq sir.
Yes, sir. Sir.
Sir.
Sir.
Yeah, you can share your views, sir.
Yes. Sir, very good afternoon to everyone. I feel it's my privilege to congratulate the Department of Botany for this wonderful conference, because the topic they have selected is not only useful for the botanist, but it's very much important for life. And life is a concern of every human being. Just now I was seeing the deliberations from the morning itself. They were much helpful. Many scientists have also spoken about the topic. But sir, really this topic, plants and environment, is a concern of today. Even sir was pointing out the constitutional provisions for the safeguarding of our forest and everything. I express my gratitude to the Department of Botany under the leadership of Dr. P. Lata Garu and the great colleagues like Deepak Parmar and Raviraj Garu and everyone who has participated and made their efforts and they're given in making this conference a great success. It's really a great opportunity to the Giriraj College to have organized an international conference where not only the speakers of various countries have spoke, but they have also participated. Students and research scholars from other countries have also participated. So really it's a good, and it is the first and foremost international conference. I congratulate the contributions made by Dr. Halim Khan, Telangana University, and other professors of Telangana University and the Green Society of Hyderabad also. And really it's good for our college also. On behalf of our college, all the students, I congratulate one and all. Thank you, sir, for giving me this opportunity.
Thank you, Rafiq sir. Now I invite Professor Kamlakar sir, who is the head department of Botany in Usmania University, to say few things about today's international.
One second, let me introduce him.
Sir?
I will introduce him. Let me. We have a profile of sir. Wait.
Yeah, sir.
Ask him to share the screen. Okay.
Right, sir. Yes.
It is a proud moment to introduce our beloved professor, Professor P. Kamalakar sir, Professor and Head Department of Botany, University College of Science in Usmania University. Professor Kamalakar sir is MSc and PhD. His academic experiences are he was a junior lecturer from 1998 to 1999, and he was assistant professor of botany in since 1999 to 2010, and he was associate professor of botany in University from 2010 to till date. He had a number of refresher courses to his credit and orientation programs also. And he has been a co-investigator of sponsored major research project, and he published a number of research articles, more than 10, and he was the resource person in number of seminars, conferences, and workshops. And he was in his extracurricular activities. He was NSS program officer, and he was a general warden, and he was associate NCC officer. He was in charge head for Department of Nizam College, and he was board of studies chairperson for a number of times, like in 2014, 2016, 2017, and 2018 to 20. He was departmental committee member for a number of times, like in 2003, 2010, 12, and 15. He was departmental research committee member also in 2017. And he was a selection committee member for a number of events, like he was for UGC RF scheme in 2012. He was a selection committee member for Spurti Degree College in 2012. He was selection committee member for Pratibha Degree College in 2012. He was selection committee member for Bhavita Degree College, Vanastalipuram, and Elenki Degree College, Usmania University, Stanford Degree College, Bhuvanagiri, and he was member assessment committee 2 times for IICT Tarnaka. He was examiner for a number of academic responsibilities of PG, UG question paper setting, paper valuation within and outside the Usmania University. He was chairman of Viva Voce board for SV University. He was pro tem guide for retired professor Dr. Vasanta Pillai, research scholar P. Sudhakar Reddy in 2016. He was board member of examiners for PhD Viva in 2017. With these few words of introduction, I would like to request our Kamalakar sir to give his address in the valedictory event. Over to you, Kamalakar sir.
Thank you. Thank you, sir. Thank you for giving me this opportunity. Of course, I feel at the end of this conference, international conference, the resource persons you have involved, it is almost nothing is left, really. They have motivated all the scientists you have included, really. Those from like Madhusudan Reddy, Makbul Ahmed sir, and our Yashwant, Dr. Madhusudan Reddy, and Abdul Rafiq, Deepthi, all the researchers, those who are really motivated, all the research scholars who have participated, all those who have initiated their research work, they got really motivated. I just want to share some, in general, of course, those who are interested to do any research in this field related to plants, that how they are interacting with the environment around these plants. In general, once we have to understand that we are part of the nature, including the human species, particular Homo sapiens species, it is also a part of the nature. Those who are really interested in doing, once understand some basic principles related to the nature, with a view that we are also part of the nature. Any impact on the nature, any impact on the nature because of any developmental activities, or what are the components of this open system where any developmental activities, how they disturb this environment, what are the consequences, and once we understand them through understanding some basic principles, we have to understand in our local area, those who are really interested in doing some research with a view to come out with a solution, by understanding how the environmental changes in the environment just because of any activities without any sufficient knowledge on the environment, how these living organisms are interacting with the changes in the environment. Of course, in nature, there is a ratio of all these components. Let it be some biotic or abiotic components, physical or chemical factors, how they are fluctuating, how they are changing. These changes, of course, those changes may be the climatic changes like daily or seasonal changes, their fluctuations, how it interacts with the living organisms, thereby the evolutionary process and diversity. Naturally, it is in a ratio that goes without disturbing the nature, that just because of the developmental activities, how it is disturbing the plant diversity. Of course, not only plant diversity, other living organisms, how they are interacting among themselves, how it affects the entire biosystem. Once we understand that it is an open system, that system, of course, can consider this one single living organism as a system, open system, that definitely interacts with the surrounding conditions, the fluctuations in surrounding conditions. In that way, if we start understanding, of course, we can consider this our personal system, personal body also as a part of the ecosystem, how it is interacting, that we can experience and understand. In that way, if we proceed, if those research scholars or who are interested in doing further research with an understanding on these local conditions, they can proceed to the extent they can do. In that way, of course, let me appreciate first the organizers, our Giriraj College, Telangana University, and other with the collaboration of which you have been conducting. Let me thank you all for organizing such a wonderful program that really has motivated all the participants. I thank you very much for giving me this opportunity to see some presentations by the real researchers with their dedication. Really, I could not join from the very beginning. Of course, with their names, let me appreciate them. Dr. Maqbool Ahmed from School of Sciences in Maulana Azad National Urdu University, Gachibowli, and Dr. Madhusudan Reddy and Dr. Uma Maheshwar Reddy. Really, I appreciate you for your dedicated work you did, and that really you have not just observed, but you have come out with a solution to restore the nature. Of course, that nature, it was without any disturbance, it was in a perfect condition. Just because of our activities, our development activities might have disturbed that, and after with an understanding on what are the reasons and what are the consequences on the society or on the living organisms, how it affects, that knowledge you are with that knowledge you are doing a dedicated work. I thank all scientists who have already presented, and all the participants, and for organizers for giving me this opportunity. Thank you very much. Thank you.
Thank you, sir. Thank you for your
thank Dr. Lata. I thank Dr. Lata for providing these facilities to join in this meeting. Thank you.
Thank you. Thank you, sir. Thank you so much. I request now Indira madam to switch on the camera and you unmute yourself and give your remarks. Indira madam.
Good evening, good afternoon, all. Thank you for the honor and the opportunity for being a part of this evening, today's sharing platform.
Can you make your video on? Yeah, your video is there, but you're right. Now you're visible. Okay, now. Right, ma'am.
Good evening, good afternoon, all, and thank you for the awesome company of elite academicians and all participants. It was indeed a privilege and an honor to be a participant tonight with this amazing platform, no matter where we are in the world. And it has actually brought me back to my roots in India. And it also emphasizes, thank you for Dr. Shyam Kumar, who emphasized the role of and who has explained us the blotting techniques, and to also the emphasis, the protein and the complex mixtures of the tissue preparation and the specificity of the binding aspects. So this will also give a lot of insight and also inspiration to the young botanists and the environmentalists of participants of tonight. And secondly, I would like to thank Dr. Abdullah Muhammad Sohail. This is my second lecture. I was witnessing you, sir. Very inspirational, as you always do. And the problems that the plant-based medicines, the production of them, and the field production of cultivated level, the contamination caused by fungi and other bacteria, trying to bring the manufacture, the drying and the storage, the difficulties and the challenges that the medicinal plants or the people involved in trying to build the resource through medicines face are being well explained. And thank you for your sharing your knowledge and wisdom with us tonight. And Madam Anjana, good. Thank you, Ms. Anjana Singh, for you eloquently spoken about the symbiotic effect and mycorrhizae of plants, and also the past post-harvest. And Dr. Chan. And sorry, and the applications and how the applications of mycorrhizae increases the nutrients and all the elements that we try to complement the living world with nitrogen, potassium, calcium, etc., which are part and parcel of dietary requirements, and the increase in the diversity of the plants through the uniform seedling and the benefits of fungi. Thank you for sharing that, Ms. Anjana Singh. And then thank you, Dr. Chandrakanta Mahendranath, for your sharing of the post-harvest losses that and fruits and vegetables on perishable commodity. And thank you for bringing us insight into the variations in water temperature, and the way we have to find ways and means to restore the produce that we nature has already bestowed us, and the various ways and the stages that we have to look into the harvest, the transportation, the storage, the packaging, and the marketing. Thank you for sharing your immense knowledge in this field of and your field of expertise in this area. Thank you, Dr. Amaresan, for sharing about the abiotic stresses and the strategies of the defense mechanisms adopted by plants, especially in drought, salinity, heat, and other factors that can be chilling, cold, and heavy metal influences that can impact the plants, and the care to be taken and to nourish the environment. Thank you very much, sir. Dr. Madhusudan Reddy, sir, thank you for sharing about thank you for sharing about the ex situ conservation procedures of and the vast amount of infrastructure and the amount of enthusiasm and the plant species that you are trying to restore, and the water beds that you have created, and the wonders of the Victorian leaves that you have created. It is really amazing to see, and thank you. Hopefully many scientists will be inspired and try to keep continuing the same energy and enthusiasm that you have induced in us tonight. And thank you, Ms. Thank you, Mr. Maqbul Ahmad. Maqbul Ahmad, sir, thank you for the introducing us to the delightful world of mushrooms and the myriad varieties of mushrooms, which are edible, medicinal, and have a fascinating role of to play in our dietary requirements too. Thank you very much, sir. Madhusudan Reddy, sir, thank you for showing us about the ex situ endemic and plant diversity and human well-being are interconnected, and how you have explained us and shown us, taken us to this to the flora and the unique vegetation that you have created, and the vermi compost and the arboretum and the different species, aquatics, 50 aquatic species, and the cycle as the project that you have. This really has been an eye-opener for many of the botanists and all the participants tonight. Awareness and human response to the transformation is an aspect that will keep going. I guess that is the area that you have inspired or touched all the environmentalists tonight. Uma Maheswara, sir, thank you for sharing about the plants for a living planet. Your passion is really insurmountable. And then we have seen the plantation campaign, the environmental conscious to be aroused in the young minds, and to make them ambitious and to be participant of such innovative programs and be part and parcel of the nurturing and also taking care of the nature around us. So thank you, sir, for that. Last but not least, Dr. Ahmad Abdul Halim Khan, sir, thank you for introducing to about the council actions and the various activities it is involved with. Thank you for stating all the publications and the need to be take lead in preserving the earth and also find sustainable developmental solutions and trying to be a world global citizen to as an environmental wizard. Thank you so much, sir.
Thank you, ma'am. I would like to introduce ma'am. Actually, Indira madam is a colleague for me in 2001 and 2002. He is presently a faculty member in one of the College of Education in New Zealand. So this was Indira madam who was summing up the entire proceedings of the international conference in virtual mode. She was from her summing up, we can come to know that how curious she was, and she was very patient since the initiative of the conference till the ending. So with a with these words, I appreciate her efforts and summarization of the complete conference. So we feel a bit sorry that we couldn't have our president of Council for Green Revolution, Srimati Leela Lakshma Reddy Garu, and we equally express her great heart and kindness, kindness. So with these few words, I would like to request one of our students who is very curious to give her feedback. So after this student feedback, we'll be moving for the vote of thanks. And I request Deepak sir to the student to join for the feedback.
Yeah, sir. First, I request Manasi from final year to share her views about the conference.
Yes, sir. Good afternoon. Sir, am I audible?
Yeah. You can talk, Manasi.
Yes, sir. It was a pleasure to be a part of this conference. I feel.
Make your video on.
Yes, sir.
Yes, you can speak now.
Yes. It was a pleasure to be a part of this conference, and I feel lucky to be able to listen to such distinguished speakers, which normally wouldn't have been possible. Through this, I was able to gain knowledge from various fields such as microbiology, biotechnology, etc. I was also able to know about the research field and hope many students have been inspired to contribute to it. I got to know in detail about various blotting techniques and their applications. The importance of microorganisms in maintaining ecology was clearly explained. And also, a very important fact was put out that microbial threads no political or geographical barriers, and a holistic approach is needed to conduct research by all the nations to overcome them. In the lecture on symbiotic effect on mycorrhizae on plants by Dr. Anjana Singh, she pointed out their role in maintaining plant ecology. Though small in size, their impact on ecosystem is huge. In the post-harvest loss of fruits and vegetables in developing countries, ma'am threw light on a significant factor often ignored in agriculture sector, and also suggested possible solutions. Lots of difference between developed and developing countries have been observed during like about the post harvest loss. We got to know about the shortcomings and also possible solutions to overcome them. The world of mushrooms. This lecture was very interesting. Through this, I got to know about various kinds of mushrooms such as bird's nest, lion's mane, lady's skirt, cauliflower. They are not only attractive to the eyes, but also very potent that they can poison us. And also, it was very interesting to know that it is only crop containing vitamin D. The lecture on ex situ conservation of endemic plants of Eastern Ghats was also very informative. And also, I was very inspired and happy to know about important initiatives taken by the Council for Green Revolution, such as the Vanasankalpa, about the 10 million tree planting campaign. And it is true that without plants, no space exploration would be meaningful. So there is a great need for important steps to be taken to conserve them. Overall, this conference was very enlightening and insightful. And I would like to thank our college management and also the Council for Green Revolution and Department of Botany of Telangana University for arranging such a informative program. And it was very helpful for students like me to improve our knowledge and also igniting interest in the field of research. I really look forward for such conferences to be conducted more frequently. Thank you.
Thank you, Manasi. Now I request Deepthi to share her views. Deepthi.
Good afternoon, everyone. Am I audible?
Yeah, you are audible. On your video, Deepthi.
Yes, sir. Sir, it is not allowing.
Okay, you can speak.
Okay, sir. So first of all, I would like to thank the Department of Botany, Giriraj College, and Department of Botany, Telangana University, and Council for Green Revolution for this wonderful opportunity to learn and explore new things and many presentations that are very useful for academics and also uh, in future studies. Uh, Also, I found this seminar very useful and informative. I can say all the speakers kept this seminar lively. Especially thank Dr. Madhusudan Reddy Garu and Umameshwar Reddy Garu for inspiring me with their presentations about plants and biodiversity. Also, blotting techniques and mushrooms, they are useful in academics too. And further researchers, if any students wants to go for research, and thank you for this opportunity, sir.
Thank you, Deepthi. Now I request Vishnu Priya, who is present right now beside me. She wants to share. She is standing since a long time here to share her view. Vishnu Priya.
Good afternoon, everybody. I'm MK Vishnu Priya, studying in degree first year BSc Biotechnology from Giriraj Government College. As I have joined this year in my undergraduate program, this is my first international conference that I am attending. I am very glad to say that these presentations of resource persons were very simple and clear to understand. So I am thankful to my lecturers and also Council of Green Revolution and Department of Botany, Telangana University for organizing this international conference by inviting eminent resource persons from different parts of the world. Thank you.
Thank you, Vishnu Priya. Sir, over to Alim Khan, sir.
Okay, thank you, students, and thank you all the people. Yashwant, can you make yourself your video on? Yashwant and Venkat, both of you. Can you share the screen, Venkat and Yashwant? You're not visible, Yashwant, though you have made yourself video on. Please, you come onto the screen so that when we are speaking, you should be visible on the screen. Venkat is not
share
there.
Your screen.
Share yourself, Yashwant.
Hello, sir.
Yes. Okay. Now I will speak, and I think we are not. You are not close to Venkat. Venkat is not here.
No, sir. No, sir.
So it is a. Yeah, he's ringing me. I'll speak to him later on. So it was a great job what has been done from the side of Council for Green Revolution. It was really a splendid job what has been done since morning. Not only morning, but I think we since last 2-3 days we were keep on consultation with each other. We were trying to make this event all this colorful. So people who have joined us from US to New Zealand, so we can see that from one end of the world to the other end of the world, we could cover. Sure, so it was on the job. I think we should give a big round of applause to these people, Venkat and the person who is visible on the screen, Yashwant. So they are very cordial and they are very, helpful. And the entire credit goes to these people who made this technical program very green and very colorful. I should express my deep sense of gratitude to not only the technical team, but also our resource persons who were from different corners of the world. Really, we could make the thing world as a global village. We can see that a person like Dr. Shyam Kumar Bhandari from US and Mrs. Indira Satyanarayana is from New Zealand. So we can see that how vast distance in short span we could cover and make this event a worldwide or a global event. So a big, huge round of applause for the technical support given by Yashwant and Venkat. Venkat was the person who was really enthusiastic, and he was, I'm going to say, we can say sculpture of all this meeting. So Venkat was keep on in touch, and we could make this event. Without you, it is impossible, Venkat. And technically, without Yashwant also, it is we can't expect or we can't dream. So now coming to the speakers, first I would like to express my heartfelt thanks to our beloved student and alumni of Giriraj Government College, Dr. Shyam Kumar Bhandari, for readily accepting his invited talk in the international conference in virtual mode. And I was very specific to Dr. Shyam's speech, stating that it should be a very simple and technically sound kind of concept, which he could make very simple and very sweet presentation. And then next day we could go to the Bangladesh, very far distance from US to Bangladesh we covered, and soon we got a green signal from Professor Abdullah Mohammad Shohel. Professor Abdullah Mohammad Shohel sir has given a beautiful presentation of the microbes and how it could be very useful in making the world green and clean. And then I would also express my deep sense of gratitude towards Professor Anjana Singh ma'am. So it was the first time I could witness live in our meeting Professor Anjana Singh ma'am. So Anjana Singh ma'am has made this complete event very, very, very beautiful with her work or her briefing on Mycorrhizae. So Mycorrhizae, she covered so many facts that how we can characterize it, how can we isolate it, how can we apply it. So various concepts of Mycorrhizae were made very, very clear from Anjana Singh ma'am. So again, the Department of Botany from Telangana University and Giriraj Government College, both of them express our deep sense of gratitude towards your presentation, ma'am. Thank you so much. And next coming up, the most beautiful presentation of the times, I think, which is very important need of the day, that is the loss of fruits and vegetables. Loss of fruits and vegetables is the important aspect what we can see nowadays. This is the fact which is not being taken serious. So it is a time for us to make the fruits and vegetables. Production is one part where we are very rich in production of in whatever season comes, we are getting the fruits and vegetables. But the thing is, we are not very serious about its post-harvest concepts. What concepts are there? Such a beautiful explanation given by Dr. Chandrakanta Mahendranathan from Eastern University, Sri Lanka. Ma'am, we express deep sense of gratitude from Department of Botany, Telangana University, Council for Green Revolution, as well as Department of Botany, Giriraj Government College. Facing towards the national kind of speakers, we had our beloved speaker, very lovable person, that is none other than Dr. Natarajan Amaresan. He's from Ukkatarsadya University from Gujarat, Bardoli from Gujarat. He was so humble. He covered so much about the PGPR, we can say plant growth promoting microorganisms, and how can we make the sustainable agriculture, or how the stress can be ameliorated. That and all he briefly and beautifully described to us. Following that, we have Dr. Mahul Ahmed sir, who is very humble and who has given us a very practical food which is needed to be made in a diet, that is mushroom. So we also express our deep sense of gratitude and thanks to Mahul Ahmed sir. And followed by that, we have two our own speakers, that is Dr. Madhusudan Reddy sir and Uma Maheshwar Reddy sir. So Madhusudan Reddy sir, I saw in his CV, he was a PhD from plant physiology. So from plant physiology, he turned into an environmentalist, taxonomist, and whatnot, a conservationist. So it was a great journey of our speaker, Madhusudan Reddy sir, who made the Eastern Ghats to be live in the Yogi Vemana University. Now we can call synonymous that Yogi Vemana University is a conservation center of the Eastern Ghats. What is now lost property of nature. Really, we lost the property of nature, that is Eastern Ghats, and it was he who shouldered and made the Eastern Ghats to look in a small area that is called Lead Botanical Garden by Dr. Madhusudana Reddy. His decade of journey with the plants and making it a conservation center and also the distribution center of plants to the local and the other states. So we proud you, sir. You did a very big job, very big challenge you have accepted, and you did it. And you will be making many more such kind of events and make us really realize how plants can be conserved. And you are the one who will be the pathfinder of the day. And I really thank Umaveshwardi sir, because he has covered a vast knowledge, a very big knowledge. So here I have pinned him. You can see him again, those who missed at that moment. So Reddy sir has given a beautiful insight of plants for a planet. And really, sir, it was very inspiring that you have covered not only local, but also international. Who are the personalities behind making the world green? Not only people are deforesting, but also there are people who are making forest again. They are making the artificial habitats and make us to get more and more oxygen. Really, sir, you are a hero of today's international conference. We could admire you. We feel that though it was a very delay, actually, as per our schedule, when Yashwant, Venkat and myself planned, it was at 1 o'clock expected to sum up the things, but these 3:37, still we are continuing our session, and we have more than half century people in our group of participants in Zoom and YouTube. I don't know about YouTube, but in Zoom we have half century people are listening to us. So it was a great gesture, sir. Umaveshwari sir, we will be keeping in touch with you, and we want you to be in our future meetings also, and your initiatives and the Council for Green Revolution achievements. We will see much more updated profile of our council. So I also express, it is injustice, I will express my deep sense of heartfelt gratitude towards you, sir, for giving us awakening on the environment and ecosystem. So really, sir, you created a wonder today and made us to realize the environment is really, we should alarm now and need to protect it. So with this few words of vote of thanks, now I think everybody is able to see Venkatesh. So I was worried when the moment when Venkatesh couldn't be found in this gallery. So now I am very happy to see Venkatesh also. See Venkatesh, you have again switched off your camera here. So I was appreciating you. So Venkatesh is the man who was very readily accepting. This time I rightly posted one request to Leela ma'am. So I don't know, Leela ma'am couldn't join us. Still we miss you, Leela ma'am. So in future, whenever we are going to collaborate again, I think Leela ma'am will be there with us in this gallery of you. So because she was the lady behind all this CGR and making this all kind of virtual events. So it is a big thanks for the technical team, and special thanks to Venkat, special thanks to Yashwant, and special thanks to all.
Thank you, sir. Thank you, sir. Thank you, sir.
Thank you very much. And now the conference is almost to the end, and I request the Giraj College people to start the national anthem, and so that we can end the meeting.
Students. Mic. You also come.
Start.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता Punjab, Sindhu, Gujarat, Maratha, Dravida, Utkala, Banga, Vindhya, Himachala, Yamuna, Ganga, Uchala, Jaladi, Taranga. Tava Shubha Name Jage, Tava Shubha Shisha Mage, Jage Tava Jaya Gata, Janagana Mangala Dayaka Jaya He Bharata Bhagya Vidata, Jaya He, Jaya He, Jaya He, Jaya 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 He.
Jai Hind.
Jai Hind. Congratulations, all of you. And Yashwant, now we can